It's probably both. To present for face on. Cool. Okay.
Can one of you make me co-host, please?
On it. Done.
Cool. I'm just going to pause the recording. We're still streaming, but now we won't have as big of a file.
Okay, perfect.
So we'll just restart that when we're ready to go.
Okay. Good morning, everybody. Welcome to day 2 of the California Aquatic Bioassessment Workgroup meeting, CABW, and the California Chapter Society for Freshwater Science meeting. For those that weren't with us yesterday, my name is Allie Dunn, and I work at the State Water Resources Control Board and oversee the Surface Water Ambient Monitoring Program, or SWAMP. And the Bioassessment Monitoring Program is one of the programs that I oversee. I'm also joined by Angela DePalma Dow, and we'll be handing things off to her in a second here. But Angela is the president of the California chapter of CalSFS. I do have just a few kind of logistics to go through again for the second day, and then Angela's got some exciting news for the poster sessions that were held yesterday, and we'll talk a little bit about the workshop that's going to be held this afternoon. Okay, so the fun, exciting stuff. We're all virtual again, clearly, as you all could tell, this is not in person. We have the chat and video and audio disabled for attendees. However, you can use the Q&A icon. It should be under your chat controls. If you have any questions about that, you can reach out to that SWAMP email there. But use that Q&A button to submit questions. Following the presentations, we'll have a few minutes for questions there. And then we have a panel today too, so there'll be a lot of great discussion. Use that Q&A button, and you can ask questions while the speaker is still presenting, but we'll get to those at that 5-minute discussion. If you have any logistical or technical issues, please email the SWAMP email, which is swamp@waterboards.ca.gov. We'll also put that in the chat if it's not already there. But that's, if you're having Zoom issues, whatever it may be, please feel free to reach out to us, and we'll do our best to get back to you as quickly as possible. And then the last thing I just want to say on this front is we are doing our best in this virtual world, and issues do happen, technical things do happen. So please be patient with us. Yesterday went pretty smoothly. Hopefully I'm not jinxing myself by saying that for today, but hopefully it all goes well today as well. All right. So for this second day, we have some really great talks lined up for that 4th session of CABW, everything from looking at macroinvertebrates and some more holistic monitoring efforts down into different monitoring methods, looking at eDNA, and we'll talk about nutrients and cyanobacteria and all kinds of great things. We'll take a break then around 10:50, 11 o'clock, come back for that estuary panel. Again, some really great talks focused on estuaries. We're starting to do a lot of more work in that way with the bioassessment program statewide at SWAMP and moving out of wadable streams and into more salty zones. So this should be a pretty interesting discussion. And then we'll break for lunch, and I won't go into the details here, but really great workshop after lunch that CalSFS is hosting with the TEK panel, and great discussion there. And if you didn't get a chance to check out those posters, they're still up. The Zoom Rooms meeting link is in the agenda. So if you want to pop over there and check those out, feel free to. And you can do that anytime, but we just carved out some space for you if you're going to be here all day for the talks and workshop. And then we'll wrap things up about 4 o'clock. And with that, I'm going to hand it off to you, Angela.
Thanks, Allie. I appreciate that. How are you doing this morning?
Great. How are
Good.
you?
So I'll just make one correction, because the posters, we actually don't have the Zoom room up anymore, but they're available on our website. So I just put the link to the CalSFS website in our in the chat for everyone to see. And you can see the posters also from last year's CalSFS as well as this year. This year's posters were spectacular. We had 10 posters and 9 were in the student competition. You guys did a great job. All the students really knocked it out of the park. Some great scope of topics and great information, and the technical and the visualizations for these posters are just amazing. So everybody did really good. The poster session, when we do a student competition, is not possible without judges. We need to be able to have judges be able to evaluate posters and provide feedback for the students. So I just really wanted to thank John Olson, Kyle Leathers, Alyssa, Jasmine Shen, she won from last year's poster or lightning talk competition. Anna Holder, thank you so much. Ryan Carey, thank you to all the judges for help making this possible. It was really appreciated. And we did have some poster winners. You can go to the next slide. Who's ever controlling that? Yay. Okay. So we actually had a tie for first place this year. So Alyssa Scher with freshwater invertebrate composition depends on river salinity, and Morgan Clark's poster intersects condition of largemouth bass in an effluent dominated river. So both of those tied for first place, and they each won $100 for this poster competition prize, which is made possible through support and through CalSFS, through the members, donations from members and non-members, as well as some support from the Parent Society of Freshwater Science. And then second place, we had $50 go to Melissa von Meyerhauser, with her poster From Stoneflies to Spiders, integrating aquatic and terrestrial invertebrate monitoring to understand the effects of effluent on stream ecosystems in Southern California. So yay, big round of applause to all the posters and the winners here. Thank you guys so much. Did a great job. And yeah, those posters are still up on the website if you want to check them out. And they'll basically be there forever. We're trying to keep tabs on all the work done through CalSFS. And so we just really appreciate all those that participated, both that showed up to the poster session and those students that participated in that. And this is a hard time of year to get together all your posters and present, because it's the end of season. Some are still doing field work and the semester has just started for them. Some are teaching, some are attending a lot of classes. So we really understand that this is a crux time in a student's academic career, and to be able to come out and do a poster, really appreciate it. And it really helps to further the society as well as our partnerships with CABW. So thank you so much. Next slide. You made the stars teeter. Okay, there we go. Thanks. Okay, so today 1 to 4, we do have our traditional ecological knowledge session and panel. And these are our speakers here on the left. So we're going to be having a presentation from Sarah Ryan from Big Valley, Keith Parker from the Yurok Tribe, Anne Marie Mendoza from UCLA, Dr. Beth Rose Middleton and Ada Stevenson will be doing a joint presentation. And Dr. Beth is from UC Davis and Ada is from California Indian Water Commission. And we really have a unique opportunity this year because we're going to have the first part will be the special session. We'll get to hear from each of the panelists, each of the speakers on projects they're working on now, things that are going on in their systems. And then we'll have the question and answer panel will be after that. And that'll be moderated by this cohort, this CalSFS cohort of fellows. So this is our first year doing a fellowship program, and the fellows give back to the society. And this is one of the ways, by participating and moderating this panel. And they're all interested in learning about TEK and facilitating TEK knowledge across the membership. And so this is part of the ways that they can do that. And so these are our fellows here. We have Robin, we have William, we have Kyle, and we have Melissa, and they're going to be moderating this panel today. And we do have another fellowship opportunity coming up. So if anybody's interested in being a fellow for CalSFS, you want to pay attention to our website. About around January, I think, is when we'll open up the next cohort, the next application. So if anybody's interested, make sure to pay attention to our website. You can always reach out to me, and we have our email too coming up here in the next slide, or next few slides. I'll have it posted. And we will be posting this on social media. We have a Twitter account at CalSFS Social throughout the day as well. All right, so if you're wondering about what CalSFS is, we really are a local chapter of SFS, which is an international organization now. So getting to the local chapter gives you really more in-depth networking, professional growth opportunities at a local level. We are fellows. We've been able to match them up with mentors at the State Water Board through opportunities like this, working with CABW, as well as early in the year, we were at the Data Water Science Symposium. We were able to provide information exchange. And then, of course, any of the members can have direct input about local events or outreach opportunities. So if you see that there's a need in your local community or academic clubs for more interaction that would be beneficial with Society of Freshwater Science, being part of CalSFS will really help you do that and give you some resources needed to be able to do that. There's also lots of opportunities provided for students in early careers. And even if it's not something that comes from the chapter itself, it can be facilitated or coordinated with the parent society, as well as with local chapter too. We try to support our local students in early careers as much as we can. If you're interested in joining, there's two ways to do that. So the first way is really easy. If you are just renewing your membership to Society of Freshwater Science, when you're going through the renew process, there's a little box that tells you, gives you options for chapters, and you want to check off California chapter. Make sure you do that. That's really the only time that you can get your California chapter included in with your Society of Freshwater Science parent renewal. If you want to join, you've already joined, you forgot to do that, or you just want to join the chapter, you can email us down here at cal.chap.sfs@gmail.com, and then we'll be able to get you set up as a member today. Next slide. I think I have like one more, because I know we want to get to the session. Okay. Last thing, and this is my groveling, but if you want to support CalSFS, you can donate to us directly, and just know that your donations provide for student travel awards, student early career research support. We have some members that do research and they just can't afford publication fees, so we can help them with that. As well as, there's some students that really want to be a member, but they can't afford the membership either, or they don't have the resources through their institution. And so we can help with them too. So it's really about growing this membership and growing these opportunities for all the members and for any freshwater scientists here in California. If you'd like to donate directly, you can go through our Venmo, which is down here. It's at CalSFS-John Olson. He's our treasurer. So it's not just some random dude. He's actually our treasurer and he's great. You could also email him if you want to figure out another way to donate via check. And his email is right here, joolson@csumb.edu. And so we appreciate any donations. And the fellows are here today and able to contribute to the society and develop their careers with SFS through donations made possible. So we thank so much for all of that. I think that's it. Do I have another slide? I think I have one more slide really fast. Yeah, okay. So thank you again to Swamp and Waterboard. Thanks to the team for having us. We're so excited for day 2 of the CABW CalSFS. And next year will be our 10th anniversary for CalSFS meeting. So we have hopefully we'll have some really cool, fun stuff planned and we'll be able to meet in person. And again, thank you so much for letting us be a part of this meeting. And I hope everybody has a great day too. And we'll see you this afternoon at 1 for the TEK session and panel. Thanks so much, Ali. Back to you.
Thank you, Angela. And yeah, as Angela mentioned, the CalSFS is such an awesome organization. The fellows presented at this year's Data Science Symposium, and everyone was totally blown away with their work. And we're not surprised we get to work with you guys, but just some really exciting work that's happening throughout the state. So great organization to support and join if you are available to do that. Okay, so, and I'm thinking it's too bad it's on our 30th anniversary next year, because we could really do some cool 30-10.
They're off by 1 year, but you know what that means? We get to party for the 10th year for CalSFS, and then we get to party the next year for the 30th CABW. So
that's smart.
2 years in a row.
Totally. All right. So our 4th session then, and our first session of today, we've got some really great talks lined up. I am going to hand it off to Carly Nilsson with the State Water Board to moderate this session. Before I do that though, Carly, I wanted to give you a proper introduction. So Carly has her BS in biology and has been a researcher, consultant, and fellow for water quality protection. She has worked for the water boards for almost 15 years, most recently taking on a new position at the State Water Board in our office in AWEMA as the co-lead for the freshwater and estuarine harmful algal bloom program. And with that, Carly, I'm gonna hand off to you.
Okay. Hi, everyone. I'm really excited to be here. we're doing session 4 today, and so we have 4 awesome speakers. So settle in, get your coffee, get comfortable. I think today it's going to be kind of a unique, different kind of approach. We're going to talk a little bit about endangered and threatened species, some fish, and we're going to talk about some algae, which is right up my alley. So with that, I'm going to introduce our first speaker. Gary Chin is a second-year PhD student in evolution, ecology, and organismal biology at UC Riverside, advised by Dr. Helen Reagan and Dr. Kurt Anderson. His research interests are in freshwater ecology, ecosystem health, and ecosystem services. His current dissertation work is on evaluating the ecological sustainability and governance structure of the Santa Ana River watershed in Southern California. Specifically, he's focused on studying the current and future ability of the watershed to deliver ecosystem services, sustainability, and equitably. In his free time, he enjoys cooking, exercising, and attending music festivals. So I'm going to hand it over to you, Gary. Don't forget, guys, that you can send in your questions during the presentation in the Q&A. With that, let's send it to you.
Okay, all right. Give me 1 second. I'm just going to test out my remote control, see if it works. Oh, no. Oh, okay, all right. All right, I guess I'll start then. Well, hello, everyone. My name is Gary Chin, and I'm a second-year PhD student, as Angela said, in evolution, ecology, and organismal biology at UC Riverside. And today I will be talking about an assessment of threats to endangered and threatened freshwater species in California that I completed with members of my lab. And for the rest of this presentation, I'll be shortening endangered and threatened to ENT. So to begin, freshwater ecosystems supports high biodiversity through its variety of floodplain flow regimes. When we look at freshwater ecosystems, it's not just the free-flowing stream, but also the adjacent areas that are flooded at varying timescales. So looking at this figure from Poff et al., we can imagine that the riparian vegetation near the stream gets flooded regularly, perhaps annually, while the areas further away from the stream may only get flooded once every decade or century. And this wide range of flow regimes that exist in freshwater systems in turn supports a wide range of species and populations. Freshwater ecosystems contain as much as 12% of all known species, including a third of all vertebrate species, and approximately 40% of global fish biodiversity. And so a reason for this is the fact that this small amount of water in the world is not only responsible for sustaining wildlife, but also for sustaining human activities such as irrigation and municipal and industrial consumption. And this intense human demand for water and its associated stressors ranging from agricultural runoff and flow regime modification from dams to oil spills affecting wetlands puts a huge strain on such a limited and dwindling resource. Yet, as we all know, considerably less attention and research is put on freshwater systems, despite water insecurity currently impacting more than 2.5 billion people. And by the year 2030, human water demand is expected to exceed reliable supply by 40%. And just to reiterate the fact that freshwater ecosystems are biodiversity hotspots that are disproportionately affected. So when we look at this graphic from Reed et al. 2019, it shows the trend over time of the Worldwide Fund for Nature's Living Planet Index, which measures vertebrate population trend data for the freshwater, terrestrial, and marine realms. And here we can see that there is a remarkable index decrease in freshwater species relative to terrestrial and marine species within the last 20 or so years. And here you can see this graphic. So Dudgeon et al. Provides us with a useful framework in thinking about freshwater biodiversity threat. So what this figure shows is that we can categorize the most imminent threats to freshwater biodiversity into 5 categories, which are overexploitation, water pollution, habitat degradation, species invasion, and flow modification. And then you'll see all these arrows that connect those 5 threats. And that suggests that these 5 threats are strongly linked with each other. And therefore, conservation efforts have to balance and attempt to address all of these issues simultaneously. So only finding solutions to 1 or 2 of these threats at a time is inadequate in addressing the larger freshwater biodiversity crisis. And just by looking at the very nature of these threats, it really highlights the fact that freshwater is a resource that humans constantly extract, divert, contain, and contaminate in ways that compromises its ability to support its immense biodiversity. And in all the places where freshwater systems are threatened, it is especially precarious in Mediterranean climates. So outside the Mediterranean Sea region, Mediterranean climates can be found in Southwest and South Australia, the Western Cape of South Africa, Central Chile, and California and Northern Baja California. And with Mediterranean climates, they are characterized by highly seasonal and interannual variation in runoff and flow with long periods of aridity and isolation. Given these conditions, Mediterranean climates suffer from extensive land use change related to agriculture and urbanization, as well as extensive hydrological modification in the form of dams and reservoirs to protect human settlements against the flooding and drought characteristic to Mediterranean climates. For my research, we decided to focus on the state of freshwater biodiversity here in California. And with California is one of the few places where 5 climate types occur in close proximity. The desert, cool interior, highland, and steppe climates border a small region of Mediterranean climate and all contribute to the diverse biome in the state. The mild and winter rain of the Mediterranean climate in particular supports some of the highest species richest in the state. And these conditions also coincide with the highest human population density out of the 5 Mediterranean regions, along with high rates of endemism and trends associated with high freshwater biodiversity threat, such as high human water demand, massive urban growth, and extensive agricultural industry. And therefore, when we look at California, it really provides a useful case study for other places that are currently or will undergo intensifying freshwater biodiversity threat. And within California, there are 157 federally and state listed endangered and threatened, or ENT, animal species. Some examples of these species include the giant kangaroo rat, the San Bruno elfin butterfly, and the tidewater goby. Regarding these 157 species, we focused on 3 key research questions. So number 1, first, how many of the 157 species are freshwater species, meaning they rely on freshwater hydrological processes to live? Second, which are the most threatened taxonomic groups within those 157 species? And finally, third, what is the relative prominence of different threats across different habitat types? To answer those questions, we searched through online resources like the IUCN Red List website, NatureServe, and the California Department of Fish and Wildlife website to fill out an evaluation sheet for each of those species. So here is an example evaluation sheet for the San Bernardino kangaroo rat. At the very top, you'll see the at-risk classification. So FE for federally endangered, PCE for proposed state endangered, and so on. Below that, you'll see its risk factors, which we identified and evaluated using the IUCN Red List threat classification scheme. So on the very first column, you'll see the specific threat category. And then next to that is the degree of threat and spatial scale, both rated as high, moderate, and low. After that is the temporal scale of the threat, whether it is short or long term. And finally, a specific description of the threat. So for example, if you look at the very first row of the risk factors, you'll see that we found that the kangaroo rat is affected by natural system modification, specifically flood control structures and operations and groundwater pumping. And we rated it as a long-term high-level threat throughout much of its range. And under those risk factors, you'll see that there's information about the species habitat association, spatial scale, functional group, generation time, and lifespan, if any of those are available. And once we completed evaluation sheets for each of those species, we aggregated and analyzed the information and had these key findings. Regarding the first question of how many of the 157 species are freshwater species, which again rely on freshwater hydrological processes to live, we found that most ENT species in the state are freshwater species at 64.4%, compared to terrestrial at 28% and marine at 7.6%. And then on to the second question. So what are the most threatened taxonomic groups among the 157 species? So here's a bar chart with the taxonomic groups on the y-axis and the number of species on the x-axis. You'll see that the most threatened taxonomic group is fish, followed by mammals and birds. And given that we all know that freshwater species are the most threatened relative to terrestrial and marine species, and that all the fish species that we assess are freshwater, then this isn't really too surprising that fish are the most threatened. However, it's also important to be mindful of the fact that there may be some taxonomic bias in which species are actually listed on federal and state ENT species lists. We can also break down the freshwater, terrestrial, and marine habitat categories in this figure into specific floodplains. So in this iteration of the figure, the solid colors correspond to freshwater habitats, while the textured colors correspond to non-freshwater habitats. And some things that I noticed from this figure are that a sizable portion of the bird species are riparian, while a majority of the amphibians are aquatic, while a majority of the other invertebrate category, which leaves out insects, live in vernal pools. And then finally, moving on to the last question. So what is the relative prominence of different threats across different habitat types? And here is another bar chart with every assessed threat category on the y-axis with the number of species on the x-axis. So freshwater species represented here in light blue make up a majority of the species affected in nearly all the threats compared to terrestrial and marine species. And what's interesting here is that while altered hydrology through dams, reservoirs, and levees is the only threat directly linked to freshwater, freshwater species are affected by every single assessed threat. The top three threats across all habitat types are habitat loss, invasive species, and climate change. However, when we look at just freshwater species, the top three threats are habitat loss, altered hydrology, and invasive species. And these threats, along with pollution and overexploitation, form the basis of the framework on freshwater biodiversity threat by Degenet et al. That I showed you earlier in this presentation. And again, we can break down the freshwater, terrestrial, and marine habitats into specific floodplains in this figure as well. In this iteration of the figure, we can see that while species living in freshwater aquatic habitats, represented in light blue here again, make up a sizable portion of the species affected in most of the threats, the species represented by the other freshwater associated habitats like riparian vegetation in yellow and vernal pools in red make up a sizable portion of the species affected as well. So just to recap, the key findings from our research are that first, most ENT species in California are freshwater species. Second, the most threatened taxonomic groups are fish, followed by mammals and birds. And finally, threats are most prominent in freshwater species when we compare it to terrestrial and marine species. And seeing the extent of threats to freshwater species in California highlights the need to find solutions to address multiple threats simultaneously, as Dudgeon et al. Suggests, and manage the trade-offs between ecological and societal needs, ensuring that there continues to be an equitable and sustainable delivery of freshwater ecosystem services. Future work should consider the spatial extent of threats to endangered and threatened freshwater species within California. Study whether geographic location and regional water allocation are significant factors in assessing threat to freshwater species. Third, conduct a similar analysis of threats to ENT plant species in California. And finally, conduct similar analyses of ENT freshwater species in other states so that we can develop a broader sense of the spatial, ecological, and societal extent of freshwater biodiversity threat. And with that, I'd like to acknowledge my PhD advisors, Dr. Helen Regan and Dr. Kurt Anderson, and our lab's former master's student, Anna, and former undergraduate researchers Leo and Eamon, who collaborated with me on this research project. In addition, I'd like to thank my department faculty and fellow graduate students for their constant help and support. And finally, I'd like to thank you all for listening. I'll take questions now.
Okay, thank you so much for that really great presentation, Gary. For all of you guys, please submit your questions for the Q&A. We do already have a question. How does aerial spraying rate as a threat for West Nile carrying culex using BTI or the OP Temphophos, for example?
What was that species again? So I actually have the spreadsheet of all the species that I looked at, so I can actually see specifically for that species if we looked at it or what the threat is.
Temphophos.
Could you type it in the chat just so I can?
Yeah.
Thank you.
And let's see.
It doesn't appear that we looked at that particular species in our analysis.
Okay. We have another question. We'll comment first. Great presentation. Just curious if you know what types of water pollution pose the greatest threat to ENT species?
For ENT species, from what we could tell, a significant source of the pollution within the state are pesticides and rodenticides. So particularly within more rural areas that are not necessarily under the purview of state and federal regulations when it comes to pollution within the state. So for example, like one threat that is emerging within the state is illicit agriculture. So specifically for cannabis and marijuana, that affects some more inland streams where there's unregulated use of pesticides and rodenticides that are getting into the water and affecting species that would normally be unaffected by pollution.
Okay. I had a clarification that it's culex as in the mosquitoes for the species. So I don't know if you want to check that really quick for me, Gary.
Oh yeah, sure. So just to clarify, the species that we looked at within this analysis are either listed as threatened and endangered in the federal list and the California list. So when I'm looking at the insects that we assess, they consist primarily of beetles and butterflies, but no mosquitoes.
Okay, excellent. I don't see any more questions coming in. I had a quick question for you. Do you know if this kind of research, similar research is going on anywhere else in the world in terms of these Mediterranean climates, and if they're kind of coming up with some of the same answers that you are in terms of what is most affected?
We're not aware of any similar analyses of endangered and threatened freshwater species in the other Mediterranean climates, but we mostly chose this to focus on California, given that we do live here. So like there is some relevance to our own daily lives, but also the fact that there is just so much information available that might not be available in the other Mediterranean climates. So again, like we had access to the IUCN Red List NatureServe, as well as the California Department of Fish and Wildlife website, which in itself was so helpful in getting the information that we needed. So, but it would be definitely interesting to look at the other Mediterranean climates as well in the future.
Okay, I think we have time for one more question. What information or paper did you use to cite pesticides and insecticides as the greatest harm to T&E species?
So this isn't coming from any specific paper, but it's just coming from our own experience reading through a lot of the reports that were coming in. So just based on our observations, pollution through pesticides and rodenticides seem to be the most common cause of pollution threat. So in particular, a lot of the reports that we were using are the 5-year plans from the US Department of Fish and Wildlife, and they'll list all the threats and then give specific narrative details about what that particular threat is. So, but it would be interesting again to actually look at what are the sources of pollution, just to give like a specific number of like how many species are affected by rodenticides versus pesticides versus some other chemical in the water.
Okay, thank you. That was an amazing presentation. I think we're at time right now. There's one more question that I think we'll send you to answer independent of this. And I think everyone can reach you by contacting the swamp email or Shuka directly. But that was awesome. Thank you so much for your time, Gary. I really appreciate it.
Okay, all right. Well, thank you so much.
And with that, I think we'll move on to our next presenter. And that was actually a great segue, especially talking about fish is one of the most threatened taxonomic groups. So now I'm going to introduce Steve Blumenshine, and he's a biology professor and director of research and development for the California Water Institute, both at CSU Fresno. His research program focuses on inland surface waters as habitats, primarily for fish. His current focus is the San Joaquin River Restoration Program, where he sustained work on juvenile Chinook salmon ecology in the system through a combination of empirical approaches, fish bioenergetics models, and coupled ecosystem simulation models. Earlier this year, he was fortunate to start a new research front on the C. Wandel Lake Change collaborative project in Lake Constance, which borders Germany, Switzerland, and Austria. And with that, I'll pass it over to Steve.
Hi, Carly, thanks. Let's see, I got to get the video started. No, I don't have it. Yeah, that was a great and important topic, Gary, and Carly's right. It set us up well here for what I want to cover. So I'm sure that many runs of Chinook salmon in California are on your list. And so what I want to cover, and I want to clarify one of the terms in my title, is macroinvertebrate diversity. So given that Gary covered some taxonomic aspects of these threatened species in California and the Mediterranean climate, our focus on macroinvertebrate diversity is really in the distribution of their abundance over space and time in juvenile Chinook salmon habitat in the San Joaquin River. So what we're focusing on, and again, a good carryover from Gary's talk, is with the San Joaquin River Restoration Program, which is a multi-year, multi-agency effort to restore this southernmost population of spring run Chinook salmon. So in this topic that I want to cover today about the San Joaquin River Restoration Project, for the material that follows, why does it matter? Well, there's goals for this restoration to meet the main objective of a sustainable population of Chinook salmon that doesn't need wholesale inputs from hatchery raised juveniles and adult broodstock, for example. And in order to meet some of those restoration goals, we need to understand what's the carrying capacity of this system And how does that vary in that juvenile rearing area over time and space? Okay, so in California, many of the historical salmon runs were quite extensive and went into foothill and mountain areas. But from this map that many of us have seen, that many of the reproductive areas and juvenile rearing habitats for Chinook and other salmonids in California are greatly truncated, such that dams preclude their return to natural spawning areas. So in the last century, that was a rather abrupt and serious habitat change and interruption to their life history needs. So what I'm showing in the bottom left picture here is an aerial photograph of Friant Dam, which creates Millerton Reservoir here. And so what's allocated after what goes down canals to feed agricultural interest is this little trickle of a river, that's the San Joaquin. And here and about 20 kilometers downriver is the current rearing area for Chinook salmon in our study system. So closer to the ground, what we could do is take a lesson from Peter Moyle and take a look at the current landscape that we have, for example, and it mentions Mediterranean climate regions. So it's the new reality for streams in California and other Mediterranean climate regions. So Gary, there's more of a connection than I initially realized. And the fact is, with restoration programs, we can't go back to what was before human alteration of hydrology, for example, in these important habitats. So we have to work with what we have. And so our focus and our approach in our role in the restoration effort is to recognize this altered hydrology, as well as prospective changes due to climate change, which are impending. And so overall, we look at the thermal experience for the fish. And that is really a thermodynamic approach, looking at energy gain through prey consumption and energy costs, say through swimming against the velocity of the river, and the metabolic energy, which are based on the temperatures of the water. So we look at this in a big picture is the scope for production. What's the potential production for juvenile salmon cohorts, given the thermal and other energy regimes in the rearing area? So let's take a look at a table from the Fisheries Management Work Plan for the San Joaquin River Restoration. And we kind of zoom on this table, which is based on HSI, or Habitat Suitability Index models. But that doesn't really jive with our approach we've used for years and years for a lot of different fish populations, in which those are based on thermodynamic principles and trophic bases for production. So for example, one of the objectives for juvenile growth rates of spring run is 0.4 grams per day for spring run or fall run, or 0.07 grams per day for those that are still there in the summer. And we've argued with them or pointed out that this metric is not really energetically based, and there's some issues with it. For example, if we consider this metric, 0.4 grams per day, well, that really matters. These small fish grow very rapidly, we find. And if we're looking at a 2-gram fish, 0.4 grams per day is 20% growth per day. That's fantastic, almost impossible. But for a 10-gram fish, that's just 4% per day. That's not bad. But if we get up, say, to a 20 or 40-gram fish, 0.4 grams per day isn't that great. And so we argue that it's not really connecting with the carrying capacity of the habitat and the scope for production. And we're asking for some consideration of some changes of the goals of the restoration program to better look at that approach. So for example, using this 0.4 grams per day growth rate as a baseline, we find from a lot of data that very few fish meet or exceed that 0.4 growth rate in grams per day, but we find the growth rates on other measures are quite good, actually. Rather, we prefer to look at the bigger picture and have an allomic approach to growth rates, such that small fish, like puppies and human babies, can grow very fast, but as they grow, their growth rates, again, based on temperature, tends to diminish. So we have an interaction between temperature and the size of the organism in assessing this scope for growth. And we use this value omega, which standardizes the growth rate across these different sizes, recognizing that very small fish have a very high capacity for fast growth, and that diminishes as the fish grow in size. So this omega value, for example, standardizes growth rate. So we have a 1-gram fish growing at 1% specific growth rate, mass per day, and everything else, bigger fish, for example, are relative to that, as well as their specific growth rates. So that standardizes that allometric interaction we have between temperature and size, for example. Okay, so our focus is on looking at the thermal environment plus prey energy. Those are both based on thermodynamic principles. So for example, we know that the thermal environment in the rearing habitat is quite variable. Areas that are blue are cool over time and space, the two X and Y axes here. But as we go further downriver over time, things get a little hot. So in our simulations of growth and the habitat, we consider this very dynamic thermal landscape. And then we have the prey energy. And if we have a lot of prey energy, fish will do okay because they could fuel that furnace that's burning pretty hot in these warmer temperature times and places, for example. So overall, we coupled in a prior iteration of our research, which is ongoing over a number of years, empirical information, collecting data like this thermal history and the macroinvertebrates with simulations using tools like bioenergetics, which is an IBM or individual-based model, and InStream, which is a habitat bioenergetics coupled model. So our overall approach, I'm not going to walk you through all this, but in our proposals, we're looking at water allocation. So we know that surface water is scarce and there's a lot of competing users for water in California. So we want to look at the efficiency of water allocation for the restoration based on bioenergetic principles to better assess juvenile Chinook habitat and their needs to meet these restoration goals. So the currency we look at are energy units like calories per grams of fish, and we use these coupled type models to predict some different scenarios. So the InStream model, for example, there's a new version 7 that we worked with Steve Railsback on that. And that has a lot of physical variables as inputs. And then finally, we get down to prey quantity here, a biological input that fuels the metabolic furnace that the fish need for growth. And with the simulations, we could estimate growth, survival, and condition of juveniles in any cohort. So I'm showing you this, it's a lot of data, but the main point here is that this thermal environment is quite variable. For example, we can't just say that we have a drought year because we know from the last big drought time that the drought cycles aren't running in high precision. They could be early or late, and they differ quite a bit from normal hydrologic cycles in the river, for example. And we keep track in our simulations of what thermal environment individual and cohorts of fish are experiencing by tracking how temperature in the river changes over the cohort days. So when they're born, approximately November 1, at different locations in the rearing area. So we kind of interpolate their thermal experience as they undergo successive days in the river and even move downriver, for example. So what we could do in our simulations is to incorporate drought years or normal hydrology. So based on the river restoration program, there's designations of water years going from normal wet to critical low. We could couple that with different baseline abundances of prey. I'm going to skip this and move on. I think it's just showing some of our empirical data showing that temperature isn't that related to fish growth. So some other factors must affect growth rate. So with that, we could take these scenarios of water habitat and prey abundance I have on the right part of the schematic, but the inputs are the prey abundances and availability, and that's the macroinvertebrate diversity part I had in the title. So we need to consider a diversity of habitat conditions and where the fish might forage. Search mode is when they look along the bottom of the river for benthic prey. Drift is most traditionally studied to assess habitat. But then there's macrophyte habitat. And there's a lot of that in this rearing area of the San Joaquin River. And that's not very common to salmon habitat, so it's really understudied. And then we have several methods to estimate growth rates. And we can couple all this information to calibrate some best fit models and undergo more scenarios of combinations of the type of water year and the amount of prey. So for example, the macroinvertebrate diversity, if we look at drift samples, which is what most people look at when they assess juvenile salmon habitat, we get lots of tiny prey like this. These are little crustaceans like copepods and cladocerans. And so that's like breadcrumbs. If you're a forager, it's not very efficient. But when we look in this understudied macrophyte habitat, oh my gosh, that's where all the big stuff is hiding. And this is a different packaging of energy that if we follow optimal foraging theory, is more energetically beneficial, we'd suggest, than foraging in this crumb-like environment here versus having a whole piece of the rice cake or bread in this type of habitat. So we're trying to figure out how to incorporate these variables and these differences in the prey among habitats in our scope for production estimates. And then just a few results that we have so far. Overall, it's showing that there is some variation in drift abundance, say in time and space. And then in the macrophyte habitat, I'm gonna have to skip to this one. We see different invertebrate abundances among different plant types, so that makes our analyses a little difficult. We have to account for degrees of freedom there. And then overall, we see a significant month and site interaction for macroinvertebrate abundance in different months and different sites along the river. So that's gonna mean that we need to chop up our simulations in more tightly spatial regimes in the river. Okay, so to assess how the fish relate to those different habitat types, we use stable isotopes and mixing models. We have data from the invertebrates. We just got data yesterday from the fish, so I can't include them here. But one thing we see in the clouds of the stable isotope signatures from carbon and nitrogen for invertebrates is that we see a nice downriver signal, which is really good because then we can kind of assess where the fish got their nitrogen and carbon along the river reach. Okay, so I'm told I have 1 minute left. So we use mixing models to look at the probability of the fish and their carbon and nitrogen and what type of prey it came from, for example. So overall, we're really focused on this fish habitat relationships from a bioenergetics point of view and the heterogeneity of the habitat and the prey energy that they contain. So we're focused on this thermal and energy source considerations and using integrative simulation-based processes and interdisciplinary groups with lots of different expertises. And so we want to take that theory and those simulation approaches coupled with the empirical data and help those to advise and guide some applied goals like Chinook salmon restoration. So finally, I want to thank a lot of people who've worked on this over the years, both agency and people in my lab group. So thanks, and I'm interested to see what questions you have.
Thank you so much, Steve. That was super interesting. We're still waiting for some questions to come in. So with that, I guess I can ask a question just kind of based on your research. Did you guys do any bioassessment on like the proper for macroinvertebrates at all to kind of couple with this, or was it just looking at the fish species?
Oh, we've spent lots and lots of effort on macroinvertebrate sampling and analysis in the lab. That's very labor intensive. Yeah. Okay.
And how would you envision kind of incorporating this kind of research into a restoration project that is specific to kind of fish habitat for some of these larger river systems?
Yeah, larger rivers are going to be a tough call. San Joaquin is big enough you could nearly walk all the way across it, but you need to consider, say, for the Sacramento River, the morphology of the habitat. So you're going to have a lot of volume per surface area, for example, with respect to the river margin and contact with any bottom habitat. And so you need to look at your prey energy sources appropriately and scale that to the morphology of the habitat, whether we have a low width, shallow river or a broader river that's very deep and high in volume.
Okay. And we're getting some other questions, so I'll read those to you. How can you test if fish are actively feeding in the macrophytes? Do you think macroinvertebrates can avoid fish predators in that habitat?
That's exactly what we're doing, and that's our new front now. So actively feeding in the macrophytes, we could do that chemically with the stable isotope signatures, but that's inferential. And just in the next couple of weeks, we're going to put out GoPro cameras to monitor fish movements, both the juvenile salmon and predators there. And then we're going to work with our agency colleagues to put in some minnow trap type systems to look at who's entering and exiting the macrophyte habitat.
Cool. And great talk. Do you have any data on macroinvertebrate production in addition to density diversity?
Hey, Kurt. Yeah, that's a great question. So, a lot of estimates of macroinvertebrate productions are like by size frequency method and things like that. So I don't have anyone in my lab dedicated to that exclusively, but maybe let's follow up with that. I'd like to hear from you and see where we could go with that, for example.
Awesome. I guess that's going to end it for the questions. Kurt says thank you. So I just want to thank you so much, Steve, for your time. It was great to hear a little bit about the fish species, because I know we've been focusing so much on the bugs the last 2 days. So I really appreciate you taking the time to kind of share a different, lens with all of that.
Thanks, Carly.
Yeah. So with that, I'm going to go ahead and introduce our third speaker, Byron Furman. I might have butchered the last name. I apologize. Byron is an aquatic technology development scientist at CPRO Corporation, and he is the Northern California director of the California Lake Management Society, commonly known as CALMS. He received his PhD in environmental systems from UC Merced, where he studied redox and oxygen control on water quality in California reservoirs. He also holds a BS in chemistry, an MBA, and an MS in environmental engineering sciences. He has previously worked as an instructor for an environmental engineering course on water quality and as the quality assurance manager at an environmental laboratory. And with that, I'm excited to introduce him because we're going to start talking about my jam, algae.
Thank you, Carly. Let me see here if I got controls. Oh, perfect. Just a little bit slow. Let me go back to my first slide. Okay, so yeah, thanks for everyone for having me. I'm really excited to talk about nutrient and mineral cycling and just how it relates to aquatic ecosystem structure. For my job, I mostly work on aquatic technology. So just kind of trying to find new solutions to water quality problems or ways to go about managing water bodies. And so this talk is kind of derived from like the research that I do, but it's really just how can we use water quality and algae data to better inform management. And that can be whether it's preventative management, reactive management, or even just predictive management. So understanding there's going to be a problem before it occurs will give us a lot more tools to work with that. So I'll just start off with the basics of nutrient management. So most water bodies are naturally oligotrophic. So they're pretty blue, clear. You have a healthy food web. So you don't have excessive algae because the algae growth is kind of controlled by the zooplankton and the fish and the birds and the wildlife. And so that's like your perfect, really functional food web. But with a lot of humans, we just love nutrients. It's just an integral part of our society, and especially of having large communities. So with things like intensive agriculture, stormwater, urban runoff, it's inevitably just leads to a lot of nutrient diversion into water bodies, and these nutrients will fuel huge algal blooms. So you'll have these naturally oligotrophic systems becoming mesotrophic, eutrophic, even hypereutrophic. And nitrogen and phosphorus are the main drivers of this algal growth. Some of the cyanobacteria can actually fix atmospheric nitrogen, so it makes it even tougher because you could try to limit the nitrogen, and then you might even just be stimulating the algae that produce toxins. The other thing is that algae need about 10 to 15 times as much nitrogen as phosphorus. This is pretty much all organic matter or all things that are alive. This really smart scientist, Redfield, he went around and studied a lot of different organic things and found that they all have a very conservative ratio of carbon to nitrogen to phosphorus that's pretty predictable. No matter whether you're a human, a bird, a cat, a dog, an insect, algae, whatever. And it's mostly because you just have so much protein and amino acids, which contain the nitrogen, whereas phosphorus is in DNA and ATP, and it's just less abundant in terms of overall mass. So overall, because phosphorus has been really limiting, and because cyanobacteria can get nitrogen from the atmosphere, just historically, nutrient mitigation to prevent harmful algal blooms has focused on reducing phosphorus. And I think it's important, and it's probably number one, but it's definitely not all that matters. There's a lot of other players in the game, and a lot of things that you can monitor and look at in order to determine, how your algal taxonomy is going to be just how, it's going to change or how it's going to be structured. And so the other players in the game that I would consider would be carbon, silicon, nitrogen, and iron. And these elements are important because algae have different chemical needs, and not all algae are created the same. So some of them are an integral part of the healthy food web. I would say green algae and diatoms are very important parts. And then some of them, they produce a lot of toxins. They produce taste and odor compounds. Cyanobacteria in particular, zooplankton don't like to eat them. And there's a lot of theories on the cyanotoxins, but one of the major theories is to avoid predation. So when they're producing these toxins, zooplankton are less likely to eat them. So it's a competitive advantage to produce more and more toxins. And it's just a byproduct that humans have health consequences from it. So Carly, don't hate me, but I'm just going to generalize a little bit. And I'm going to say that green algae and diatoms are the good guys and the algae that we want to preferentially stimulate, not to obscene levels, but they're a very important part of our food web. And then the cyanobacteria are the bad guys. So not all of them are bad, not all of them produce toxins, but we're learning every day about a brand new toxin we had no idea about before. So the more research that comes out, the more it leads freshwater water body managers to say, okay, a lot of cyanobacteria are causing a lot of problems. And so because they have these different chemical needs, we can just kind of keep it simple and say, good water chemistry is going to drive good algae. And if we can't control our water chemistry, we can monitor it at least, and we can know if our water chemistry is going to lead to good algae or if it's going to lead into bad algae and if we might have a problem. So starting off with some of the elements that I mentioned, carbon is really going to be CO2 and the form that it is in based on the pH. So because algae and cyanobacteria photosynthesize, they turn carbon dioxide into oxygen and get some energy from doing that. So based on the pH of the water is actually going to determine the form of carbon that they can uptake. So at a low pH, it's mostly in the form of carbonic acid, and that is in equilibrium with carbon dioxide. So if you look, carbon dioxide plus water is the same chemical formula as carbonic acid. So carbonic acid can dissociate into carbon dioxide, and that can poof out of the water. And so in general, at a low pH 2, you have a lower alkalinity. So you're going to have less overall carbon that's available for algae. And in these situations, green algae are good at carbon concentrating, a little bit better than cyanobacteria. And then diatoms have a lower carbon need overall. So they're going to be a little bit more competitively advantaged. And in general, you're going to have less productivity at lower pH because you'll have less carbon availability. Now, at a medium or a high pH, you're going to have a lot more bicarbonate or even carbonate. And that's just because a factor of, it all being dissolved at a higher pH, you're going to have a higher alkalinity generally. And so you'll have a lot more carbonate minerals that are releasing this carbonate into the water column. So you're not as likely to be carbon limited, and therefore cyanobacteria tend to be a little more aggressive and a little bit more competitive under these circumstances. It's also a product of, they're not limited by anything, so they can take advantage of their other aggressive tendencies to get nitrogen or to get phosphorus. So silicon, I would say, is very underrated because diatoms are awesome, and they don't actually have to use carbon for their cell walls. So green algae make their cell walls out of cellulose. Cyanobacteria make them out of peptidoglycan, which is another sugar. But diatoms just polymerize silica. So if there is enough silica around for them, they actually have a huge competitive advantage in that aspect, that they don't need to photosynthesize and waste that energy to make cell walls. They can just do that with inorganic compounds like silica. And so if there is enough silica, they're stimulated. But we've decreased silica loading into a lot of water bodies by damming and just in general, by pumping a lot of nutrients in, you can have a big diatom bloom early on in the season. and they'll use up all the silica and crash, and then you won't have any silica for the rest of the season. Whereas before humans started pumping in all these nutrients, you had a much more progressive growth of diatoms, and they wouldn't deplete all of their silica resources immediately. And diatoms are really important because they make a lot of lipids, which are really energy dense. So they're like an excellent fuel source for driving a really healthy ecosystem. The zooplankton love them, the planktivorous fish love them, and they're great water quality indicators as well. So moving on to nitrogen, the classic way that people look at nitrogen for water quality is using an N to P ratio. And the simplistic thought is just, well, toxin producing cyanobacteria, a lot of them have heterocysts, and these are specialized cells that can fix nitrogen from the atmosphere. So if we have really low nitrogen and we have an abundance of phosphorus, all the good algae that can't fix nitrogen aren't going to be able to grow. But then all of our nitrogen fixing cyanobacteria, well, they can grow and they can take advantage of these conditions. And so you're going to select for them in these conditions. But that's kind of a simplification, because there's two different forms of inorganic nitrogen that algae can use. So there's nitrate, which is the oxidized form, and diatoms require this form. Green algae like it under certain circumstances. They're pretty flexible. They can, use nitrate or ammonium. Whereas ammonium is the reduced form, and cyanobacteria really, really prefer this form. It's a lot easier to incorporate into proteins, and so it just gives them an energy advantage. Diatoms can't use the ammonium, and green algae can, they can use either form. So in general, the high nitrate to dissolved phosphorus is going to be a lot more helpful, and having a level like that is really going to suggest that, hey, I can grow the good diatoms or green algae preferentially to the cyanobacteria. Whereas if you have you have a high N to P ratio, but it's all ammonia form nitrogen, then that's not really helping you. It might just say, instead of having nitrogen fixing cyanobacteria, you're probably just going to have non-nitrogen fixing cyanobacteria. And so the last one I want to talk about is iron, which I would consider definitely underappreciated fuel for cyanobacteria growth. So nitrogen fixing cyanobacteria need an insane amount of iron. That molecule on the right-hand side is a cofactor in nitrogenase, which is the enzyme that fixes nitrogen. And it's like 60% iron by weight. So they need a ton of iron. And even non-nitrogen fixing cyanobacteria still have a higher iron requirement. And iron's kind of a tricky one because iron oxidizes very rapidly in the presence of oxygen. And so if you have oxygen in your water column, you're not going to have any dissolved iron around because it's going to precipitate out to the sediment. So in a shallow water body, now the cyanobacteria have these cool little tricks, and they can secrete these enzymes that will dissolve this iron in the oxidized form and turn it bioavailable. But they can't use those tricks in deep oxic lakes because they can regulate their buoyancy, but they can only travel like 20, maybe 30 feet max before they're kind of out of whack. So they can't go 50 feet down to access the sediment in a really deep lake. But a lot of these deep lakes, especially because of all the nutrient infusion, they're getting really limited. Or they grow a lot of algae and they become anaerobic. And so when they do become anaerobic, they can release iron in the dissolved form. And because they're anaerobic, they don't have any dissolved oxygen in that bottom part of the reservoir. So then now the cyanobacteria can dive to that part of the water column, the hypolimnium, and access the iron from there. So the best way to not have the nitrogen fixing cyanobacteria is just to have an oxic deep lake and to avoid their access to the iron. But obviously you can't really control that. So it's more just about understanding what you do have. And the other thing about iron is that the iron oxides bind a lot of phosphorus. So when you do have no oxygen and iron reducing conditions, you poof off not only dissolved phosphorus, but you also poof off the dissolved iron as well. And so both of those are really good at stimulating those cyanobacteria blooms. And so here's like a little just diagram of what I would consider like the collusion between the normal bacteria in the sediment and the cyanobacteria up at the surface. So you have the normal bacteria at the sediment, and they're going to eat up organic matter. And if they eat up enough of that and use up all of the oxygen, then you'll have iron reducing conditions, and they're going to dissolve the iron by reducing it. And then the phosphorus that was bound to it gets poofed off as well. So now you have all the fuel to grow a cyanobacteria bloom. Now the cyanobacteria bloom is going to produce a lot of toxins and avoid predation by the zooplankton. So it's not going to get eaten, and instead it's just going to decay. And so, it lives out its life cycle, and it falls back to the sediment, and it returns all of that iron and phosphorus in the cells in the form of organic iron and phosphorus. And now that sediment bacteria can do the process all over again, and it can degrade that organic matter, release the phosphorus and the iron, and drive another cyanobacteria bloom. And so I'll just leave with this final little diagram, and this is just hypothetical, but it's kind of telling us how we can use, nitrogen and phosphorus abundance and ratios and forms, or we can use things like the abundance of silica and iron and the pH of our water body to understand how we had different algae communities throughout a season. So I'm a huge proponent of seasonal monitoring and starting that monitoring before you actually have an algae bloom to know what your baseline water chemistry is and what nutrients you have and what nutrients you don't have before the algae actually start to grow. And then if you track these algae throughout the season, which a lot of these agencies are doing, which, all of the data we're collecting is really great. And so it's a good way to kind of integrate everything together. And I would say, if you are having problems, but you're not looking at algae identification and kind of determining, what percent cyanobacteria, what percent green, what percent diatoms, even looking at, into the species level of cyanobacteria to see which ones are producing toxins or which ones have the ability to fix nitrogen. I would suggest doing this because it's so helpful to know how this algae community transitions throughout an entire season. And it's very classic to see a diatom community at the start of the season, and they'll use up all of the available silica if you have a lot of nutrients around. So they'll bloom and then they'll crash, and then you won't have very much silica for the rest of the season, especially in California where we get most of our rainfall during the winter time. So if you don't get much rainfall during the spring, summer, and fall, and then you have all of the runoff that comes in the wintertime, well, you regenerate your silica in the wintertime, but after your first bloom of diatoms, you probably won't see any throughout the rest of the season. And that just makes less competition for the cyanobacteria. So it gives them more preference. Some lakes, you could very easily correlate the amount of iron in like the depth that the cyanobacteria can access to your cyanobacteria abundance. And so you can probably, if you just look at it through one season, you can figure out, okay, when do we actually start releasing iron? And maybe it's only in one part of your lake or your reservoir, but then that cyanobacteria bloom that's triggered spreads to other parts of the lake as that community, migrates out to find a new niche for them. And so, just looking at simple things like the silica and the iron abundance throughout time can help a lot. If you track your nitrogen to phosphorus ratio, if you look at when you have nitrate versus when you have ammonia, this can really help. And so, all of these are just really ways that we can use the information we have at our fingertips to try and, better predict what's going to happen with our water bodies or to figure out some management strategies for them. So thank you everyone for your attention, and I'm happy to take any questions.
Thank you so much, Byron. I really appreciate you commenting about, monitoring before you actually have a bloom so you can actually implement some management strategies. Super important. We have a question: in a lake or river, do you monitor nutrients in subsurface or depth integrated samples?
That's a great question. If you have the ability, I love depth integrated. So, sampling every 3 to 5 feet, or if you're doing it in meters, classic is doing it every 3 meters. So you can get a surface sample every 3 meters down if it's pretty deep. And when you do these kind of samplings, you'll tend to find that your surface, like if your water body is stratified and you have different layers, you'll tend to find that your surface, it's all pretty much the same, and then your hypolimnium is all the same, and then you have this really small transition zone, the metalimnium. And so if you have like a data sonde, then you can figure out where you're in the surface water, the metalimnium, and the hypolimnium, and then you can get away with taking 3 samples. But if you have active like nutrient or iron release, then you're going to have a higher concentration. The hypolimnium is kind of like stagnant. So if it's actively being released, you'll catch more of it down closer to the sediment as opposed to like the upper hypolimnium. So I guess that's a bad answer. It kind of like depends. But yeah, the more data you have, the better.
Awesome, thanks. In a resource-constrained public agency managing a lake, what's the most effective yet affordable method for collecting the biological data, i.e. diatoms, green, and cyanos, needed for the seasonal pattern analysis? And what frequency would be best and most affordable?
I would say if you can justify it, having someone with a background in algae, like someone like you, like we have some experts here where I work that do cyanobacteria or just algae identification, and they're so quick. Like I'll look on the microscope, but I'm a chemist by training, so I'm not the one to know like, oh, this is this type of species of cyanobacteria, this type, or no, that's a green algae, or this is pico cyanobacteria. I don't really know that stuff, but there's a lot of experts that do that would be able to serve that capacity. And just with given a microscope and a centrifuge and a water sample, they could tell you exactly what's in your water. I would say that's probably like if it's a public agency that has multiple water bodies, it's a good investment to just have one person that does that for all of the water bodies. And then they can also do research into like how these communities are changing. There's so many things you can do with that information versus sending it out to a lab. There are a lot of labs that do it, and our lab does it, but we're out in North Carolina, so there's I'm sure a lot that are closer to California that you could probably get a contract with to say, hey, we want to do 40 samples throughout the year at a discounted price. From my understanding, they're not extremely expensive because it's like 10 minutes for an expert to do that identification. I would say it would be one of those things where if your water body is dramatically changing, like community structure, like so frequently, you would want to start off doing it more frequently and then figure out when you can actually, like what period it shows like dramatic changes. I think you have a much better handle on this, but my perspective is like, an algae life cycle is like around a couple of weeks. So if you monitor, monthly, you should see fairly decent changes in between that month because you won't have the same algae that was alive at the beginning and at the end of that month when you sample. If you can do every 2 weeks, I think that would be better. But I think, once a month throughout the period of, algae abundance, obviously you don't want to be, doing that before you have a ton of algae. It's just kind of more of a waste of money. But, start maybe April or so, April, May, June, July, August, September, October seems to be kind of reasonable.
Awesome. Yeah, and always feel free to reach out to our Swamp Have program as well. We'd be happy to help with that. How do you suggest incorporating impacts from higher trophic level species such as sediment resuspension from carp?
That's a great question. It's not really in my area of expertise. I think I've primarily focused on deeper lakes. And I know that carp are a huge issue in like the shallow lakes. Like I work on Utah Lake a little bit, and they used to have a lot of aquatic weeds just aquatic vegetation that reduce resuspension from all the waves and everything. And then they stocked it with a bunch of carp. carp ate all the plants, and then you have suspension from the carp and the waves or the wind. So it's definitely not ideal. I think that plays a lot into, like just nutrients getting into the water column. So it's a lot tougher than just having a stagnant water body with minimal resuspension versus, when carp are kicking things up, you can assume that you're not going to be phosphorus, nitrogen, or iron limited. But in that aspect, you could probably assume a little bit more silica. So yeah, I think that's a great question. And that's why, holistic understanding of water bodies is very important. So, someone that focuses on just like the water chemistry and maybe a little bit of the algae here and there wouldn't have all the answers. Whereas, like someone that focuses on fisheries and someone that focuses on aquatic plants and, the whole big picture would be a lot better way to kind of understand these more complex ecosystems.
Awesome, thanks. I think we have time for a couple more questions. Does deep lake cyanobacteria methylate mercury? Can your information be used to stop methylation?
So I don't believe the cyanobacteria are the ones methylating mercury. And that's just the like iron reducing and sulfate reducing bacteria primarily. I did study that like in grad school. It's not my focus right now. From my recollection of what I was studying, I would say sulfate reducing bacteria were, in my opinion, they were like the key methylators. So you have some iron reduce, and like if you look at like the redox ladder, so you have like oxygen reducing conditions or oxic conditions, nitrate reducing, manganese reducing. So in those three, there aren't any known mercury methylators. Then you get down into iron reducing conditions, and there are some iron reducing bacteria that can methylate mercury, but they're not like super prevalent. Then you have sulfate reducing conditions, and the sulfate reducing are the predominant genre of mercury methylators. And then you have methanogenic conditions, and they can methylate mercury, but they're also very good demethylators. And so I think one of the takeaways from what I remember was actually bloom dilution. So that concept is that under anaerobic conditions, you can have more mercury being methylated, but because you release more nutrients and you grow more algae, you actually have a higher algae to toxin ratio for when you're putting it into the food web. So like if you just think of like, okay, the amount of carbon I have in the water column versus the amount of methyl mercury I have in the water column, even if you have more methyl mercury, if you have 5 times as much carbon, then you're going to have more nutrients to toxins. So, there's kind of conflicting studies on like things like oxygenation to repress mercury methylation, because maybe it would repress it by keeping it oxic. But then on the flip side, if you reduce your overall algal productivity, and then now the zooplankton have less to feed on, and there's more methyl mercury in that algae, it can become a problem. So mercury was really a tough one. But yeah, if you want to send me an email, I'm happy to talk about that stuff. It was fun back in the day.
Awesome. In shallow lakes, what do you see as the best management strategy for controlling the effects of iron mineral reduction and phosphorus released from sediments?
Shallow lakes are tough. So I would say, you really just can't get away from it, because the cyanobacteria can always access the upper sediment and get that iron oxide. You can mitigate phosphorus chemically with different ways. I haven't seen oxygen being very effective. If anything, it's detrimental in shallow lakes because you're just stirring it up even more. So I'd hate to say that on a presentation like this where I wanted to talk about all the other things, but it comes down to phosphorus, chemical inactivation. I'm not the biggest fan of alum. And then I work for a company that sells like Phoslock, so I don't want to just sit here and be the chemical salesman. But yeah, I would look into like phosphorus mitigation. I guess iron's actually a decent, I've seen some studies where they added enough iron to, because you have oxic conditions and you're not going to have the iron dissolve, then you can actually kind of enhance the sediment binding capacity of phosphorus and keep it in that bound up state. But then another thing is that a lot of the shallow lakes I've worked on, they underestimate external loading. And so they'll think of, oh, okay, well, we get so much. And if you have carp and you have a lot of like resuspension, you do get a lot of phosphorus from the sediment. But if you don't, then you're probably getting a lot from your watershed and from like nutrient loading in like the early spring. It's like a huge factor. And so if you get all this phosphorus that gets dumped in before you're even monitoring it, you won't know because there's not enough light to drive that algal growth. And then, by the time the light hits, you have this, a ton of algal growth, or you have a lot of that phosphorus that's just like slightly bound up to the sediment, but stuck in the pore water. And so then the algae can access that. So I would say in a lot of shallow lakes, they kind of, you underestimate the loading from watersheds. But again, a complex question. So if you want
we're getting heavy hitters today.
happy to talk about that more. But yeah, great questions.
I think there's time for one more and then we'll have to pass. But have you ever seen evidence of bloom dilution before and or ripple effect after an algae bloom?
And is this in relationship to mercury methylation?
I think the participant actually said that, oh yes, it is in relation to that.
So yeah, I have seen bloom dilution. My PhD advisor is Mark Butel, and so he looked at oxygenation in these twin lakes in Washington. So they had this just model system to study where they were these two geochemically very similar lakes, similar depth, and they oxygenated one of them and didn't oxygenate the other one. And they wanted to see, okay, with oxygen, you usually see lower methyl mercury. They saw that, but then they saw higher methyl mercury in the zooplankton and in the small fish. And mercury studies are tough because they take so long to actually translate up the food web. So to change the concentration in large fish, whether you're trying to reduce the concentration or whether you're trying to see if the concentration is increasing because of enhanced anaerobic conditions, it takes a long time. But they saw increased methyl mercury by dry weight ratios in the zooplankton and in the small fish in the oxygenated reservoir. And they linked that to the reduction in productivity. So when they oxygenated it, they stopped all of the nutrient release and they had lower algal abundance. So the zooplankton had less stuff to prey on. And then the organic matter that they're eating inevitably had higher methyl mercury to carbon ratios in it. So they kind of stocked up their food source with the toxins, unfortunately. And it's not, all lakes are different. So some oxygenation systems will stir up the sediment water interface, and then you'll release more nutrients. So in that aspect, maybe that would be the best. So you have oxic sediment water interface, no methyl mercury, but then you have more nutrients. But then that's a tough situation too, because sometimes increased productivity leads to other increased toxins. But yeah, it's always fascinating to talk about.
Yeah, definitely. Well, thank you so much for your time, Byron, and for helping answer and explain some of these difficult concepts. We really appreciate it. With that, we're going to move on to our last speaker in this session, Dr. Jamie Smith. She's a senior scientist at the Southern California Coastal Water Research Project, known as SCWRP, who studies the ecology of harmful algal blooms in aquatic systems. She wanted to highlight some of the project contributors. Dana Schultz is a senior research technician at SCWRP who specializes in HAB ecological studies. Rich Vadnais is an engineering geologist with the North Coast Regional Water Quality Control Board and has extensive experience with benthic cyanobacteria in Northern California. Dr. Emily Duncan is a senior environmental scientist with the Los Angeles Regional Water Quality Control Board and is the regional HAB response liaison. Dr. Susanna Thoreau is a senior scientist at SCORP who focuses on the development of molecular methods for bioassessment and the study of microbial communities. And with that, I'll pass it on to Jamie.
Great. Thank you, Carly. Appreciate the introduction. And again, I'd like to thank my co-authors for their contributions. I'm honored to be able to present on all of our behalves. So let's see. Oh, okay. That's right. They warned me there would be a lag. So today I'm talking about DNA-based methods for identifying toxin-producing cyanobacteria. I think Byron gave me a really great lead-in for some of the topics that I was going to cover today. But to kind of just take a step back, cyanobacterial HABs are a major issue in California. I'm sure many of you have heard about them, particularly in the last couple of years. There's been a lot of kind of high-profile news stories regarding harmful algal blooms. And California has really done a lot since 2016 to understand the extent and magnitude of the issue. And it truly is a statewide issue. I'm showing here on the left-hand side the 2020 HAB reports and advisories, and they span the entire state. And we have a number of different issues with different toxins. Microcystin has been a historical focus, but there are issues with other toxins like antitoxin and cylindrospermopsin, issues with multiple toxins simultaneously. And so this is a, let's see, this is a cross-cutting issue as far as beneficial uses are concerned. Harmful algal blooms are a cross-cutting issue that affect basically all of the core beneficial water uses, mainly due to the toxic secondary metabolites that they produce. Cyanobacteria have been around for 2 billion years, so they make all kinds of chemical compounds. They obviously cyanotoxins, as well as taste and odor compounds, and a lot of other things, like as Byron also mentioned in his talk, there's the more we look, the more we find different kind of toxic metabolites that cyanobacteria make. And so this obviously impacts, drinkable, fishable, swimmable, and aquatic life uses, as well as a lot of tribal uses, which I'm sure this will come up later in the tribal use panel today. But there's been a lot of study, but we still have a hard time really assessing the overall impacts on beneficial uses. And that's because there's a lot kind of going on when it comes to harmful algal blooms. So I just want to kind of put in your mind a framework for what makes a toxic algal bloom. So we have cyanotoxin production, right, which is the source of a lot of the issues, but that kind of requires both the presence of a toxigenic species as well as environmental conditions that promote toxin by those species. Toxigenic species don't just pump out toxin all the time. Toxin production is a biologically mediated process based on environmental cues. And so your nutrient conditions really, and temperature and other factors really influence the toxigenic species to either ramp up or kind of ramp down their toxin production. And so understanding this issue is we kind of really need to look in these different boxes, right? So a large focus thus far has really been on cyanotoxin production and characterizing where we're seeing the toxins. But there's a lot less focus on identifying, the environmental conditions that lead to toxin production and the toxigenic species that we're actually dealing with. And really, to effectively manage cyanotoxin blooms, cyanotoxin producing blooms rather, we really need to kind of have this holistic approach where we're looking at all of these things because again, Byron gave me a really great lead in. Depending on the species that you have and the environmental conditions, you can have very different kind of cyanotoxin outcomes. And today I'm going to really focus specifically on the presence of toxigenic species. So there's multiple ways that you can go about really characterizing your algal community. And the most traditional way is microscopy. So you go out in your environment, you collect samples, you do morphology-based taxonomy, and then you can kind of get a sense of what's going on in your community. But this is actually a bit, challenging, and there is a high level of training that's needed to do this. I'm just showing here kind of on the left panel here, just to give you a sense of what these guys actually look like. So morphology-based taxonomy is possible with a trained expert, but it's challenging, and there's a number of issues that it actually can, depending on the level of taxonomy that you want to do, it actually can be quite time consuming, as you might need a dedicated staff person to basically work through these samples, depending on how many you have coming in. And the other issue is you do get disagreement among taxonomic analysts, and so you can end up with variation depending on who's analyzing your samples. So another way of going about characterizing your community is a molecular-based method. So instead of collecting samples and doing morphological IDs with microscopy, you can basically collect the environmental DNA, extract that out, and run it through a biological bioinformatics pipeline on the computer, and you get taxonomic information that way. And kind of what I want to talk about today is that might be, a nice complement and a more streamlined and standardized approach, and potentially faster than kind of your traditional morphological IDs. So today I'm going to show two different case studies where we've used molecular barcoding to look at cyanobacteria in kind of cyanohab assessment scenarios. So talking about how you compare your DNA with your toxin analysis across diverse systems. This works across many different systems. Also identifying our problematic genre, and not so much environmental risk factors in this talk, but I think that's the ultimate goal of both of these studies that I'm going to talk about. And then evaluating, kind of discussing how DNA can be a complementary tool to some of the more traditional or common HAB indicators like cyanotoxins. So to start, I'm going to talk about a case study, and this is a large contribution by Rich Fadness. So I just want to acknowledge all of his hard work in the field collecting all these samples. But in 2019, we have 18 samples collected throughout kind of the rivers up in Northern California, where we collected samples for cyanobacterial barcoding. And so we used a cyanobacteria specific primer set and a curated cyanobacterial reference database that's based on Silva and GenBank, and as well as traditional microscopy and cyanotoxin measurements. And so what we found, or kind of what I want to show you, is that DNA-based methods do generally offer you a bit more of a refined view of your cyanobacterial taxonomy. So I have two arrows on this bar, and unfortunately the circles aren't showing up as they do in my version, but essentially what we're looking at here on the x-axis is what the microscopy ID for those samples were, and these are benthic mat samples too, to help you kind of, so these are kind of algal mats that sit on the benthos. They're usually filamentous typically, and so on the y-axis is the microscopy ID, and on the on the x-axis and on the y-axis is the DNA. The numbers are the number of samples with those IDs, and then we're looking at how they agree. So the purple arrow, what I wanted to bring your attention to is, yes, a lot of times we can get good agreement between our morphological ID via microscopy and our DNA based identification for genus level taxonomy. The red arrow pointing at the 33 is an interesting scenario. This is a scenario where we have Formidium and Microolius. These can look very similar. They also, their taxonomy was recently split, and the Microolius is more associated with toxic species, whereas the Formidium is not. And so in this case, these likely are more of an instance of this taxonomic change based on the toxin producing profiles of these different species. But with the DNA samples, once you have your DNA sequences, you can rerun them extremely easily through the pipeline, whereas with a microscopy sample, that's much harder to do. And so that's an advantage that DNA-based methods offer. They also offer, much deeper taxonomy. So here I'm showing just kind of a snapshot of what the diversity found in each of the river sites. And I wanted to also just call your attention that, these are, 10 plus genera, and each of these genera from a genomic standpoint, we've combined species. So there's usually generally multiple species within each genus. So we could even drill down deeper into this analysis if we wanted to and look at all of the different species of Anabaena or any of the other genera that we would be interested in. And so that was all of the cyanobacteria present in those mat samples. You can pull out just your toxigenic taxa, for example. So now I'm just showing toxigenic taxa. And this blue arrow here is indicating the area of the river where we found the most toxin detections. And so that, when we look at this, that corresponds with the highest presence of toxigenic taxa. So that can help kind of guide, you can use this tool in a way to kind of guide where you might have your biggest problems based on the biology. But it's not a perfect tool, and I don't want to sell it as your cure-all, but you can have instances. What I'm showing you here on the left is our toxin profiles from the mats, and on the right is the community composition. So you can have samples where, yes, you have toxin present and you have relatively similar communities, but then you can also have, as I highlight on the right, scenarios where you have dominance of one taxa associated with toxin, but then in another mat that's dominated that same genre, you don't have toxin. So it can help guide you, but it's not going to be a 100% there's toxin here, there's not toxin here. So paired sampling gives you the most complete picture of what's going on. So our second case study is on regional lakes in the LA area. So we did a regional lake survey back in 2020, where we looked at 17 different lakes and characterized the cyanobacterial community, the cyanotoxins present, and the basic environmental conditions. I'm going to give you a little snapshot of this study because there's not a ton of time left. But basically what we found was there were cyanotoxins in 7 of these lakes. And cyanotoxin genera that we found were more diverse than kind of your usual suspects. In lakes, a lot of times Microcystis kind of gets the bad rap as being the causative genre of microcystin. But in this case, that Microcystis is present. So that's that light kind of green color. So if you look at Leg Lake, that lake had microcystin and was dominated by Microcystis. But we have these other lakes where we have, relatively, detectable levels of microcystins, and you do have Microcystis present, but there's other taxa, meaning, you can't just say, if I have this, I have that, right? Similarly, we have a couple of lakes with more novel toxins, things that haven't been studied as much. And so this is a nice example of, these are relatively understudied lakes. So we can get a sense of, well, what's the toxin present in these lakes and what are the potentially associated producers? And so if you have a monitoring program, you can start to kind of get an understanding of, oh, if I start to see, in the instance of Machado, if I start to see Sphaeospermoptis, which is this purple, maybe I need to be concerned and I can guide my cyanotoxin sampling appropriately. And then I want to bring your attention to this last slide, my second to last slide, where these are lakes where we detected toxigenic genre and we had no toxin, right? So again, this is a good tool to understand these lakes have toxigenic production potential, but there was not toxin at this point in time when we went to sample. But you might want to then pair this with observations of eutrophication or other indicators to say, are these high risk lakes? Should I how should I adaptively kind of manage this system to make sure that I'm assessing my risk of cyanotoxins appropriately? So in conclusion, Just some final thoughts. So DNA-based approaches, obviously I hope I made the case that they can offer a robust tool for morphological identification. You can get a high level of diversity and you can assess that effectively across your systems using DNA-based approaches. And I would highly recommend, pairing this and having like a holistic system by understanding your trouble species and the typical toxins that they produce, as well as the environmental conditions. You can really start to work on adaptively managing your system. And again, I think the speaker before me, Byron, mentioned, you want to start sampling before you have an issue so you can really understand how your system's changing. So again, understanding drivers requires consistent sampling, characterization of the toxin diversity, and the environmental conditions. And so with that, I really thank you for your attention and I look forward to any questions.
Thanks so much, Jamie. Really appreciate it. We do have a question. Since toxin production and or release can be at the species or strain taxonomic level, is there hope that DNA-based approaches can attain that resolution?
Oh, absolutely. I think that, I started kind of working on algae back in 2012, and it's just amazing the progression of molecular approaches just in that short period of time. And I think that, there's multiple ways you could use molecular approaches. There's tools where you could take, so what I'm showing you here is barcoding. So we're not saying this taxa is producing the toxin, it's just giving you kind of an idea of what's there. And you can develop kind of correlative relationships of like trying to understand who your problem taxa are. But there is starting to be approaches where you could sequence the community and pull out both the taxonomy and the toxin producing genes and link those two together to say, this is who's producing this strain or species is who's responsible or who's the common toxin producer in my lake. So I think that we're really, the possibilities of doing that are just increasing and are becoming more and more accessible over time.
Awesome. First of all, great presentation. Did you look at environmental conditions to see if there was any correlation with toxin production?
So we did do that. I didn't have time to put that into this presentation. And that's kind of I would say that's an active analysis that we're doing. From a high level, I think that we did see, correlations between more eutrophic systems, particularly in the lakes case study is where we did that. And I think you do see a general kind of correlation between your more eutrophic systems and the presence of toxin, but that's an ongoing analysis. So hopefully I'll be able to present more information in the future on that.
Excellent. And at what concentrations do these various toxins become harmful? We've done lots of sampling in streams across California this summer and have gotten a lot of hits, but I really want to figure out which of these sites would have harmful levels of these toxins.
Mm-hmm. And that's a great question. So from a human health perspective, in planktonic systems, the swamp program has great kind of guidance levels on what concentrations are harmful. There's the, your yellow, orange, red for the three main cytotoxins of concern. And so I point you to the swamp FHABS program to look at that guidance from a human health perspective. From an aquatic life perspective, that's a more difficult kind of question to answer. And same thing for mats. I think that's an ongoing research. But generally, I think that what the common use is to use those human health thresholds right now to assess kind of your relative harmful versus not harmful or less harmful concentrations. So hopefully that helps. But that's an area of active research of really understanding your chronic versus your acute and all of those effects. So good question.
Awesome. And just to kind of piggyback on that, yeah, please look at the My Water Quality portal. That also has some information, which is on the State Water Board's website. So with that, I think that's all of our questions. And I just want to thank Jamie for her time today and all of the speakers in this session. It was super awesome. And with that, I will pass it on to Allie before our break.
Cool. Thank you so much, Carly. And thank you all. Those talks were all super interesting. It was nice to kind of get some biodiversity talks in there and then more focused in on cyano and algae and eDNA. So thank you all. We're ready to break. We've got a good chunk of time ahead of us with this estuary panel. So please take a little break, bio break, get coffee, what have you. I do also have a poll that I wanted to share with you guys. So if you get a chance, go ahead and provide your answer there. Just a question about if you've attended CABW or CalSFS before. And we'll share those results after the break. Thanks, everybody. right, hi everybody again. We are going to go ahead and get started here. And just I wanted to quickly share the results of that poll. Thanks to those of you that participated. We got a pretty even-ish distribution of, yes, you've got some fully committed CABW, SFS folks. Some have been coming off and on, but a lot of first timers. So welcome to all you newbies. I'm excited to hear that we're getting new people in and a nice new influx of fresh faces and people. So thanks for joining us. And with that, I'm going to go ahead and kick off our final panel and session of CABW. And before I do that, I'd like to introduce the moderator, Jennifer Sue. She's a senior ecologist at EPA Region 9 in the Wetlands Office, and she serves as a regulatory expert for Section 404 and 401 of the Clean Water Act and the Marine Protection Research and Sanctuaries Act, with an interest in forging effective partnerships with agencies and stakeholders to ensure multi-beneficial environmental outcomes. She is currently working on several initiatives to improve regional wetland outcomes, including development of a SFA Wetlands Regional Monitoring Program, supporting the Bay Restoration Regulatory Integration Team to improve wetland restoration permitting, and serving on interagency project teams to promote the beneficial reuse of dredge sediment for bayland restoration. Jennifer has over 23 years of experience in aquatic system characterization and management, including water sediment and biota monitoring, rapid ecological assessment, wetland jurisdiction delineation, compensatory mitigation analysis, and NEPA CEQA compliance. So thank you so much, Jennifer, for joining us today and moderating this important panel.
Thank you very much, Allie. I wonder, is my video showing at all?
I believe so.
Okay, great.
Yes, sorry.
Yeah, I'm super excited about this panel. We've got three discussions that we're going to hear today. Dedicated estuarine scientists who are going to talk to us about bioassessment and monitoring tools for estuaries and developing, somewhat of a standardized framework and tools that they are using currently. So with that, I'm going to introduce the first speaker, Jan Walker. She's an ecologist at SCORP, who specializes in developing assessment tools and programs for coastal wetland ecosystems. She received her BS in environmental science from the University of Virginia and her PhD in ecology from the joint doctoral program at San Diego State University and UC Davis. Her research interests focus on developing condition assessment tools for estuaries and other marine habitats, as well as causal assessment methodologies to identify and predict potential stressors to these ecosystems. Take it away, Jan.
Thanks, everyone. Can you see me? And let me see if I have control. Okay, awesome. So hi, my name is Jan, and I just want to first start by kind of setting up the session today with myself, Kevin, and Michelle. Estuarine and lagoonal systems provide an abundance of beneficial uses and functions and are often considered receiving waters for rivers and streams. And estuary management is gaining increasing attention due to the effects of changes in stream discharge and rising sea level. And although California has invested an estimated $600 million over the past 2 decades to acquire and protect estuaries, the state really lacks a coordinated statewide monitoring program for assessing estuarine health in a systematic, consistent manner. And so this session, Building Capacity for Monitoring and Bioassessment in California Estuaries, is really aimed at talking about developing assessment tools and monitoring frameworks for California estuaries. And so first, I really want to begin this session by asking this broader question of why should we focus on assessing and monitoring estuaries? And so I'm going to start with kind of 5 broad reasons why I think that we should be assessing estuaries. And this first being that, estuaries provide a broad suite of important functions and beneficial uses. We all know that estuaries have a number of ecosystem services, from providing recreation to storing carbon to controlling diseases and vectors. Additionally, estuaries are affected by many stressors in complex ways, right? So estuaries are impacted by many anthropogenic activities, such as changes in land use, like coastal development and farming, which can lead to runoff and pollutants. And there are many climate change stressors, such as sea level rise and ocean acidification. And of course, these estuaries usually host ports and marinas, which can lead to invasive species. Specifically in Southern California, wetlands historically supported large areas of vegetated and unvegetated marsh. And around 1815, there are approximately 33,000 acres of all coastal wetland habitats. However, in the following 150 years, more than 62% of these wetlands were lost. And by 2005, only 12,800 acres remained. So it's important to really understand the baseline conditions of these estuaries in order to get a sense of how these critical habitats are changing. And so thirdly, estuaries offer protection across the land-sea interface. And so a key objective for management is to understand this land-sea connection. This is especially true for the 30 by 30 movement, and specifically in California, where we are attempting to conserve 30% of California coastal waters by 2030, where a main strategy is to build protection across the land-sea interface. So estuaries are critical piece in this connection. And of course, estuaries build coastal resiliency. So a huge emphasis on both the state and national level is in regards to coastal resiliency, and estuaries are a key component to building resiliency due to natural flood protection and buffering. And so lastly, most aquatic resource management programs affect or are affected by estuaries. Every coastal region has estuaries, where there are 534 estuaries in California alone. And each coastal regional water board recognizes this estuarine habitat, or EST, beneficial use in their basin plan. And so, however, the state's 303D, 3035B integrated report does not include estuaries, despite the fact that they are included in many basin plans. And this is most likely because we don't have the tools to assess this use. Therefore, it's unclear whether these California estuaries are in fact supporting this beneficial use. And despite the extensive number of estuaries, there is no standard monitoring or assessment of California estuaries. And because of this lack of monitoring, we really have limited bioassessment tools because we lack the baseline data to build these tools. And so bioassessment can play an important role in estuary assessment. Sorry about that PDF on the left here. Looks like we had some formatting issues, but there have been past efforts to adapt marine bioassessment tools to brackish environments. For example, there have been efforts to adapt the AMB, which is a macro-benthic based index designed to assess the degree of anthropogenic disturbance in coastal waters, to more brackish and tidal fresh environments, like in San Francisco Bay, which this index for these systems is called the MAMB. And these past efforts have largely focused on shallow subtidal environments, and we have yet to develop bioassessment tools specifically for estuarine habitats that include both the intertidal and the marsh habitats. And so bioassessment tools for estuaries that can be incorporated into routine assessment and reporting programs could be based on a variety of assemblages, including plants, SAV, benthic infauna, fish, or multi-assemblage index. However, the first step in developing bioassessment tools is to generate a consistent data set across a gradient of conditions that can ultimately be used to support index development. So that's kind of where what we're going to be talking about today comes in. We have recently collaborated with a statewide team to develop and test a standard assessment approach and a set of SOPs that can be used to evaluate estuarine conditions statewide and eventually provide the data necessary for index development. So the remaining part of my talk is going to be focused on this framework for bioassessment. So to help address this monitoring gap, the Ocean Protection Council, or OPC, is supporting a development of a statewide estuarine monitoring and assessment framework that will initially be used to evaluate the efficacy of the Estuarine Marine Protected Area Program. So I'm going to briefly tell you about kind of the backbones of this program. And then Kevin, who is our next speaker, will go into more detail using a case study. So to give some background, in 1999, California legislature passed the Marine Life Protection Act, aimed to protect California's marine natural heritage through establishing a statewide network of marine protected areas, or MPAs. And by 2012, California completed the implementation of the MPA network, spanning the state's entire 1,100-mile coastline, protecting 16% of state waters. And so California MPAs include a number of coastal habitats, from kelp forest to rocky intertidal benches to deep and midwater areas and estuaries. And for the last decade, long-term monitoring has been essential for evaluating the efficacy of MPAs. For example, 2 networks exist to monitor the rocky intertidal benches. So this is the marine network. And then the kelp and open water areas are monitored by PSCO. And so this long-term data has been critical in evaluating the effectiveness of MPAs and building large data sets for evaluation. And so of the 124 MPAs that are in California, 23 of these MPAs are estuarine. And the problem is that most monitoring has been focused on the offshore habitats, and there are currently no long-term monitoring programs for the estuarine MPAs. And so this lack of estuary monitoring has recently become even more apparent because the decadal review of MPA efficacy is due to the commission next year, and there is no data to determine effectiveness of these estuary MPAs. So this is where we have come in. OPC funded the statewide team to develop the framework to address this fundamental question of estuary MPA effectiveness. But a key element to the development of this standardized monitoring protocol is that it can be used not only by the MPA program, but by any program aimed at assessing estuary function, condition, or health to provide data that can be easily compared across systems and between programs. And so a key component of this framework was to build consensus among the framework and the monitoring protocols. And so we've engaged with a diverse advisory council to achieve this. And so our framework has 4 key principles for long-term monitoring: flexibility, comparability, interpretability, and practicality. And I'm just going to briefly kind of go over each of these tenets. So flexibility. This EMPA framework takes an ecosystem function-based approach to assessing the condition of estuarine ecosystems, with the underlying principle that all healthy estuaries should provide a suite of ecological functions and ecosystem services, and that failure to do so is an indication of degradation. So this approach is unique in that it focuses on assessing function rather than assessing species or habitat. And this builds in flexibility for application within our highly heterogeneous systems and our highly heterogeneous state like California. So our team has chosen 11 priority ecological functions of estuaries, and here they are listed here. So users focus on functions and then choose specific indicators and collection methods to evaluate their given function. So I'm showing only a subset of our SOPs, but this table links functions to indicators to collection methods to SOPs. So for example, if you wanted to mainly monitor your estuary for shellfish support, you would pick the indicators that match those functions. So there's a lot of flexibility built into this framework. Next, to make sure all estuaries are sampled in consistent manners and to ensure comparability and interpretability between estuaries, our users follow a prescriptive framework to characterize estuaries. So as part of this framework, users first classify their estuary by geomorphic form. So for this specific project, we use the frequency of the mouth being opened or closed to divide estuaries into 3 classes: lagoonal, embayment, and riverine. Next, users identify the main landscape features or habitat types that will allow them to assess ecological conditions. So for each estuary type that they identify, users use these function to feature tables to link their priority ecological functions with landscape features. And the purpose of this is to make sure that users are sampling where they want to assess these functions. So a table might look like this. Here's a large embayment table where you have functions in the first column and landscape features along the top. If our users were interested in surveying nekton habitat, sea level rise amelioration, and nursery habitat, then they should prioritize their sampling in the high marsh, low marsh subtidal channel and SAV. And so the prescriptive nature of this framework and the direct connection between function and habitat or landscape feature has two main benefits. One ensures standardization, where users are sampling the same habitat types across estuaries, and it's transferable system to system and resilient over time. For example, if an area of the system changes, then users can move or sample elsewhere depending on these landscape features. Third, our third principle is interpretability. So where comprehensive and consistent sampling increases our ability to interpret data. So our outline walks users through a process for choosing sampling locations on site maps prior to entering the field. And Kevin will kind of go a little more into this using a real world example. But one of our key focuses is on cluster sampling, meaning that we choose 3 sampling stations, so 1, 2, and 3 within our given sites, and then collect a number of different metrics per sampling station. So this allows us to interpret across multiple estuaries. And so lastly, to increase the feasibility of the sampling protocol, we have chosen to limit data collection to what can reasonably be accomplished in a 3-day sampling campaign. So a 3-day campaign should reduce personnel costs and allow users to implement the protocol across multiple sites. Now we are piloting this assessment framework at 15 estuaries, half MPAs and half reference sites. In our site selection, we have a variety of different estuary types, such as barbelled estuaries or lagoonal estuaries, embayments, and riverine. And we are monitoring these sites regionally, dividing up our partners regionally. And so we just completed our spring and fall sampling, and we'll report back to OPC regarding the effectiveness of this framework for assessing the condition of estuaries. Now, there are a number of products and outcomes that are going to be coming out of this work, one being a standard set of protocols and monitoring tools, a baseline assessment of estuarine and PAs, procedure for data management, and then a structure and approach for ongoing coordination of estuary monitoring, which is going to be really crucial in ensuring the longevity of this monitoring program. Oops. And so lastly, the hope is that this statewide monitoring program will help move us collectively forward with bioassessment. So initially, the program will provide the much needed information on estuary condition and stress. However, over time, as consistent monitoring is a necessary first step to build the data sets necessary to develop bioassessment tools. And so with that, thank you so much for listening. And next, Kevin will kind of go into more detail regarding a case study specifically for one of our estuary sites.
Thank you, Jan. Yeah, I just want to remind everybody, as Shuka's put in the chat box, that we're going to be answering all questions during the Q&A panel after all the 3 speakers have presented. But go ahead, feel free to submit your questions now, and we will be keeping track of those for so when we get to that discussion. Now I'd like to introduce Kevin O'Connor. Kevin is an ecologist specializing in wetland restoration, monitoring, and assessment, and is the project manager for the Central Coast Wetlands Group at Moss Landing Marine Labs. He's been there since 2008. Kevin is involved with wetland and upland restoration in the Lower Salinas Valley, the development of a rapid assessment method, several rapid assessment methods, CRAM, RIPRAM for multiple wetland types, along with the estuary MPA monitoring program in California that Jan just told us about, and participation with the California Wetlands Monitoring Workgroup. Thank you, Kevin. You're up.
Thanks, Jen, and thanks, Jan. Thanks, Jen, for the introduction, and thank you, Jan, for introducing the program here. So as Jan said, she went over kind of the larger framework of our project. I'm going to zoom in on a little bit more of the implementation and how it's worked so far on the Central Coast. So I'll be talking about the application of the EMPA assessment tools in the Carmel River estuary, but I'll also be doing some comparison to other sites on the Central Coast, as well as our regions, the North, Central, and South regions. I just want to reiterate that this is a collaborative project between multiple academic, NGO, and government agencies. So we've got Long Beach down south and SQUIRP. On the Central Coast, we have Los Angeles Marine Labs. And up in the North Coast, we have Sonoma State and UC Davis Bodega Marine Lab. We're also working with the Nature Conservancy and the SFA Water Board, and then UCLA is helping out with the eDNA work. So just general, the strategy of this project is developing this multi-layer monitoring framework. And to do that, we're doing our best to leverage existing work that's going on in the state. So we've got our National Estuarine Research Reserves and our NEPs, and collaborating with them on existing monitoring efforts. And then partnering with regional organizations, tribes, other universities, and stakeholders to help get their input on the protocols, and then hopefully eventually implementing the protocols in a consistent way across the state. And then finally, we didn't want to reinvent the wheel. So we're using existing monitoring and assessment tools that have been developed in the state. The Bay Foundation down in Southern California, with funding from the US EPA Region 9, put together a great level 3 monitoring manual for estuaries. So we have incorporated those SOPs into our program. And if there were factors that were not included within those SOPs, we imported other SOPs that would cover the factors or components that needed monitoring. Our general approach has been to identify key data gaps within monitoring programs across the state and locations across the state, and then develop a systematic data collection protocol that can be implemented throughout California to answer these statewide EMPA management questions, and then be implemented by other organizations in their own monitoring efforts. And by the end of this project, we're hoping to establish baseline understanding of estuarine condition in our selected sites, using both new data that we've collected and existing data from previous monitoring efforts. I put up here the list of selected sites that were put together by the EMPA team with input from our Management Advisory Committee. Since we have our North, Central, and South regions of the state, each region has 5 estuaries to monitor, and those estuaries are a combination of MPAs and reference or control sites. On the Central Coast, the 5 sites are the Pajaro River Lagoon, the Morrocoho Slough, the Carmel River Estuary, Arroyo de la Cruz, which is down near San Simeon, a small coastal, small river mouth estuary, and then the Morro Bay Estuary, which actually has 2 MPAs. So Jan went over this somewhat, but I wanted to just rehash the various monitoring protocols that we are implementing in this program. So it's broken up into 4 main categories. We've got abiotic factors, biotic factors, estuarine habitat surveys, general surveys, and then watershed processes and stressors. So with abiotic factors, we are deploying in situ water monitoring probes, those being CTDs and DO sensors and temp sensors. We're getting basic water chemistry at multiple locations across each estuary, doing water column profiles using a YSI, and getting nutrient samples, grab samples for nutrients. And we're also doing sediment cores of the marsh plain, intertidal, and the subtidal zones. For biotic factors, we're doing fish surveys that includes beach seines and bruvs, which are baited remote underwater videos, which we deploy at each station. Crab surveys using shrimp pots and minnow traps, and then benthic invertebrates using cores at the intertidal and subtidal zones. For general estuary habitat surveys, we're using the California Rapid Assessment Method, of course, at multiple assessment areas across each estuary. We're looking at marsh plain vegetation surveys, macroalgal surveys, as well as topographic surveys to characterize the topography of the marsh plain. General SAB surveys, including eelgrass surveys, community composition assessments, looking at eDNA, and then sea level rise vulnerability and marsh plain accretion rate estimates using set stations and feldspar markers. Finally, for watershed processes and stressors, we're looking at general landscape stressors using GIS analysis for the for the watersheds of each estuary, as well as previous efforts in historical habitat change analysis, mainly in Southern and Central California. Now here is a theoretical example of what one of the monitoring stations would look like in an estuary. So you would have 3 of these stations, as Jan said, throughout your estuary. And here you can see all the various protocols being implemented in one small location. So we've got the red lines, which represent vegetation transects with the quadrats. We've got dashed yellow lines representing the topographic surveys. Then out in the water, you've got beach seines, crab traps, eTNA, purple lines representing SAV, the white box representing feldspar. So there's a lot going on at each of these stations. Over in the real world, when you get out to an estuary, so here we have Carmel River Lagoon on the Central California coast. There are limitations and whatnot when you get out to a site, and combination of the habitats that are within a given estuary. So here in Carmel River Lagoon, we've got the North Marsh, we've got the main channel of the river, we've got the South Marsh, and then we've got various access locations in each area, limiting or putting various restrictions where we can get to the lagoon to do our assessments. So in the north, we've got the State Beach parking lot, which provides great access to the North Marsh. On the eastern edge, we've got the water treatment plant driveway leading to a restoration site where we can get to that south arm. And then on the western edge, we've got the Carmel Meadows neighborhood where we've got recreational trails and the water district has an access road. So these various access points and then the different subhabitats within the lagoon helped us focus where to put our monitoring stations. In addition, as we said earlier, this is a collaborative effort with existing monitoring programs. So we've got 2 purple dots or stars on this map here. There's a number 1 and a number 2. So the number 1 is an existing monitoring station put out by the Naval Postgraduate School. That's Mara Oriskanen. She's got a DO sensor, a depth logger, a velocity meter, temperature and whatnot at that station. And then the water district has a long-term water elevation probe at the star number 2 there, which is put online. So in the conceptualization and placement of our new probes at this lagoon, we worked with Mara and identified locations within the lagoon that could use more data collection. So the green stars are where we deployed CTDs and DO sensors, and then the yellow stars are where we deployed the temp sensors. We then went ahead and established our monitoring stations. So station 1, we always put in the main bay of the lagoon. So that's right near the mouth of lagoon, number 1 here. We then had 2 additional monitoring stations, station 2 up here in the North Marsh, station 3 down here in the South Marsh along the south arm. This is a large scale restoration project put in probably 15, 20 years ago. And then we really wanted to get some information on the input from the river. So we established a 4th monitoring station up here. And that each of those stations, we then employed each of the SOPs. So we've got water column profiles and nutrient grab samples as the red dots. We deployed the BRUV, which is the green X's. We had worked with Mara to deploy a set station in the North Marsh and the South Marsh. And we also put out feldspar markers to compare the 2 types of accretion studying methods. Beach seines, sediment cores, crab traps, you can see it's all here. And back in spring, when we did this first round of sampling, it took about 3 days to get this all done. The deployment of the in situ water monitoring probes, we did about a week earlier, so that added a little bit of time. But in general, we stuck to the 3-day monitoring effort that was planned for. Some initial observations from all parts of California, all regions. After that initial monitoring effort, there were some general framework considerations. Estuarine size is highly variable amongst the regions, which can lead to a general range of representation of 3 monitoring stations. So if you've got 3 monitoring stations in a small estuary, you're getting a better representation of overall estuarine condition as compared to larger estuary. Mouth condition is very important. So here on the right, we have a picture of the Carmel River Lagoon. When we deployed the crab trap, we came back the next day, and at the bottom, you can see the lagoon was completely empty. It had breach and our crab traps were high and dry. So knowing the mouth condition can be very important in setting out those traps and various SOPs when you're employing them. And of course, there's a variable degree of urbanization in the watershed and estuarine use throughout California, with higher pressures in the south in general than on the central and north coast, leading to various levels of stress on these estuaries. And additional logistics to consider was sampling effort, team size, travel time to get out to these estuaries to do the assessment. It was a very long drive for the North Coast team as compared to the South and Central Coast team, and then permitting and site access issues. So let's go into some of the initial results that we got. So I just want to remind you that Carmel River Lagoon is a barbell estuary, so you've got highly variable water levels through space and time, as well as water chemistry. So here's some data looking at spatial and temporal variability of dissolved oxygen in the Carmel River Lagoon. This is from the Naval Postgraduate School back in August 2020 through the beginning of 2020. And you've got 4 different locations. The first 2 are near the mouth. The 3rd row is on the south arm, and then the 4th row is up on the north marsh. And you can just see when you've got intermittent, open, intermittent, and closed conditions of the mouth, the DO is highly fluctuating through these times. But even when you do have DO crashes, as noted as those blue dots and yellow dots along the various rows, you do have other locations within the estuary where DO is just fine. So having a single monitoring location is really not sufficient, which is why we deployed loggers throughout these systems to really get in on the variability in DO temperature and whatnot, characterize them. Here you can see the grab sample and water column profiles data for all sites along the coast. I've highlighted the Carmel Lagoon here in yellow bars for each of them. So we've got water temperature, air temperature, and DO across the top, salinity and pH in the middle, and then total dissolved solids and conductivity in the on the bottom portion here. It's just interesting to note here the salinity of the Carmel Lagoon is highly variable based on where you were in the water column and in the lagoon, but we have very consistent DO levels. Another interesting to note is the general increasing trend in water temperature from the northern part of the state all the way over to the southern part of the state in the top left graph. Again, these are water column profiles and all the data for all stations at all sites. So you're seeing the total variability of all of these metrics in one graph. Here we have the proportional abundance of the fish species for the south, central, and north. You can just see in general, there was a higher diversity of species on the south coast. On the central coast, we had a lot of 3-spine stickleback and staghorn sculpin, whereas on the north coast, a lot of topspelt and sculpin and stickleback. We're hoping the fall sampling would see a little bit different results of the fish beach sites. Another way to look at the data of the fish is on a site-by-site basis. On a basic looking at a heat map here, we've got the Carmel Lagoon on the central column, the CCCAR. You can see we only caught the stickleback at that site, but other sites or other estuaries had additional species on the central and north coast, and then again the south coast had a high variety of fish species. And finally, the vegetation data looking at the south and central coast sites. We had some issues with plant nomenclature and identification in the spring amongst the 3 regional teams. So for this fall, we've decided to adopt the Jepson herbarium plant list in our data checks. So we will not have a problem of plant species name inconsistency across the state. So from final take-homes, protocol refinement and development, we've been successful in the development of the monitoring framework, but we've identified the importance of developing some guidance documents to crosswalk management concerns with SOP implementation and outputs. And we need to consider the effectiveness of the methods versus the time invested and the costs. So some methods take longer, which require more time. There needs to be an equal balance to keep on that 3-day time frame. Some other keynotes. The categories of protection of these EMPAs are more of a gradient and not a category, which can make looking at the effect of MPAs on estuarine habitat and estuarine condition difficult. Low replication within sites. So if you have a larger site, only 3 stations may not be sufficient to get at overall condition. Again, mouth condition is very important and critical for sampling time and estuary condition. And there has been some consideration of some kind of like module framework being developed. So dependent on the question, as Jen was saying, you may have additional duration, sampling effort, and higher frequency to get at those questions. There'll be a baseline level of assessment, and then there will be some enhanced efforts. Finally, our next steps is connecting with additional monitoring programs and finishing up our fall sampling. To finish off the presentation, I have a little slideshow here of the various estuaries we visited. So here's the South Coast estuaries. And then we have the Central Coast estuaries and all of our field team out there doing great data collection back in the spring. And then finally, we have the North Coast team doing their sampling. So it's been a great project collaborating with all the various CSUs, UCs, and NGOs, and look forward to your questions at the end of the panel. So thank you.
Thank you, Kevin. That was a lot of great work that you guys did this past year, and a lot of great information that you're providing to us. I'm going to segue to the next speaker, Michelle Tarian, California State University at Monterey Bay. She's in her second year at Cal State, and she's in the Monterey Bay's Environmental Science Master's Program. She received her undergraduate degree from Ohio University in Environmental Geography and Women and Gender Studies. Michelle's studies focus on estuary and land management. She has been developing a fish assemblage model for the Russian River estuary that will be applied to all estuaries in California in order to assist estuary management. Michelle also works with the Garrapatta State Park, assessing sea cliff buckwheat habitat range to support the Smith's blue butterfly. Thank you. Take it away.
Hi, I'm excited to be here. Thank you everyone for tuning in. I will be presenting on modeling fish assemblages in the Russian River estuary. The work was conducted by myself and Dr. John Olson through California State University, Monterey Bay. So I know the previous two speakers mentioned why estuaries are important, but I wanted to talk a little bit about tidal inlet bar estuaries, which Kevin did talk about. But tidal inlet bar estuaries tend to have more varying bathymetry compared to large estuaries in California, partially because of sediment blocking the exchange of water and shallow water depths. And an example, like we had seen previously, of a tidal inlet bar is shown here. On the left, you can see that the sandbar is completely blocking the exchange of water. And on the right image, it's letting the exchange of flow back and forth. And this is the Santa Margarita River estuary. And I do want to point out that when sediment fully blocks the estuary inlet, the exchange of saltwater and freshwater is disrupted, and this has an impact to critical fish habitats in California, and that includes salmon, steelhead, and tidewater goby. And then additionally, the sandbar closure can cause flooding of low-lying shore properties, which we know we have lots of those in California as well. And currently, the timing of when to artificially breach a sandbar is dependent on balances between developmental, recreational uses, and then the challenges of flooding, degraded water quality, and fish passage. And the breaching can be done naturally or artificially. A natural breaching of the sandbar occurs when enough water moves the sediment to allow the exchange of water, and artificial breaching is normally done through excavation. So we know that species utilize different parts of the estuaries in different ways. And what we want to know is what environmental factors drive the utilization and apply that to the management of estuaries. And to do so, we use species distribution models, and they will predict the distribution of a species across geographic space and time using environmental data. So in particular, the goals of this project was to model environmental effects on fish assemblages in the Russian River estuary specifically. The Russian River estuary is located in Sonoma County, California, and it is shown here in this image. And you can see that the sandbar is open in this image, the water's being able to exchange. And then we want to take this model and apply it to all California estuaries to improve estuary management practices. Some considerations for the models that we built are that we used random forests to model the associations between the presence and absence, and then also the abundance of 5 fish species and environmental data. And all data, presence, absence, data was balanced when we ran our initial models using the imbalance function, and both models were run using random forest SRC package in R. And we balanced the data because machine learning modeling has problems predicting a balance between presence and absence. So we wanted to make sure we gave the model a fair chance of predicting correctly. And then here in this image, you can see the 17 sites along the Russian River where we were able to collect data, or where data was collected from 2004 to 2019. The 5 fish species that we use as our response variables included and shown here are coho salmon, steelhead, Chinook salmon, 3-spine stickleback, and starry flounder. And this is a comprehensive list of all the predictor variables that we use for initial models. I won't go through all these, but I wanted to point out that overall, we were really fortunate to have good long-term sampling data that was provided to us by California Trout. We also had available upstream water quality data from a USGS water gauge. And then we also had assistance using GIS to determine land cover data on the banks of the river. And I do want to point out that we included fish species data, presence, absence, and abundance as part of our models to assess interspecies relationships as well. Here we see the 5 different species that we modeled, the life stages that were modeled, the total abundance is modeled, and the presence total is modeled as well. I just want to point out that we did not model adults for any of the top 3 salmonoids. That is due to differences in life stages, and then we also had low adult sample counts as well. Our modeling process, just quickly, was to use all the predictive variables and then remove any variables with low importance or any correlated variables, and then assess the overall model. So I will go through each of the 5 species and the model results, and then some trends that we saw per species. But I want to talk about how we assessed our models first. So this top left model performance is going to be for the presence absence models. And we used a confusion matrix to assess the performance here. So you can see that for the coho salmon, our final model of presence absence correctly predicted 507 absences and correctly predicted 83 presences. And it incorrectly predicted 114 absences and incorrectly predicted 11 presences. The class error is telling us that the models were well balanced, with neither error being significantly higher than the other. But I do want to note here that we are over predicting coho occurrences with this 0.21% class error. And then finally, we looked at the AUC, or area under the curve, scores, and the AUC for coho for the presence absence model was a 0.90, and that is showing us that the model performed well and was able to predict most presences as presences and most absences as absences, and an error rate of 17% as well. And then for the remainder of the slide, all of the data will be representing the final results for the abundance model. And the abundance model overall performance is explaining 46% of the data variance. For biological models, the percent variance explained between 20 and 30% is considered good. Anything above 50 is considered very good. So here we're close to 50, so that means this model did well. And then the error rate, or the mean squared error of 0.03 is the difference between the estimated values and the actual values of the data set. So that's a good error rate. And then, sorry, the second table is the list of variables, and the from highest to lowest, they make up the final abundance model here, and they represent the predictive power of each variable based on its contribution to the model. And then the next slide, I'll pull out the top 4 variables, and we'll talk a little bit more about the trends we saw with those. So the plots shown here represent the relationship between the variable on the x-axis and the coho abundance response on the y-axis. And I do want to note that for our model of abundance, we log transformed the species data. And here on the y-axis, it's noted that it's back transformed. The ranges can be seen there to better understand the coho abundance probability. And then major trends we saw for coho salmon include that their abundance increased with increased Chinook abundances. They show up early in the year, and they prefer to show up when lower water temperatures are entering the stream. And then they also like higher conductivity. The Chinook salmon model performed well with an AUC of 0.88 for the presence absence, and for the abundance model, we explained about half of the variance. And again, the top predictors that made up the abundance model for Chinook are seen on the right here. Some trends that we saw for Chinook salmon in the Russian River estuary are that Chinook continue to show up 2 months after coho salmon, but they overlap in the early months. So they must be segregating by space in some way. And they prefer low stream temperatures with a gradual decline of with warm waters. The steelhead model performed well with an AUC of 0.80, and the predictability of the abundance model was about 32%. And then on the right again is the variables, and we'll talk about the top 4 variable trends that we saw. For steelhead, they prefer to be further away from the river of the mouth or the bar, the sandbar, and they show up early in the year, and they prefer stretches of vegetation cover on narrower segments of streams. The 3-spine stickleback, the model performed well. The abundance model had 55% variance explained. The variables on the right are the variables that made up the final model. Trends that we saw for 3-spine include they like to be far away from the mouth of the river. They prefer to be close to large stretches of vegetation, and they prefer cooler temperatures. I'm running out of time, so I'm going to just go through this one quickly. The model performed well. It had a higher error rate, but that might be due to high abundance numbers here for starry flounder. Trends that we saw for starry flounder is that they show up early in their year, and they prefer high flows with warmer waters. So overall, our models for presence absence performed well. All the AUCs were above 0.8, except for starry flounder. The abundance models, they also performed well. They explained overall a range of 32 to 55% variance. The error rates had a little bit more of a range, but we did have a wide range of abundance numbers, which impacts the error rates there. And even though the predictors changed per species, there were some patterns that are important to note. Our top predictors included temperature and chemistry at our upstream sites. All fish influenced were by the day of the year and the temperature as well. And in every model, we saw abundance of other fish species included. So there's some sort of interspecies relationship that we noted. And then location was important to most of the models as well. So where they were in the river, the width of the river, or distance to freshwater inlets. And then the bar status was very weak in the models. And this might have to do with the fact that the majority of the year, the Russian River estuary bar is usually open, and that is because they're required to manage that bar for steelhead passage. So some implications for management is, again, there's a little selective bar status on the fish assemblages. I'd be interested to model an estuary where the sandbar is not managed or manually opened. Managing for one fish may hurt the other fish for antagonistic relationships. Like we saw in coho and Chinook, it's assumed that they share spaces. And freshwater inflow and day of year are important for estuary management. And some considerations for this would be modifying inflows by changing dam operations if there is a dam upstream. And the next steps that we will be conducting is modeling other estuaries. We are in the process of doing that right now. We want to compare different size estuaries, so small and large. And then our data is similar between estuaries. We want to do a multi-estuary model. And overall, we want to use the models to create a framework that can be used by estuary managers in California. And that's going to be essentially called the development of the California Trout Aquatic Assessment or Species Assessment Tool, the ASAT tool. That's our end goal. And then I want to say thank you to everyone involved, California Trout, Sonoma Water, Paula Maladondado for her GIS work. And I want to thank everyone for attending this presentation.
Thank you, Michelle. That was great and really informative and interesting. As were all the talks, I was really I'm excited about new monitoring tools, be it actual data collection and modeling efforts. So we are going to, I'm going to look at questions. I think we have a few coming in. Let's see. All right, we have a question from Terry Fleming, and I think this question would probably mostly be for Kevin and Jan. What is the relationship between the monitoring effort and CRAM? Do they work together in a level 1, 2, and 3 framework, which I think you kind of walked through, but I'll pass it over to you guys.
Yeah, so absolutely. Well, I mean, we're implementing CRAM at all of the estuaries. So CRAM is one of the monitoring protocols we're utilizing. As far as level 1, I mean, that's really getting at, which estuaries are we focusing on? So we've got our MPA boundaries, we've got maps of habitats within those estuaries. And then level 3 is many of the other protocols that we are implementing. So the beach seines, the crab traps and whatnot. And they are really working in harmony, let's say, to come up with an overall assessment of estuarine condition across this MPA network, as well as the reference sites and hopefully additional estuaries that we hopefully get to utilize these monitoring protocols in. Jan, you want to add to that?
I just wanted to add to that one of our other big focuses is on trying to manage all of the data that we're collecting with our monitoring. And so for OPC, our data will be going to data 1. But we're also working on building the data infrastructure to be able to assimilate this data and make it more digestible for users. So a more user-friendly interface that we're working on, data templates for the data to be entered and then QA/QC'd. And so a long-term goal would be able to have it easily walkable from level 1, 2, and 3 with these data structures that we are creating.
Thank you, Jan and Kevin. I think that would be really useful for potential, management users or even regulatory entities reviewing these tools and the data that are collected. You talked, I want to ask Jan and Kevin, just as a follow-up, you talked a little bit about SOPs that you are developing. What is the timeline for development of these SOPs? How much data do you need to collect to kind of pull this together? It's a little bit of a logistic question. As well as, sort of your outreach piece.
Well, first, I guess I'll go first, Kevin, and then you can fill in some gaps. A lot of our SOPs, so we're building, Kevin mentioned that we're leveraging a lot of existing work. And so the SOPs that we pulled together were built upon a previous EPA WIPDIG, Wetland Program Development Grant, for those that are acronym crazy like I am. And so we're building off a lot of those SOPs that were developed as part of Santa Monica Bay's foundation. And so those had been in works for about 3 years, and then we took them and then we've been developing them and testing them in the field. So I would say they're close to ready to go. That's what we're testing now. And so that's what we will be reporting back to OPC. Kevin, you want to add to that?
No, I mean, that's yes. Jan, yes. There you go. We didn't want to reinvent the wheel. We didn't want to create new SOPs. So because EPA had already invested in California developing level 3 monitoring protocols and SOPs for estuaries, all we did was take those and start testing them out at additional estuary types, so the barbel estuaries and whatnot. And it seems to be working pretty well so far.
Yeah, so we're testing them and then we're linking them all together, right? So we're putting them into a monitoring framework, while previously they were just standalone SOPs. So now you can use them together and then link them to these ecosystem functions, which really are the groundwork of the monitoring program.
And will you be using the California Wetland Monitoring Workgroup as an outlet for this tool?
Oh, yes, of course.
That is definitely our next priority, is to connect with people and disseminate all of this information. The State Coastal Conservancy just received an EPA WPDG WIPDIG grant to start to help bridge the gaps between all these different organizations and some of the frameworks that we are creating. So hopefully that can be kind of another avenue to get this information out. We definitely want a transfer of knowledge for sure.
Yeah, in year one, so we're at the end of year 2 right now. So year 1 was really pulling together all of our thoughts, pulling together the management advisory committee, pulling together all the SOPs. Years 2 is going out and testing them in the spring and the fall. And then year 3 is working up the data and conducting a lot more of the outreach to the various organizations. So it's definitely coming, Jen.
We look forward to hearing more about that. Thank you, Kevin and Jan. All right, we have a question now for Michelle. It's from Sean Melindy. Great talk, Michelle. Do you think your model can complement presence-absence models of fish higher in the watersheds?
Hey, yeah, thanks, Sean. Yeah, I think this model is extremely flexible because it's a random forest model. You can kind of pick what environmental variables you want to put into and what fish species you want to represent. And I think because we're doing it kind of over a range of different sites, if those sites moved higher in the watershed, it would tell us different things. So I think that you could either completely remove the estuary aspects and move it up the watershed, or you could just continue with sites up the watershed and keep the different parameters such as upstream river length. And that would really tell you a lot still. I think so. The fish we chose specifically were for indicators of estuary, or ones that were important to estuary management. Yeah, you could always change the species or the environmental variables.
Michelle, can I ask you a follow-up question from that?
Yeah.
You primed the model with just Russian River data, right? So
Yeah.
what do you think is the spatial extent of your model, right? Do you think you could apply these models to other estuaries? And then what constraints do you think are where you can apply these models, right? Like obviously this probably wouldn't work in Southern California because we don't have any of those fish or a lot of those. So where do you think your spatial constraints are?
That's a good question. And that's definitely something that I'm in the process of learning right now. Because we started with Russian River, we had a great data set for Russian River. That's why we started the model there. And we're in the process of modeling other ones. So we're going as far north as Redwood Creek. And then we're hoping to eventually model all the estuaries in California. So the spatial extent is still something that we're working out. We're hoping to eventually create a model that is able to represent every estuary, and that would be kind of used in like a platform where you could pick which species that you wanted to, fish species that you wanted to highlight, and then it could tell you what management operation would impact them the most. So it'd be interchangeable, if that answers it. And then the fish species we chose were important to the Central Coast, but we're going to change the fish species based on going more north or more south. Yeah.
I think that's a really good question regarding the connection with the upper watershed and you know, using that model in those upper areas. I would like to make, along those lines, I'd like to make a plug for the Russian River Regional Monitoring Program that's under development, Michelle. So I'm sure you might be aware of it. So that would be a good place to kind of make connections with that program as well. Obviously, they're looking at a lot of water quality variables, sediment, temperature, which would affect fish assemblages. Rafi from Scorp asked the question, Michelle, do the data collected by Jan and Kevin's projects enable them to use Michelle's model? Actually, that's a question for all three of you.
Yeah, I think so. Right now, my model is specific to fish species and estuaries, but the more data, the better. Right now, some of the estuaries that we're trying to model, we need some more data. So there needs to be some sort of standards for how people monitor fish in estuaries, because that's an issue that we're running into is data gaps, unavailable data. So yeah, we could definitely use more data and standards.
Yeah, so we are collecting all of the abiotic water quality data that you have, and then of course we're fish seining, so we have that data as well. I think this is a great question in the sense that you know, all of our protocols and monitoring framework are in like our drafts right now, and so if there are specific groups that need specific information, making sure that we are collecting it in a way that would allow them to use it is definitely important information that we need to know. So, Michelle, if you have anything specific that you think would help us or we would help you, like, please reach out.
Yes, you guys should get together. We have another question related to management of these systems. And, you know, as Michelle pointed out, and I think you did too, Jan, these systems, these estuaries can be naturally or human opened, breached, if you will. And particularly along the central and southern coasts, there are a lot of management actions that require, you know, regulatory permits, and that those kinds of regulatory analyses require information, you know, standardized information on the health of these estuaries under those conditions, under those management conditions. And so this, one of the things is there good information on when the bar is opened, which I think Michelle touched on. Seems like that would,
Oh, that's my fault. All right,
Kevin, you have now appointed yourself to read the question.
Okay, it just disappeared. But basically the question was, is there good information on when these breaches occur? And I would say if it is a regulatory breach, then there would be information through the various regulatory agencies when the breach cannot occur, and you know what time of year and what restrictions or considerations need to be put on that breach. If it is a non-regulated breach, then no, there isn't like some live location where all estuaries have water level data that is being put up to watch. But we have 20 or so of these barbel estuaries gauged along the California coast. We've deployed pressure temperature loggers that we go out once a year in exchange, or we work with state parks to do it for us. So then we can see when the breaches have occurred. We don't know if they are natural or unnatural. I did post a link to a document that I drafted with John Largere at Dega Marine Lab a couple years back with funding from NOAA on considerations for the management of the mouth state of California's barbel estuaries. So it goes through a whole synthesis of the processes and phenomena going on in these systems, as well as the potential impacts of anthropogenic breaches to the estuaries. Did that link show up? I hope it did. So I encourage people to look at that. It's got various appendices, the main document, and then various appendices looking at the data compilations we put together for Russian River, Scott Creek, and Los Penasquitos, as well as a list of sites that have permitted actions on them and some of the special conditions that we put on those permitted breaches.
Kevin, why don't you, I'm going to throw you here under the bus again. Will you also maybe mention the JPL project where we're trying to link and monitor some of these opening and closing events that you and NASA and Eric and all those important people have been working on?
Yeah, so over the past 2 semesters, we worked with the NASA JPL group called Develop, and they have built an online tool that is meant to predict breaching events in barbel estuaries along California, as well as some water quality and parameters. And It's still in development phase, but we shared a lot of our temperature depth data with them for various systems along the California coastline, and they kind of put together a predictive model based on aerial imagery, because the whole point of develop is how can we better use satellite imagery to manage whatever it may be. So we said, well, we want to better manage our estuaries. They went and found a bunch of satellite imagery on various coastal estuaries, zooming in on the ones that we were interested in, and then linked the aerial imagery with the temperature depth data we have for those going back 3 or 4 years, and came up with kind of a predictive model of breaching events. And once that goes live, we can share that with the estuary managers in California. It'll be a great tool.
Very, very exciting stuff. Can't wait to see the results of that as well. So no pressure, Kevin. I have one last question. I don't see any other questions from the audience, but I have one last question on level of effort. And this question really is for Kevin. You talked about 3 days to perform this monitoring for the framework that you are developing. And you also talked about modularization, potential, making this a module type of thing, depending on your management question. How modular is that 3-day, sort of level of effort if a regulatory agency wanted to require that somebody take information related to collect this data? Is 3 days the minimum, or can you cut it back a little bit? Can it be more modular in that way?
Well, I would say 3 days is kind of the minimum for the 3 stations that we've set out in implementing all of the SOPs. Now, what Jan was getting at with the link back to the functions of the estuary, if a manager is only interested in a specific habitat within the estuary or a specific function of that estuary, that then kind of culls the number of SOPs that you would want to implement, which would reduce the overall time needed to do the monitoring effort. I would say that in a larger estuary, the difference between Drake's Estero and Limantour versus Carmel River Lagoon or even Arroyo de la Cruz. Arroyo de la Cruz is so small, we only did 2 monitoring stations because there was no way we would have put in a 3rd one. But Drake's Estero is gigantic. So in those larger systems, you might need more stations and a little bit more time. But it all goes back to if you're implementing all the SOPs as we are right now for the MPA monitoring effort, or if you're reducing those to get at a specific habitat type or function within the estuary. Does that answer your question?
Absolutely. Very thorough. Thank you.
I will add a caveat that it really, and Kevin mentioned this, that it does depend on your estuary size. So our Southern California team, and it depends on the number of people you have. Right?
Absolutely.
So our Southern California team now has cut it down to 2 days because we have smaller systems. So we can do Malibu Lagoon in 2 days. We can do Galuda Slough in 2 days. But it does require you to shift things around a little bit. Like if anything requires 24 hours, then you need to hit like a traps to require 24 hours of deployment. Then you need to hit every single station in 1 day. And just, you have to kind of change how we've prescribed it in the framework, but it can be done in 2 days. Right now, Eric and I at SWRP are also testing this framework down in Baja California, Mexico. And so those systems are really different. There's a lot of more logistics in terms of how you're going to get to every site in terms of boat. But what we found is that 3 days is really can be the standard. And then depending on personnel, how much money you have and things, other logistics, you can kind of shorten it or expand it around that.
Yeah, that absolutely makes sense. And I think, as you lay that out in your guidance for use of the framework, you'll be able to kind of elucidate that for, users. So thank you. I have another question for Michelle from the audience. Great talks, first a comment. And then the question is, do you think that your findings on minimal effects of bar status on fish assemblage suggests that bar closures do not delay coho and steelhead ocean entry timing?
Hey, thanks for that question. I want to just point out first that I'm not a fish expert. I'm good at modeling, but I don't know as much about fish as probably some people here. I do not think that suggests that there's minimal impact on steelhead and coho entry. I think that the Russian River estuary is well managed. I think because they're required they're required to have access for steelhead migration. And like I pointed out, the majority of the year their bar is open, and I think that usually it's manually opened. I think that bar, we were initially thinking that bar closure would have a really big impact on fish, right? It changes so much of the water chemistry, and they're required to go from the ocean upstream. And if they can't do that, then that should have a really big impact. So I'm hoping to get better bar data from estuaries that we continue to model and see how that plays out moving forward. But I would expect it to have a big impact. We didn't see it in this model. But again, we'll see how the other ones go.
Yeah, and for better or for worse, Michelle, there's lots of estuaries that you could test that on that are non-naturally closed.
yeah, I'm excited to see the comparison. So stay tuned. Yeah.
Information.
Mm-hmm.
I just wanted to say thank you to the presenters. This was super informative and enlightening, and I again just can't wait to see the results of all the good work you guys are doing and the connections that you're making. So with that, I'm turning it back over to Allie, I think.
Thanks, Jen.
Thank you.
Yes, thank you all so much. That was an excellent discussion and really great questions posed, and kind of from the swamp statewide perspective, have me thinking about what this future framework might look for the statewide bioassessment program if we had $1 million or 10 million, what have you. But I'd love to follow up with you guys and kind of pick your brains and talk more on that front too. That said, we've got our lunch break. At this point, that time is not right. It's actually 12:15 to 1 p.m. And I'm going to hand it off to Angela just to give you a little teaser and next steps transitioning over to the workshop this afternoon. This is kind of it for me as the main face and host, and Angela will take it over from here. But I just wanted to quickly express my sincere gratitude to all the participants, the panel, the presenters, the moderators, all the newbies that showed up. Thank you so much for your participation to date, and hope to see you all at the workshop. Angela, handing it off to you.
Thanks, Allie, so much. So everybody can go have your lunch, but come back because we're going to have more meeting after this. So we're going to have a special session by CalSFS for tribal traditional ecological knowledge. And that'll be followed up by a panel where we have some questions for our panelists, moderated by our fellows, and then we'll have lots of time for audience questions and discussion. And this is where we're really going to get into the meat of learning how we incorporate this TEK into all of our research project management projects, and hopefully provide a tool for any future freshwater scientists that'll have a legacy for us as we're going forward, managing freshwater systems into the future. So just to give you guys a little tidbit, we're going to have a talk by Sarah Ryan about Clear Lake Tules and beyond, collaborative tribal beneficial use development. We'll have a talk by Keith Parker of the Yurok Tribe of California, and he'll be talking about fisheries management and tribal ecological knowledge in the Yurok Tribe. We'll have a talk by Anne-Marie Mendoza of UCLA, talking about bursting through concrete, indigenous water planning, and Tova Gengar. And I definitely pronounced that wrong, and I'm going to make sure I correct that when I talk to Anne-Marie. And then lastly, we'll have a co-presenter talk by Dr. Beth Rose Middleton from University of Davis, California, Ada Stevenson from California Indian Water Commission, and they're going to be talking about building understanding across worldviews to increase recognition of tribally specific beneficial uses. And then we'll have a panel, and we'll get to talk to all these panelists together, and we're very excited for that. And this is put on by CalSFS. And yeah, if you have any questions about this, you can always just put that in the question answer. And I can type that response to you over lunch. I'll be here. And we hope to see you guys all in about a little less than an hour, 1 o'clock. We'll start promptly. I think that's it. Did I forget anything, Allie?
Nope, I think you covered it all. Thank you.
Okay, great. Thanks so much. See you guys back at 1.
Bye.
Hey, Angela, are you there?
Yeah.
I just wanted to make sure you're co-host. I think she has to accept. Did you get it?
Yeah, it looks like I can
Oh,
I have.
Ew. Sorry, I'm being dumb for a second. You got it?
Yeah, can you hear me?
Yeah, sorry. My volume was down. Okay, cool.
Do you need something to eat? Go get something to eat. Go get some coffee.
All right, I'll be back.
Okay, cool. Me too. Okay, so it's 12:55 and we'll get started in 5 minutes or so. Thank you, welcome back.
Hey, Angela, I stopped sharing, so I can, do you want me to give you controls or you got it under control?
Let's experiment
Okay.
here. Just a moment. I think this will have to do.
Looks good from my end.
You can see that? Okay.
Yep.
Okay. So we've got 2 minutes before we will start the session, the CalSFS session. Okay. Okay, welcome everybody. This is the last part of the CABW CalSFS meeting for 2021. So right now we're going into a special session with CalSFS for our traditional ecological knowledge. My name is Angela DePalma-Dow. I am the chair president of California Chapter Society Freshwater Science. And during every annual meeting, we try to do a workshop or a special panel, something that provides a little bit of extra information or a tool or a resource that's valuable for our membership. And this year, during some surveys, questionnaires, interactions with our members, they expressed an interest in having a traditional tribal ecological knowledge session. And we really wanted to do it in person and have some hands-on demos, building some tule boats, maybe some basket weaving. However, with COVID, we weren't able to do that. We are still planning for that in the future. When we have this meeting, we'll be downtown Sacramento. And so we are still planning for some sort of those hands-on things in the future. But for today, we have special session speakers. So we have 4 speakers today. I know the agenda and the schedule says that we are going to have 5. Unfortunately, Keith and his whole family are not feeling well, and he's not going to be here today. Our thoughts are with Keith and his family. That can be very rough, and so totally get better. Do everything that you can to get better, and then we will have just the speakers in the same order. So we'll start off with Sarah Ryden from Big Valley, a band of Pomo Indians. And then we'll have Anne Marie Mendoza from UCLA. And then we'll have co-presenters, Dr. Beth Rose Middleton from UC Davis and Ada Stevenson from California Indian Water Commission. And so the way this will work will be the first part will be the special session. And so each of the presenters will present on their field of research, field of management, some projects they're working on, things of interest to them. So we and the audience can get to know the speakers a little bit. And then the second part of this special session will be a panel question answer with some discussion. And that part will be moderated by our CalSFS fellows from this year. So today we have Robin, we have Will, Kyle, and Melissa. We'll be asking questions of the speakers, of the panelists, so we can start this discussion and learn some things about traditional ecological knowledge, particularly for our membership at CalSFS. We want to provide some research education so that going forward, our future freshwater scientists can work collaboratively with tribes, alongside tribes, when collecting data, when working in cultural resources. And so we want to make sure that we're establishing those relationships now and providing this panel as a tool, as a resource for our membership. All right, so again, we will be having the presentations first. There are going to be about 12-15 minutes. We have a little extra time now because Keith will not be speaking. So we will allow some questions during the first part. If you have any questions about the speaker's research or what they're working on, you can go ahead and put them into the question answer box below, and we'll be moderating that. Some of these questions we'll answer live. Some of them might be really juicy. We might want to hold on to them until we get to the Q&A discussion panel a little bit later. And some of them, maybe the speakers can answer or elaborate on types. We will take all sorts of questions and comments, and anything we don't answer during the speakers portion, we might carry over and answer later into the Q&A discussion. That's what that's for. But we will allow some questions right after the speakers talk, if it's related to their work and what they talked about. All right, so once again, welcome to CalSFS, and this will be our special session. I do want to plug some things about CalSFS first before we go on. We do have a Twitter account, and we are going to be tweeting throughout the session today at CalSFS Social. Feel free to follow us. We always post any opportunities such as students travel awards, student opportunities, as well as fellowship opportunities through our chapter, as well as the parent Society Freshwater Science, the full society. And if you're interested in joining CalSFS, really easy to do that. If you are or have been a CalSFS member or a Society Freshwater Science member and you're renewing your membership, whenever it comes up for the renewal, there's a little box that asks if you want to join any chapters, and make sure you just check the California chapter box. And if you haven't done that, or you just want to join the chapter, you can email here at cal.chap.sfs@gmail.com. And between myself or John Olson, he is our Treasury Secretary, he will be able to get you connected to be a member. And some of the benefits of joining CalSFS is that we provide student early career resources and opportunities, also networking, professional growth. We have mentoring, information exchange, both with the Parent Society as well as other organizations. One year we had a session at American Fisheries Society in Reno. We were able to interact with American Fisheries Society students and other members of that organization. There's also opportunities if you're a member of CalSFS to have direct input on local events and outreach opportunities. So if there's something that you want to see in your local club or your organization and you feel like CalSFS would provide opportunities for that, this is a great way to connect and be able to make those opportunities realized. And then there's lots of other benefits I can't list here because that would just go on and on. And if you want to support CalSFS too, so we can provide some of these resources for our students' early careers, you can directly donate to our Venmo here at CalSFS-John.Olson. He is our treasurer. Or you can also email him to donate directly to him in other ways if you don't have Venmo. And again, the donations provide things like student poster awards, student travel awards, things that are useful for our memberships to grow, to expand, and so can make sure providing opportunities for those that might not otherwise have them. All right, so this is the breakdown. We're going to go into the first part of the session here. And I just want to make sure that our first speaker is here, and that is Sarah. We are like a few minutes early. So Sarah, oh, I see your face. Great. Okay. So again, this first part will go until about 2:15, and then we'll have a little break just so we can set up the panel. And if you have any questions, we can, put them in the box at any time, and we can touch base on them again during the Q&A, unless you have a question specifically for the speaker and some of their research or some of their projects. And then we'll have some time after each speaker for them to be able to address audience questions directly. And yeah, questions in the Q&A box. And I think we're ready to go. Let me make sure I'm not missing the next. Nope. Okay. So Sarah, I saw your face. And I also just want to make sure that, yeah. Okay. So we'll have you, I'll stop share here in a second. So this, our first speaker is Sarah Ryan. She's from Big Valley Band of Pomo Indians. And her talk is going to be Clear Lake Tules and Beyond, Collaborative Tribal Beneficial Use and Development. And Sarah works around Clear Lake, which is near and dear to my heart, which is where I work. So little biased having you first, full disclosure here. But we both love Clear Lake. Sarah's been working there for a very long time, and I'm really excited to learn about her work with Tules. And, Sarah, we will have you come back when we meet in person, and we can have a demo on the Thule boat building. I'm really excited about that in a future year here. So I am going to stop share. And you should be able to share your screen. Awesome. I see it.
All right.
And sound check looks good. Okay, great, Sarah. Thank you.
Thank you very much. And you'll give a time check, right?
I will, but we have a little extra time today for all four of you guys. So but yes, I will give you a time check probably in the chat.
Wonderful. Well, thank you, Angela. And it's wonderful to be on this in this meeting and presenting on tribal beneficial uses and Tules and the work that is happening on Clear Lake. As Angela mentioned, the original plan was to have one of our elders here, who is also a Thule boat builder, to come and do a demonstration for all the participants, and to really give that visual and sort of hands-on experience along with a talk, because until you see Tules, until you see the plants and the animals and the lake, it's a lot of this is just words and kind of a description of what we're doing. So I really look forward to, and I know the elder who was really sad that this this wasn't going to move forward in person. Definitely looking forward to doing a hands-on thing next year. But today what I was going to talk about was some of our research on Clear Lake and the work that we've been doing collaboratively with other tribes and also agencies, including the one that Angela works for at the Lake County Department of Water Resources. What you see, the reason why this is titled Clear Lake Tules is because this plant that rings Clear Lake, or at least used to ring Clear Lake as thick marshlands this plant is so important to the Big Valley tribe that I work for, and I've been lucky enough to work and fortunate enough to work with them for the last 20 years. But this is such an important plant for so many reasons. It's consumed, it's used in ceremony, it's used for pollution prevention purposes, it's used for tule boats. And you see a picture here during one of our annual tule boat festivals of a little girl who's playing with and engaging with the tule boat during one of our festivals. But as I mentioned, today I want to talk about some of our programs and projects that we're doing on Clear Lake. Because this is a group that might be new to tribal programs, I wanted to give some background on that. We are going to, there's a lot that tribes are doing, a whole lot, and not a lot of it is necessarily shared with just kind of the general public. So it's good for you to do your research. If you're wanting to reach out to tribes, do your research and find out about that tribe. And when you approach a tribal government, it's always good to have some additional information about the tribe. So we're going to talk a little bit about tribal beneficial uses and some programs and projects that we have on Clear Lake. So just some background that there are 109 federally recognized tribes in California. There are still many more that are also petitioning to get recognition. And they are in most counties of California in terms of the federally recognized, but tribes throughout California have traditional lands. And here's a map of California with the traditional lands and language areas of the people. And you can see it takes up the whole state, of course, and the tribes, the tribal territories that you see on this map are very much still present. And where I am and where Clear Lake is in the Pomo area. So you hear about the bands of Pomo Indians, and that is something, there are Pomo tribes in Lake Mendo and Sonoma counties. So tribes are self-governed. Here is just some language from the Tribal Government Affairs website. But you don't, they're not a homeowners association. They're not an environmental club like the Sierra Club or something. They have statutes that support the work that they do with the federal government, and they are sovereign nations. So again, as I mentioned, when you look at the work of tribes and you look at the things that they're trying to protect, it is based on taking care of their people and their resources as a sovereign nation. Tribes have water monitoring programs. Not every tribe, that might not be the priority of some tribes, and some tribes it is. If you're getting any sort of federal monies, you have to have a quality assurance program plan, a QAP. So the data is something that's not only legally defensible, and that's important in the eyes of the funding agency, but it's also something that is used now more than ever in the reviews that are happening, the evaluations of the water quality in California. Tribal water monitoring programs can be funded with a variety of sources. I've listed some of them there. And their data, the tribal data that is supported by federal funds is put into the Water Quality Exchange, which is a federal water database. The state CEDN goes ultimately feeds into the Water Quality Exchange. So the monitoring programs are also used for tribes, of course, to assess their and evaluate the water quality based on the standards and the impairment thresholds that tribes have. But you can see that it all leads into a way to assess whether tribal beneficial uses and other beneficial uses of a water body are being met. With tribal uses, it's not like if there's a ceremony that's supposed to happen, it's not like they can just postpone it necessarily for 3 weeks until the water clears up. Ceremonies and uses, tribal uses, are site specific and time specific. And this is one of the reasons why it's so important at Big Valley and most likely other tribes as well to do water monitoring programs. So you have that information. In 2017, the State Water Resources Control Board adopted a resolution that includes tribal beneficial use and subsistence fishing consumption uses definitions. Here are the definitions, and they are now being integrated into basin plans, which are basically where the water quality standards live throughout the different regions of California. And these definitions were worked on for a number of years by tribes throughout the state to make it broad enough for tribes to find those ways to ultimately come up with some water quality standards to protect their uses. Right now, except in Region 1, there are no uses on water bodies in California that will protect tribal traditional uses. And that, as I mentioned, tribal traditional uses can look because they're site and location specific, they can look really different from what's currently protected. Oops, let me go back one. So we created a graphic. Sorry, we created a graphic. Hopefully we're moving to that. We created a graphic about a freshwater harmful algal bloom and how that the toxins can impact a variety of activities that are happening that are traditional cultural activities of a tribe, and how that toxin can be the different routes of exposure for the cyanotoxins. And just put any pollution that's in the center or put that in the center of this picture, and you can see that exposures to pollution is something that is can be much more extensive than what's currently protected for recreation. And as it lists here, ingestion of water, ingestion, inhalation of particles, or skin exposure. There's plants and animals that are being eaten that nobody thinks of. Like, for example, the tules. The tule is something that's consumed. There are also herbicides that are used on the lake that have been found in the tules of Clear Lake. That's something that we have communicated to the agency that is doing that herbicide application. They did do a risk assessment based on that, but knowing what pollution is out there and measuring for it is one of the ways that tribes can get a handle on and start to change that the management of a watershed and of a water body, because these uses should be protected. I want to go a little bit into our cyanotoxin monitoring program, cyanobacteria monitoring program. Big Valley Vanderpoma Indians and Elam Indian Colony started it in 2014 on Clear Lake when they realized that there just wasn't enough information out there about what the toxin levels were. We used a variety of sources of funding, and as you can see from these couple of pictures, it was quite concerning to see a lake that's blue and frothy with dead fish in it. That's something that we've experienced now multiple years in a row in various parts of Clear Lake. So again, to protect the uses of a water body and the tribal beneficial uses, we started this program, and we know based on our studies that cyanotoxins can impact tribal uses as well as other uses. So exposure is one use. Fish consumption, we did, and I have a couple slides on that. We did a cyanotoxin study on fish and shellfish on Clear Lake and found that many of them and certain species were above the guidelines for fish consumption. And that was at a sports fish level. Subsistence consumption is even more. There's 65% of the county gets their drinking water from Clear Lake. So this pollution, cyanotoxin, is something that impacts thousands of people in the county, including tribal members. And and particularly when you have cultural activities that include swallowing water, a deliberate swallowing of water, you have to be concerned about toxin levels. We started a task force, so in a collaborative manner, we worked with, and have been since 2014, working with local, regional, state, and federal agencies to stay on top of cyanobacteria and cyanotoxin issues, really support the research that's going on with outside agencies now, and also support the work that we're doing to try to limit what's causing the cyanobacteria, which is nutrients, both from the past that are now sitting at the bottom of the lake to things that are still coming in with stormwater. There's some pictures of a bloom that looks and smells like raw sewage. We identified some locations that were tribally important, as well as where some other activities occur to develop our cyanotoxin monitoring program. And we are sampling at more than 20 sites around the lake. We also educate the public, and by doing that, we're also helping the public understand why it's important to protect these uses and these plants and animals that are so important to the tribe. When you've got toxins and when you have an overabundance and a bloom of cyanobacteria, it's something that's impacting everybody. And over the years, with thousands of people that are reading our outreach, they've come to the same conclusion that we have to find some solutions to this and to be engaged. We also issue advisories to our members about toxin levels. We do have a lot of research that's going on about cyanotoxins. We've found that the health threshold exceedances are happening in certain areas, mostly the Oaks Arm. We have areas that don't really have toxin levels of any importance, and that's in the upper arm. It's rare when we see toxin levels that are above the health threshold. Here's a few slides about our fish cyanotoxin study. Again, it's to protect the tribal beneficial use of consumption. We collected 44 fish and 49 shellfish in multiple areas. And here's our cyanotoxin results. We found that we actually looked at fish from 4 different years and found areas where there were exceedances in terms of the health thresholds, in terms of season, in terms of species, and even not in terms of the arms of the lake, but we found that the higher levels were in the upper arm of Clear Lake, which is not where we're finding the toxins. And we're doing this because we want to know, is it safe to eat the fish of Clear Lake, especially the native species that are of importance to the tribe. Some other projects that we're working on, we got a Bay Area Council Climate Resiliency Grant where we've installed some time series sondes to have better information on Clear Lake and the water quality. We've done some work on fish kill observations and interagency fish kill protocols to inform our local partners about just to get on the same page when there's a fish kill in terms of tracking it and also communicating about it. We also have a couple of projects I'm going to go into, CalWatch and our wells in Clear Lake Hitch. Here's a few things from the Bay Area Council grant. We have a citizen science, and you can actually look this up now, citizen science, harmful algal blooms and fish kills. This is a free 3-part training. This is level 1 that will talk about eutrophication, harmful algal blooms, cyanobacteria, Big Valleys programs, and this is something that we're going to very soon start encouraging everybody to join if they'd like to. Here's some of the topics that are being covered in our Udemy classes. And this is where fish kills are being linked to our website through the iNaturalist program. And this is a way to get more people engaged in identifying fish kills. Our CalWatch program is a CDC grant that we're doing with Tracking California to look at the tap water. Again, the users of Clear Lake being drinking water users, as well as other uses. We wanted to look into what kind of contaminants people who are on their own private systems were being exposed to. So we communicated with the county and the California Department of Food and Agriculture to look at herbicide treatments on the lake, as well as, so we were able to do herbicide tap water testing. We've also, because of the cyanotoxin levels that were occurring on Clear Lake this summer, we had the highest value that we've ever had in terms of this liver toxin, microcystin. But we were concerned about our tap water participants, that all of the people get their tap water from Clear Lake that are on their own private systems. And so, and because we had seen some really vulnerable drinking water systems of these private systems, that 45% of them had total coliform present, 57% had cyanotoxin present, although 89% of the intakes had cyanotoxin present. We reached out to the public health officer, again, in a spirit of collaboration with our projects, and let them know that we thought that they needed to do a health advisory for these private water intakes. And so this is what you see on screen is an example, or is one of the press releases that went out, where we actually were able to help get fill stations for drinking water for these private intakes that were on Clear Lake in these 2 areas. And it's, you know, it's still, health advisory is still in effect, but it will be released shortly is what I understand. So in summary, just a brief look into some of the programs that we have at Big Valley. And just to let you know that there are tribes in, I have Northern and Central California, but Southern California as well, tribes throughout California have some strong monitoring programs because they're taking care of their beneficial uses and the resources that are so important to cultural and traditional ways. And when you look to tribes, remember this, you know, remember that they're watching, they're paying attention, and they're very active and engaged in protecting their resources. So remember that when, you know, whenever you can and always to look at the tribal programs and to look at tribes and bring them into the work that you're doing and help to support the work that they're doing, because we'll all be better off because of it. And thank you.
Great. Thank you so much, Sarah. Really great presentation. That's a great summary. You know, I've always, because we work on the same system and we sometimes attend the same meetings, I see little snippets of your stuff for that meeting, but to have it all together in a summary, it's just great to see all the work that's going on there. I did have a question, and of course, any of the attendees, if you have questions, you can put them in the Q&A box down there and we will get to them. But I did had a question because, so your work expands, you know, obviously beyond your tribe, but how have you felt received with the public, with getting trust in the public and all of the scientific information that's going out, and how has that been received? And is there things that you've had to do to establish more trust, or do you feel that the public has that trust for that information? Particularly right now with science, it seems sometimes is really questionable, and people are wanting to do their own research, even though, you know, you've done research for 20 years on this, so you would know what's going on and would be a trusted source. But how have you been navigating that with this breadth of research and data and concepts that you've been working with and being very familiar with, and basically the expert now for this system particularly?
I think it's a great question, Angela. And you know, part of why we've been really pushing outreach and education is so that the public can come to their same conclusions. We hope the same conclusions, that not only is it possible to have high toxin levels in certain parts of the lake and contaminants in certain parts of the lake and the plants and animals of it, but also that there's a need to continue monitoring. And you're right. What we found in the beginning was that people were like, what are you doing? What is this about? And it's taken a few years to really get to, I guess, convince them that this is the tribe engaged in some studies that are of importance to them. And you know, a lot of what I hear though from the public is, if they want anybody to be in charge, it's the tribes. You know, they see that this is not for profit, that this is not for you know, to try to sell something, but it's really to better govern the watershed. So I love the fact that people are that a lot of the feedback I've heard from people is that, hey, we're glad you're doing this because somebody needs to. So it's been a positive experience in the long run.
And that's a great reward, right? That helps to keep going day out when those days are really rough and we get those bad blooms, is getting that positive feedback. And people appreciate what you're doing. So that's great. We do have a question here. So are the vast range of parameters tested in the same sites along the lake? And then how are those sites chosen? And then is the monitoring done by folks who are indigenous? And have state agencies done anything with the data from the WQX yet? They're saying that they recognize that's a lot of questions. You can choose one or all of them. We do have some time because it is 1:30, and because we don't have Keith today, we have Anne Marie was scheduled at 1:45, and Anne Marie might bump you up just a little bit. So Sarah, I think you definitely can answer all those questions if you would like.
Yeah, sure. I'll definitely give it a shot. So the parameters that we're testing, so we have a Hawk Hydro Lab, and we are doing water chemistry at every single one of our sites. That's just our cyanotoxin monitoring sites. We also have stormwater sites. We have creek sites, we have lake sites that are protective of the tribal shoreline and whatnot. But we're always using our hydrolab and a fluorometer where, well, the hydrolab now has a fluorometer. So we're looking at phycocyanin and chlorophyll A, dissolved oxygen, temperature, pH, salinity, specific conductivity, resistivity. We have the time series sondes that we have fixed in a couple of locations on the lake are testing for most of those, including, yeah, I guess they're testing for most of those. So we do see that water chemistry is an extremely important thing to analyze. There are water quality standards associated with those, with the water chemistry. We have not done much benthic macroinvertebrate testing, and it's something I really want to include in our work. The sites are chosen for the cyanotoxin monitoring. The sites are chosen, as I mentioned, some of those are tribally important sites where there are activities going on. We reached out to the tribes of Mendo Lake and Sonoma counties to discuss and identify the sites they thought would work best for the different uses that are happening on Clear Lake from the tribes outside the county. And they settled on 3 sites that are all actually in the, well, they're in the Oaks and upper arms of Clear Lake. So the rest of the sites were chosen. We were looking at a variety of, to design the study and program. We looked at a variety of information and criteria, such as we wanted to situate some sites where tules were. We wanted to situate some sites near septic systems. And then also a lot of them are in public areas where it's easy to access and controlled by either the county or the 2 cities. The monitoring is done by, we do have Big Valley members who are on the within the environmental department. And so it's a combination of Big Valley members and staff of Big Valley. With Elam Indian Colony, their monitor at the time was their environmental director. They've had a couple of environmental directors since we started the program, and they are not native. We do have an internship that we've started at Big Valley for tribal members to be engaged in this, and we have been actually going down to the lake and looking at water samples under the microscope and working with the hydrolab. So there are some activities that are going on with that. State agencies done anything with the data in WQX? Yes, fortunately, this last 305B evaluation that occurred for Central Valley did look at the microcystin data from both Big Valley and Elam, and it looks like Clear Lake is is listed in the 305B evaluation or the 303D listing for microcystin. As well as some of our other data. So yes, the state agencies are using it, but it actually took years to kind of convince that to happen as well. I guess I think I've answered all your questions.
Yeah, good job. So we'll give you we got more questions in, which is fine. We have some time. So the next one is, if you have additional thoughts on the higher fish microcystin concentration that was in the upper arm, despite it having lower overall cyanotoxin concentration. So it seems kind of the fish had higher concentration in the part of the lake that had lower concentration, right? And is it potentially due to lower productivity in these areas and therefore less dilution of the toxins compared to the Oaks arm, which is usually, for those not familiar with Clear Lake, the Oaks arm is usually where the majority of we see higher bloom, higher cyanotoxin concentrations? Or do you think there's something else involved?
Well, it is actually specifically related to the fact that we were able to get a lot of shellfish samples in the upper arm, but not the lower and Oaks arms. And the shellfish had just a higher percentage of the shellfish had cyanotoxin levels. If we're able to get funding to do this again, we will be expanding on shellfish collection at both the Oaks and lower arm. We tried. In addition, we had some of our samples were older and therefore kind of questionable in terms of, whether they had the full concentration of cyanotoxins or not. But anyway, we did also share the data with OEHA and want to be able to continue this program of testing the fish and shellfish for cyanotoxins in the tissue and in the liver, and hopefully be able to come up with a fish advisory if it's warranted. We are also asking them to develop a subsistence consumption guideline, because right now their guideline of 10 nanograms per gram for microcystin is just for sports fish consumption, which is 1 meal a week.
Great, and we'll do another question. So great talk. Has anyone looked at how water currents in Clear Lake may influence the location and density of cyanobacteria? And just to give some scale, Clear Lake is a huge lake. So there are a lot of currents out there. And each arm, there's 3 arms, have their own kind of microclimate with wind patterns and stuff. You'll be on one side of the lake, you'll drive to the other lake, and it's like completely different, no wind, totally different environment. So just to give that backup for that question.
Well, yeah, and the lower arm of the lake is where the dam is. So water is releasing out of that, and it is kind of like a drain. We are looking at the trends of how the different arms are over time have the higher toxin levels. And the currents are actually being studied by UC Davis, I believe, in terms of how, you know, how the nutrient driver or how nutrients are being driven around on the lake. So yeah, there are some studies going on regarding currents.
Okay, great. And I have one more question before we'll go on to Anne Marie. Is how would this monitoring program be affected if the Central Valley Basin Plan included tribal uses? And I know this is something near and dear to your heart that you've been working on. So I think this is a great last question for you. And then heads up, some of the other questions that were submitted, I actually really like them for the panel. So we might save some of those questions and we'll answer the panel as a group after at about around 2:15, 2:30-ish.
Yeah, I think, you know, it's surprising, but monitoring doesn't necessarily occur on water bodies. I mean, there has to be funding for it. Somebody has to develop it. And I was really surprised to find out that even though there were water quality standards for Clear Lake, and there were all of these beneficial uses that have been adopted, it didn't mean that there was any monitoring going on regarding E. coli levels or pretty much anything else except for what the surface water purveyors were doing on Clear Lake. So would our monitoring program be affected if, you know, I think it would just be a little more supported, because beneficial uses should be protected partially through monitoring. But again, as I mentioned, it doesn't necessarily happen. And most water bodies in California are not monitored for cyanotoxins or anything else.
Great. Well, thank you so much, Sarah. That was a great talk. Great questions answered. I really appreciate all that you're doing here on the lake that I work with too, but also in general, the information that you're providing for projects across the state. So really appreciate it. And we'll get in touch when we can meet in person, and we'll do those Thule boat building workshops. I'm super excited for that, and I cannot wait to drive down to Sacramento with all those toolies that we get to put together. So thank you so much again. So thank you. All right, so our next speaker is Anne Marie Mendoza. Are you ready to go, Anne Marie? I see your face. We want to make sure you can share.
Yes, I'm ready.
Great, okay. So, and we are starting a little early for you, so that's okay. Take your time to presentation, and then we'll have a little bit more time for questions on your research or on your talk. So Anne Marie will be talking about bursting through concrete, indigenous water planning, and I pronounced this incorrectly before, so how do you say that?
Tovangar.
Tovangar, okay, great. Thank you so much. And it looks like you're ready to go.
Awesome, thank you so much. Special thank you to the Society of Freshwater Scientists for inviting me to this very important conversation. My name is Anne Marie Mendoza, and I'm a Gabrielino Tongval tribal member. And I also just started my 3rd year in the urban planning department at UCLA. My ancestral is now known today as what is known as today as Los Angeles. My people called this place, which is the Los Angeles basis Tongvangar, which translates into the Tongva language to mean the world. So the geographic space that you see below my name is the entirety of the Los Angeles basin, which is our territory, which includes parts of San Bernardino and also the Southern Channel Islands. When I first began my PhD journey, I really wanted to understand the functions of power within my homeland. Los Angeles is a lot of things to a lot of people, but it is seldom recognized as an indigenous space. And who gets to make the decisions about land and water projects is ultimately kept from tribal people and participation. So through my experiences as an organizer, then turned planner, and now a researcher, I realized that urban planners have a lot of power in dictating the visions for my homelands. And if I was serious about creating political change, I needed to question and interrogate planning as a practice and its theories. So the perspective I offer today is, documenting that journey that shaped my academic study. I am a Tongva tribal member who is going to school in their homelands, and I'm dedicated to changing the hearts and minds and values of the people who now call Los Angeles home. So as an organizer turned planner, that has shaped the academic study that I facilitate today. I have spent most of my time navigating institutions and engaging with city politics to understand and combat modes of exclusion that my local tribal nation faces within Los Angeles. We are a landless state recognized tribe, so we have barriers to our sovereignty that federal recognized tribes do not have to contend with. A lot of the work that I have been involved with is in advocating for Indigenous representation in water planning. And in my quest to build capacity for tribes to participate in water planning, in particular planning for the Los Angeles River, I've encountered many barriers. And I guess that's what ultimately inspired the name of my presentation today, Bursting Through Concrete. Concrete for me represents the tools of colonial planning that exerts power and control over land, water, and ultimately Indigenous people. My people are relegated to being a radical minority in our own homelands when it comes to planning the future of waters. Planning scholar Libby Porter has helped shape my ideas about planning and helped me realize that planning is not natural, but a cultural artifact of colonialism. Planning sees itself as scientific and rational, and it works for the generalized ideas of what public good is, but is actually political and social and reproduces relationships of power. Our traditional ecological knowledge is very much channelized and controlled, just like our rivers. So for me, the Indigenous water planning movement that is going on in my homelands right now is really trying to break through what the colonial planning process has thus been, which has been erasing and tokenizing our participation. On the screen to the top left corner, we have what is the infamous LA Aqueduct that enables us in Los Angeles to be physically and intrinsically linked to the Owens Valley, a region that was formerly named Paiute Hunadu, which translates in Paiute land where the water flows in Paiute. But after the city of Los Angeles entered the Owens Valley in the early 1900s, the water does not flow like it once did. And the lower picture on the bottom is what is now known today as the Los Angeles River, which is a sacred river for my people, and currently going through a vast revitalization effort. And both of these built environment planning projects will be discussed today. So today, I'm going to cover two things, which is establishing the methodology of working with tribes that I was able to really think through when I developed the Aqueduct Between Us, which is a 5-part social justice series about water in Los Angeles from an Indigenous perspective. And then the second part of my presentation will be about what Indigenous water planning is to me and how the first part of my research really shaped the second part, which is, how to really work with tribes in establishing visions for our homelands. So I did not grow up with the critical consciousness of where my water comes from. And this is pretty normal in Los Angeles. People, turn on the tap and the water's always there. So we have really no reason to question that. A lot of my research has shown that people, use media to understand it. Chinatown is still something that people believe shape the narrative of water in our homelands. People in Los Angeles do not have the connection to water and land like they need to or they should. And it wasn't until I went to college that I understood why this was the case. I was able to understand how settler colonialism functions in terms of erasing Native presence on land in order to make way for settler propaganda and values. And as you peel back the layers of historical erasure that we experience in Los Angeles, we come to find out that water infrastructure projects, are major sources of dispossession for Native tribes in LA. So as I was radicalizing myself during the time of Standing Rock, I was realizing that a lot of people in Los Angeles wanted to talk about water issues from far off places and didn't really necessarily even understand where we were getting our water from here. I had the privilege of hearing a Paiute Shoshone guest speaker come to one of my classes at UCLA and teach me the history of water in my own homelands. And what I learned from that guest speaking time was that Los Angeles helped carve out 3 reservations in the Owens Valley. They established the Big Pine, the Lone Pine, and the Bishop reservations, and which the water rights were never exchanged, and it was to be determined at a different time. I also learned that, the breakdown of where our water comes from, 29% from the LA Aqueduct, 57% from the Colorado River Basin. And, I felt very motivated during that time that people in my homeland need to know where their water comes from, because in Los Angeles, 2/3 of the voting population needs to pass any resolution to do with water. And I thought that, knowing the basics was the most important part. So as a very motivated undergrad, I organized Aqueduct Between Us, a social justice water symposium, and I invited cultural teachers, water coordinators, and activists from the Owens Valley to come down to Los Angeles and explain to people of Los Angeles about their history, about the current political problems being had with the LA Department of Water and Power. And, this was a really great event, but it was very eye-opening that it just wasn't enough. And that at this point, I decided to keep establishing my relationships with this Paiute community. And it was really great. But I didn't know at that time what this would all lead to. I just knew that I wanted to be a responsible person in LA using water. And ultimately, this led me to creating a film with my brother, who is a film student. And we worked on this project for about 2 years. And we had a total of 9 participants, 5 from the Owens Valley and 4 from Tongvangar, that came together to tell the history of water in Los Angeles. And it's in 5 parts. It's very digestible, short series of film. I thought it was very important that people in Los Angeles do not have a chance to learn about where their water comes from. And it's so important that Indigenous people get to lay out that history for them. So it is during these 2 years that I learned the importance of relationship building, which ultimately helps shape the research that I'm doing today. And, relationship building is not something we're taught in school. And working with Paiute tribal members, I've come to realize that elements of our relationship are directly influenced by our relationship to the land. If our relationship to the land is one of ownership and extraction and exploitation, our predominant social relation will reflect that as well. So if our relationship that we foster is one of compassion, generosity, humility, reciprocity, that our relationship to each other will reflect that as well. Making the documentary really opened up my eyes about methodology and how we should engage with tribal communities who are not our own. So I'm a tribal community member, but I'm also an outsider to Paiute. And collecting oral histories is something that isn't necessarily regulated by the institution. So I had to build in my own protocols of how to collect stories and to building these processes of consent, which ultimately has helped me shape the research that I'm trying to do today, which is establishing Indigenous water planning protocols. So like I stated before, for me, indigenous water planning represents a disturbance in colonial planning, which is extractive. It's based on ideas of development and ownership. And, indigenous water planning is the opposite of that. It is based on TEK for any given water planning project. And that is something that I was able to get from the participants of the documentary, how to prioritize that within our storytelling, and then hopefully to being able to prioritize it in the way we plan. What I really took away from my elders, some of whom have passed, is that water is not a resource, but a relative. And I am excited to think through in the future about how does that look when you use that as a basis of devising a river plan. I also learned through my processes of interviewing various tribal folks is that indigenous people have the right, but also the ancestral obligation to shape the water futures in their homeland. So people where I'm from in my tribal community, we're not even at that point where we get to carry out any type of projects or to test our waters or to even be asked what we want for our rivers. We're very much in this beginning stage of politicizing the rest of the people around us because we do not have a land base. So therefore we do depend on building out coalitions with people. And I think that's where a lot of my work is coming from, is how to make our message of our knowledge the main hegemony in Los Angeles. Through my processes working with the people, I also developed processes of consent, which I based on the principles of free, prior, and informed consent, which is something that is reflected in the United Nations Direct Declaration of Rights of Indigenous People. And I love the language within that document, because it says that Indigenous people have the right to withhold their consent for any action that would affect their lands, territories, and rights. And when I made this documentary, before I released it to the public, I made sure that all the participants consented to the ways in which that their story was told. And this is something that took long time to make sure that people really appreciated and also felt appreciated and felt that their story was being shared in the correct way. And oftentimes when researchers are working with tribal communities, there's no processes of consent that are built in. And I think that that's something that needs to be talked about and needs to be put forth when we're talking through these planning projects concerning water. And then a big anchor of what I think Indigenous water planning could be and should be is reciprocity. So with the project, the Aqueduct Between Us, there is a written portion that I did for my thesis where I try to break down the laws and understanding the various policies that affect the tribes in the Owens Valley and my tribal community out here. But, writing a paper wasn't necessarily giving back to my community. Not a lot of people are going to read through a thesis. So for me, the documentary was a way of giving back and to show that the knowledge that was shared with me wasn't just going to be for me and to help build up whatever I could be in the future, but to give back, because this is something that people ultimately wanted from the Owens Valley and from here, was to create visibility, because we are so erased from not only planning, but the histories of how Los Angeles is looked at. Developing protocols was big for the Aqueduct Between Us. And I feel within my future project, the Indigenous water planning, or developing Indigenous water plan, that developing protocols would be something that would need to happen in the very beginning and be able to take shape as the project developed. So protocols are very important, how to establish respect and the way we talk to each other and to prioritize land. And the last two that I learned through the Aqueduct Between Us and will be taking on to my next project is prioritizing building relationships that are not relegated to a one-time project. So oftentimes in planning, it's very specific to a one and done. And if we are reached out as Tonga folks in our homelands about water planning, it is very tokenizing And oftentimes people will come and ask us for like a picture or some kind of our traditional ecological knowledge. And then, you know, they disappear like Houdini. We'll never see them again. So that is something that I think Indigenous water planning really goes against, is that norm of just a one-time project, and that's that. And, you know, not even to hear back from the people that you're working with. That's something that Indigenous water planning wouldn't do. So we prioritize relationships that go beyond one-time projects. And lastly, we acknowledge and talk about power dynamics within our relationships. So with me, I understand that I wear a lot of hats. I'm a researcher, I'm an activist, I'm Tongva, but I do know that I'm also representing an institution, which is UCLA, and that I need to talk through those power dynamics with people that I work with. And I did that during the aqueduct between us. And I was learning as I was going, but now I'm seeing how powerful that is. And I need to prioritize that as I move forward with my current research, which is Indigenous water planning. So currently right now, my homelands is under, well, they're going through a Alley River Master Plan timeframe, which has been a 4-year planning process. And during this process, Indigenous people have been really erased from being able to shape the future of what river revitalization could look like. In this 4 years of planning, only one event was held to, I guess, listen to what Indigenous people wanted for the river. And it wasn't like a regular listening session where the county comes in and asks you questions. It was very performative, where we were actually on stage at the Autry Museum. And, you know, to me, that doesn't represent true engagement and true respect for our sovereignty, where we totally lacked any decision-making authority. So with the frustration that came with that 4-year planning process, and, you know, constantly being on the activist side of things, of pushing for us to be recognized, I saw the need to develop our own plan, where our vision and our way of thinking about the river would not be an afterthought or something to be performed out. And that's where I'm at in my program right now, is starting to think about planning for the Paimepa Heights, and to develop an Indigenous water plan for the Alley River, which its original name is the Paimepa Height, which translates in Tongva to the West River. And, you know, already that's putting the Alley River in relationship to the other rivers in my homelands, which a lot of people do not know. There's 4 sacred rivers in the Alley Basin, and the Alley River is just one of the channelized rivers that I focus on right now. And my current research is guided by 3 questions. What is the Tongva vision or guiding principles for planning for the Paimepa Height? How do Tongva community members understand their shared and communal responsibilities about the river? And third, how does the Tongva community build broader community support in Los Angeles to carry out their vision for their ancestral waters? So I know my presentation wasn't typical science one, but, you know, I wanted to share that we have so many political barriers to get to the point where we're able to do projects and to, you know, really be taken seriously as decision makers in our own homeland. So thank you for your time.
Thank you so much, Anne Marie. That was a great talk, and you're totally fine. The talk is great when it bridges those gaps between implementation, projects, planning, and the science, because we could have science all day long, but if we don't have the right planning in place, where we're bringing in all the community and all the partners, then it falls short. It doesn't mean anything. So I think that's a great point. We do have a question for you here. Oh, my audio is a bit messed up. How about now? Can you hear me now? Okay, so the question we have for you is, thank you for your talk and all the work that you do. And this question loves that you brought up talking about power dynamics. Do you have any suggestions for those that want to do that in their work but don't know where to start? I hope you're able to hear that. My audio is still bad, apparently. I'll try to fix it.
I'm sorry, I couldn't really hear that, but I'll look really quick into the chat. I'm sorry, I didn't.
I can repeat the question.
Okay, thanks again.
And it was for me, full disclosure. Thanks so much for your work, Anne-Marie. And I really love that you brought up talking through power dynamics. I think that's something that we need to normalize and do more. But I think for a lot of folks, it can be intimidating. And just wanting to get your thoughts on or suggestions for those, emerging academics like yourself, or even, agency folks or anyone in between, to start those discussions and make space for that when they really don't know where to start.
Yeah, I think that, in the future, there should be a mechanism requiring you to do that, so then it won't seem so awkward and unnormal. But I think because I have such great love and respect for my Paiute relatives and my own people, that like I put myself out there, there's no handbook on how to do these things, even though I probably will write something in the future about how to overcome that. But I think as long as you are prioritizing the health of the water and to really push yourself to become a good relative to the land and water, that things like that, our ego, our fear starts to melt away, because you start seeing yourself as a part of this bigger system, and that there's no room for that kind of thinking. It's about being humble and to have the humility to be like, you know what, there are these power dynamics, but I understand that what you have to offer is as valuable as to what I have to offer. And I want true collaboration in any project that I'm working on. And once, you come with a humble heart, you'd be surprised how many Native folks like really can see that and will want to work with you even more. And you could bypass a lot of the distrust, because I know the people from where I'm from, we're kind of afraid to work with people because we've been burned in the past. But as we move on to this era of planning and thinking, and really confronting the climate change, that we need to prioritize the relationship with land and water, and let that fear melt away. And I don't know if that's helpful, but that's just how I had to look at my interactions with folks.
Perfect. Thank you so much.
Okay, thanks, Anna. Does my audio sound better? I did have a follow-up question to that one you just asked, Anne-Marie.
Yeah, it sounds better now.
Okay, good. I did some quick spring cleaning in fall. Okay, great, thanks. So I had a question about that, because you did mention that you want to establish relationships and partnerships for the longer term, not just for these patchwork short-term projects. And it's really disheartening when you're working with someone and then they just like drop off the planet, or they drop off, you don't hear from them again, and you want to establish those long-term relationships. As someone that's worked for agency, and I'm not tribal, but we're trying to bridge these gaps and have these relationships on projects and just overall programs that will last forever. I sometimes feel like I'm bugging folks, or I'm being too insistent, or maybe they're just not interested, and I'm being annoying. And so sometimes, and again, this doesn't apply to every single person, and everybody just has different styles for working. And I feel like I'm veering into some of our panel topics, so you can have a quick response here so we can go on to Dr. Beth and Ada. But I'm always looking for assurance when I'm trying to reach out to tribal partners, future partners, or current partners, and just make sure that I'm still, extending that olive branch, not being offensive and not being annoying at the same time, right?
I think that that's a normal way to feel. I feel that a lot of people within my homelands, because we're a very small group of people that, maybe we're not as responsive as maybe, it would make you comfortable in being. But I think that it's so important that even if you do kind of feel annoying, that, you are showing that you're a good ally, that you are, gonna be on top of these things, even if it does seem like you're a little bit out there nagging a bit. But, I know for me personally, I love when our allies are keeping us in the loop, and maybe I can't respond back because, we're very much under-resourced. As long as we have the chain of information and emails that, you're going to help us when the time comes, and that, when we are at that impasse to, participate in a project or to have questions, that you are the person to go to, and you've shown that time and time again. So I would say don't feel that way, and to just keep doing the right thing and being persistent with sharing information with tribal communities.
Great, thank you so much for that. Thanks so much for your talk today, Anne-Marie. Really, it was great, and can't wait to dive into more of this in the panel here in about 20 minutes. So let's go on to Dr. Beth. I see you there, and Ada, you are here. Let me make sure I can. And then let's make sure we can get your screen shared.
Hello?
Yes.
It says I can't screen share while Anne-Marie is screen sharing.
Yeah.
1 out of 2 is not bad.
Sorry about that. All right. Make sure it gets in full. Awesome. Okay, great. I can see that. Awesome. So up next we have Dr. Beth Rose Middleton from UCD, Ada Stevenson, California Indian Water Commission, talking about building understanding across worldviews to increase recognition of tribally specific beneficial uses. And after this, we will be going into the panel, but feel free to drop any questions you guys have in the Q&A box here. And we won't take like a break, but we'll just take a transition after your guys' talk. So yeah, take it away. Thank you guys so much.
Thank you so much for having me and having us. I apologize for the typically long academic title, but it really gets at what I hope to talk about today, which is building understanding across worldviews in the beneficial uses context. My name is Beth Rose Middleton. I'm a faculty member in the Department of Native American Studies at UC Davis. I've been there about 11 years. And I'm Afro-Caribbean and Eastern European, born and raised in Northern California in Miwok homelands. So I just followed 2 amazing panelists. I really appreciate and acknowledge Sarah Ryan's knowledge about beneficial uses. She went over much of the context, so I'm going to focus more on the collaboration. And then Anne-Marie's talk was wonderful, really bringing to mind a lot of important aspects of collaboration. So I have been following the beneficial uses work for a number of years and teaching my classes about it. And just this last year, I got the opportunity to work on pushing for acceptance, adoption of tribally specific beneficial uses. And I'm very excited to work on this because I see it as a very significant environmental injustice issue due to very much this last point. Once a beneficial use is recognized, water quality objectives build on that recognized beneficial use. And if there isn't a recognized beneficial use, that means some people, particularly indigenous people and subsistence fishers, are left unprotected. So these are some of the examples of the statewide beneficial uses. And the fact that indigenous beneficial uses are not included here is an environmental injustice. As many of you know, I'm sure, EJ, the fair treatment and meaningful involvement of all people, it has specifically in respect to environmental regulations and policies, with both the distribution of benefits and harms and the ability to participate in processes that set environmental regulatory standards. And Indigenous environmental justice is specifically attentive to settler colonial structures, institutions, processes, practices, epistemologies that are constitutive of ongoing deep environmental injustice. So it's a different lens on environmental justice that foregrounds settler colonialism. And some of the ways this is expressed in this sphere are unequal exposures, unrecognized culturally specific exposure pathways, and generally lack of protection for Indigenous peoples within their homelands. So Sarah Ryan talked about some of the process. I have been watching this process, showing my students the hearings as tribal members advocated and tribal staff advocated for recognition of tribally specific beneficial uses. One of my colleagues, Fraser Schilling, in Environmental Science and Policy, conducted a tribal fish use study in collaboration with one of our students, April Negrete, who's Tamoic Western Shoshone, and others. And it set out to quantify tribal fish consumption throughout the state. And I'll speak very briefly, I got a timer going, so I don't take too much time, but I'll speak briefly on why this is a challenge, particularly in a university tribal relationship. But the goal of his project and also of the projects I'm working on is to address environmental injustice, particularly indigenous environmental injustice, and to protect the health of tribal members. His study was modeled after a previous study done by the Columbia River Intertribal Fish Commission, and we are drawing on his study as we develop our survey tool to use to advocate for adoption of tribally specific beneficial uses on waterways in Region 5. So coming from UC Davis, building this collaboration with California Indian Water Commission, I find myself really thinking a lot about navigating the research space. I think Sarah Ryan spoke very powerfully about tribally led research, monitoring, engagement with citizen science. So the question there that comes up is, what is the role for university partners? And I feel that I'm in a bit of a unique position in Native American studies. Previously, it has been engagement with environmental science and policy, fisheries sciences, toxicology around these questions. So I'm excited to kind of bring this dialogue into NAS and engage some of our students in this work. And also, how do we participate in supportively and respectfully moving research, the results of these different studies on toxicology and pathways, exposure pathways, to policy to protect people's health broadly? But these are kind of open questions in terms of the role of university partners, especially alongside and supportive of tribally led research. These are the beneficial uses that were recognized yet not adopted around the state that Sarah showed earlier. This is the project I'm working on with California Indian Water Commission and others in Region 5. So we're really addressing the fact that although these beneficial uses were recognized, they have not been applied. And so what does it take to apply them? This is a diagram that my student advisee who's in public health, Tamara Solorzano, made. And basically she's diagramming out the process as we are understanding it in order to engage with the basin planning process and advocate for the cultural and subsistence tribal beneficial uses. One thing that I added here that's really important is that the relationship building and communication should take place first and throughout this whole process, not only in one consultation period. And this really came out in our last meeting with the California Indian Water Commission and other partners, that this needs to be foregrounded. And we're also hopefully contributing to advancing how the process itself takes place. Just again, noting the importance of tribal specific beneficial uses. This is work by my friend, Dr. Kari Marie Norgard up at University of Oregon. And again, the way to insert, or a way to insert tribal values into the water quality regulatory process, but a process that's not without its challenges. These are some of the ongoing concerns. Who is doing the data gathering to support protection of tribal values and interests? If it's tribal staff and tribes, that's probably the best possible scenario. But what about situations in which that may not be possible? Is then a partnership appropriate with the university entity? These are some questions. One thing that is really concerning, I think, to me as well as many others, is the length of time the process takes in order to document fish use, fish consumption, and to do the fish tissue studies. All that time that takes place, multiple years, pollution continues, exposure continues, health disparities continue. And then also, once there's acceptance of the beneficial use and accompanying water quality standards to protect it, what about the enforcement? Briefly, these are some elements, a draft of our research collaboration ethic. Anne-Marie talked about humility. That's central for us as well. And this is a draft, I would say, that my students and I developed also for our Keepers of the Flame Fire initiative, which is separate, but some similar principles. And in this case, descriptions of an understanding of water and water life differ between traditional practitioners, university researchers, and state policymakers. So our team is focused on respectful listening to build a collaborative project. Reciprocity is key, and also the responsibility that comes with learning about the ongoing impacts on water quality and indigenous health throughout California and beyond. So I'm speaking fast. I don't want to take up too much time. This is my last substantive slide, but this is where we are in terms of building communication between California Indian Water Commission and UC Davis, researchers in Native American studies, fisheries sciences, and environmental science. So really finding the common terms by which we can communicate about the water, the fishery, and pathways of contamination. Developing a shared understanding of the scope of the project and our goals, engaging in some mapping of different waterways to determine where we focus, slowly developing a survey tool. And there are going to be multiple tools depending on who's doing the research, if it's a tribe, maybe a nonprofit, or us as a university collaborating with a tribal organization. We're in communication with State Water Resources Control Board to make sure we are generating the information that will feed into the process in order to get these beneficial uses adopted. So we're in the process of developing a research ethics agreement. I think that will be our key next step, kind of the terms of collaboration, figuring out which waterways we focus on, continuing to build relationships between our two entities as well as more broadly, finishing the survey or the data gathering instrument about fish consumption, gathering information, developing the report, and then we'll shift to the second phase, which is looking at the fish tissue studies on the species of focus. So that's where we are. And briefly, I hope this gave a sense of kind of the challenges, the opportunities, the scope of building this collaboration between UC Davis and the California Indian Water Commission. Now I'll hand it off to Ada Stevenson.
Do we have Ada? And we're okay on time. Thanks for rushing through that, Beth, but we'll be okay. We're flexible here in the CalSFS session, so we're fine. Want to make sure you guys get the time that you need. So asking to unmute Ada here. There we go.
Excuse me?
There we go. We can hear you.
Well, thanks so much for this this verbal thing. It's kind of ticklish, the sound of that. First of all, I want to talk about the public outreach, and we talked about the TEK panel. Public outreach seems to have failed miserably here in the state of California, in the United States, when it regards to federal agency wanting to provide outreach to tribal or indigenous communities. And we see that now. And it had just rolled over generational failure, I would say. And not only are we talking about tribal communities, we're talking about tribal families. In a waterway system, there may be even just one family or one person, and that does not negate the importance of water usage. So we want to stress that when we're going out into the land. And I would like to tell the scientists that are going out to perform these outreach works, just federally recognized tribes have two categories of government. One is
And I think you accidentally muted yourself. I'm asking. There you go. Okay. There we go. Just repeat the last sentence that you had.
Okay, I just want to let all the scientists and people that are going out and studying the field, that just because they contacted a tribal elected official does not mean in any way that is a traditional person or someone who is fishing and living off water and land, because they're two very, very separate categories of people. And I know that in order to be on tribal land, that you need to notify people because you would like to remain on land and waterways. But we have many neighbors that who don't believe or won't participate in outreach. And for many of them is BLM, CalSFS, is the Federal Forestry, Department of Water, Wildlife, Park Service. Not all of those are bad people. It's just that they don't understand the concept of consultation. They don't understand in general the beneficial uses of water, lands, and plants. So what's a better way to do that? Put up a no access land of a bar or something that we can't get through. Therefore, we don't have access to traditional gathering items. And like we would always say, that water does not belong to anyone or
Anna, we lost your audio again, but I think, okay, you're back. So again, just repeat the last part. We might be having. Anna, we lost your audio. Oh, there we go.
And I think that many things that the tribes have to overcome when we talk about wanting to study water and asking people to come in and assist them is that we have a great mismanagement of water here in California. We have illegal water diversions here in California, and we have reams and reams of paper water. So as tribal communities, we're usually left out of that debate and that selling of water. And like I said, it is dangerous for us to get out there. And so we want, when people come out to study or to be partners with them, we want them to be safe as well. And we talk about nonverbal use of the power of nonverbal and verbal words. Not all tribal lands during water discussions need to be addressed, but asking information about traditional water and tribal acknowledgement is pretty respectful, but allow that to be given by tribal consent. Do not offend us by telling us our own stories, but rather let us as caretakers divulge to you the story of our water. We need to keep our water and fish healthy. And what better way to do that than tell you the history of that water rather than be dependent on the 4 to 6 weeks you may be studying it. No one knows that water, that land, and those vegetations better than the people that live on them and the people that use it for subsistence. But together as a team, I think that we can build a layer of trust and responsibility, and we want it to be treated with respect rather than to become subject matter. And that's very common. I don't know how that happens, but we end up being the subject matter rather than our water. I also want to talk about some of the dynamics out on the land. Some of the people that you will run into will be elected officials, tribal elected officials, and then you'll have everyday people who are just curious, and then you'll have traditional people who want nothing to do with you, and they hold the most knowledge. But they have years and years and generations of mistrust built, and they know the stories. And we think of the visibility they receive. Most of the tribal traditional people who know the water and the land do not want to be pictured. They do not always want to be quoted. They don't want to be seen, although they hold that knowledge. And so that's why trust is so indispensable when you're out in the field. We talk about, in fact, there will be no equality between traditional practitioners and scientists. There never can be. And by the way, what is the congressional trust responsibilities of state and federal agencies and universities toward Indigenous people when they capture our information? Where does the information go? What database? Who has access and who has control or ownership of that information? Tribes that offer information about traditional knowledge want to know who holds that knowledge for the next 50 years, next 100 years. We're talking about water, long-term water planning, because it's going to affect us out in the field, but not the scientists that come out and visit and take samples and go back and become and write papers about it. We live here 365 days a year, 24 hours a day, and nothing can change our relationship between our water and our land and our fish and our trees. We see the demise of it every day. We see that recreational and agricultural and cattle folks seem to have the power, all the power here in California. And indigenous people are not even in the discussion. And those that come out, the scientists that want to come, they still want to study water and talk about the molecular structure of water and plants and its relationship. They really need to come out more than the few times that they do. And they need to come out in different times of the year. And they need to talk to different people in those times of year. And I think that you should take into consideration when you come out on the land that many of us attend ceremonies. We are not available to speak to anyone. We are not allowed to have contact with people on the outside. So that's very important for scientists when you get ready to come out. You know, all of us, we are, we don't have a man-made calendar. We, of course, we do for our work, but we go by a natural calendar. When the seasons change, we change. We have different responsibilities and duty. Therefore, that means our duty is responsibility to take care of the land and the water, trees, etc., becomes our priority. So please don't be fed when you come out and you come out in the wrong time. It's a very simple rule. It's unwritten rule between tribal people, but apparently it must be written for those that come out in the scholarly world. And I also wanted to state too that there are many
Ada, we lost your audio again.
It looks like her audio has been completely disconnected, unfortunately. We can try, I will work with her to try and get her back on.
Okay. Thanks, Anna. We had just like a couple minutes before we have to actually go into the panel, so we stay on time for the with the agenda, just to make sure. But I did want to see if there was questions. We did have a comment, so thank you so much for your comments. This is for Ada from an attendee, so thank you so much. Let's see if Ada's there. Okay, I can see is her audio back. Oh, I can see it, yeah. Ada? Oh, and she's gone. Okay, well, let's go into the panel, and I'm sure we'll have some discourse with Anna here, which I think will be great. And again, so if anybody in the attendees audience, and thank you, Dr. Beth, for your presentation, and just stay where you are. We're going to stay here as speakers. So let's have all of the panelists come back onto your screen, and then I'm going to introduce our fellows here as well. And they will be moderating this part of the session. So the CalSFS fellows, this was our very first year for this fellowship program. We got funds for this program through the SFS Long Range Planning Committee. We did just apply to continue this project. And so far things look good. So we'll be able to have another round of fellows next year. So if anybody's interested in getting involved in CalSFS and being able to design activities like this or participate, learn about CalSFS, or you have a project that you want to do, you think CalSFS would be able to help with, think about and consider becoming a CalSFS fellow, and we'll be opening those applications in January. And so stay tuned for our website, and I'll put the link in the chat box for everybody too. And I was going to add something else. So our fellows today, we have fellows, can you turn on your video? I can see you. Okay. Okay. So we have William Oda is our fellow. We have Kyle Leathers here, Melissa von Mayhauser, And we have Robin Lopez too. Great. Okay, so thanks, fellows. So you guys are going to be moderating this part of the session. And then I'll be interjecting if there's any questions or follow-up questions or points or comments from the rest of the attendees. And did we lose Ada? Hopefully she'll come back because I want to have her participate and provide and finish her sentence. There was some stuff I was on the edge of my seat and then the audio cut out and just like, what is it? So hopefully we'll get that back. All right, so let's go into fellows. We technically have 1 more minute. Oh, the clock just changed, so we're good. We're on time. So fellows, you guys are going to ask questions. Who is going first?
I believe that is me. And my question has already been partially answered in many ways by all of our wonderful panelists today, but we wanted to try and dig in a little deeper. So reaching out, building collaborations, speaking with tribal organizations seems to be a thing that all of us can and should be doing. And as I think it was Sarah pointed out, there are just so many different people across the state who you can be connecting with who have a deep knowledge about the local area. So what are things that maybe organizations like SFS and our California chapter can be doing to encourage our members to reach out and collaborate with tribes rather than sort of going it on their own or working through sort of the more traditional academic structures when it comes to researching local areas within our state? And that is open for anyone who would like to speak on the topic.
Well, I'd like to at least partially answer that question. Within the tribal EPA world, there are lots of opportunities to reach out to tribal governments, and locally there is well, I guess in Mendo Lake and Sonoma counties, the tribal environmental departments of the tribes, most of the tribes of that region, have been meeting monthly for well over 10 years on tribal environmental issues. We do collaborative projects together. We get speakers on issues of importance. We have different topics, like we have an emergency response, like sub work group. The tribes in Lake County have been meeting quarterly since 2001 on environmental issues. So that's one place that you could reach out to, is just to find out if, you know, as I mentioned in my talk, just, you know, do some homework and research about the tribes of your area and see what tribal organizations are out there. And then also, you know, checking with tribal organizations such as the California Indian Water Commission, the California Indian Environmental Alliance, Intertribal Council of California. These are all, those are a few tribal organizations that are, you know, working on various projects and are also kind of a clearinghouse for what's going on in that region. In Southern Cal, there's a Native American Environmental Protection Coalition. I'm just talking about environmental ones. There's also Chairman's Association, Central California Chairman's Association, Southern California Chairman's Association. So there are, you know, there's websites and activities that are going on, websites to check and activities going on with tribes. And then there's events that are occurring that, you know, might be appropriate to reach out to, you know, the organizers of those events. So I think those are, that's a good start, should be a good start.
If I could just add a little bit to that, thinking about that ethics slide that I put up, and also building on Sarah's slides with the maps, which I thought were really helpful, learning the context, you know, whose homeland you're in. And then as Anne-Marie was talking about the settler colonial context and the layers of institutions that are impacting tribal homelands and lifeways, learning about that brings the responsibility to then really think about your work, your scientific work, your monitoring, your studies, and who it's for, and where it is going and what it's attempting to do.
Well, those are both really thoughtful responses, and thank you for providing those. I think I'm passing it off to Kyle, who's up next.
Yeah. So I had a question about land acknowledgement. So I think Ada touched on a little bit in terms of, the lack of commitment that's often found in working with researchers or collaborators, where they don't take the time to go out there. Maybe land acknowledgement could be a first step. But I was wondering whether those words have power or non-power, and is it helpful or harmful to include tribal land acknowledgements in outreach or research products? And how do we make those efforts more impactful or meaningful if we do include them?
I've come from the land of tribal land acknowledgements that do not go farther than that. Los Angeles is pretty infamous of saying these things, but then not carrying out the relationship that goes along with acknowledging the history of our people, which, you know, caused the whole reason for having a land acknowledgement. What I do think is important is that within a tribal land acknowledgement is a step to beginning a relationship. So oftentimes, for example, Cal State LA wanted to do a tribal land acknowledgement, and they reached out to an elder, and they worked in collaboration with this elder for weeks at a time to actually come up with the tribal land acknowledgement. And within that process, a relationship was developed with the faculty, with the staff. And I think that was a very powerful move on Cal State LA's part to be accountable to the Indigenous people here now, and to have Indigenous people define what those acknowledgements look like. And from that relationship, now the university has an elder on campus, an elder in residence that could, you know, be a part of decision-making in the future. So I think as far as just developing these tribal land acknowledgements, it's very important that you're going to go down that route to actually talk to the people whose homeland you are devising this land acknowledgement, not to just come up with one yourself.
Either the panelists, other panelists want to add to anything? Any, no? Okay.
Sorry.
Yeah, sorry.
That was just really well said, Anne Marie. I think there's lots of, elements I think about with our campus in terms of land acknowledgement and going beyond the words and really considering deeply, our institution's impact and footprint and history, and then how we address that in present relationships and plans.
Yeah, I appreciate that. And I think you probably get different ideas talking to people that you couldn't find that's trying to do a Google search or what resources you could find online. So thanks, Anne Marie and Beth Rose. And then if anyone else has questions about my question or Will's question or different things throughout all of the talks, they can put those in the Q&A, and Angela will help answer some of those at the end. I think I am passing it to whoever is next on the list.
We heard from Anastita Agustinez from the California Department of Water Resources yesterday, and during many of your talks today, about how essential it is to incorporate tribal participation and tribal ecological knowledge into policy, planning, and practice, but all while respecting tribal sovereignty. And so I'm wondering, how can freshwater research and management efforts be more supportive of ongoing tribal stewardship and monitoring efforts and responsive to questions and data needs that tribes have, while also honoring tribal sovereignty and rights?
I'm sorry, Ada is not on, because she was making, I think, some really powerful points about where does the information go. And even when I think we as researchers or collaborators work with partners and construct agreements that recognize protection of Indigenous knowledge, that we also have to look at our institutions and our protocols as an institution that we fall within that may not be exactly the same as what we are developing with a community member. So making sure that, whatever agreement we develop is actually can be defended within our institution, I think is important too, because sometimes really good intentions may or may not actually protect Indigenous knowledge. So I would just say that the importance of developing agreements and making sure that you can back them up.
I think that's a really good point that you made about acknowledging those agreements. And I've noticed with just what's happening around Clear Lake that some of the discussions about the Clear Lake water quality and what's happening with the plants and animals here has been really sequestered with just like an educational institution. UC Davis is doing work, and we've had to work hard to get them to be, more open. Even though they've agreed to be more open, they're still building off of things that they're creating rather than the work that the tribes have done on Clear Lake. And that's hard to see because, the tribal cyanotoxin monitoring program is tribal centric. That we will go out there and do water samples before ceremonies on the lake so that we can make sure that the tribes have the information. They're asking us to do that. And so to be tribal centric means that, those things there's a flexibility and an awareness of why we're doing this program and how and when we're doing this program. So for new programs to come in where it's not only building, it doesn't even acknowledge that there is a program. We've had that happen multiple times. I just think it's important to know what's already out there, and tribes might not be sharing all of, they'll be sharing the information that they think needs to go out. I know with our water programs, our elected officials at Big Valley really want there to be an understanding of how the water is being impacted by water management and land management decisions. So it's important to acknowledge that there are tribal programs, and also there are tribal uses that aren't protected. I just want to give one example. I mean, just subsistence consumption is what. So we have a Superfund site on Clear Lake, the Mercury Superfund site, the Sulfur Bank Mercury mine, which Elam Indian Colony is right on. The people there are being impacted in so many different ways, not only from mercury, but and the methyl mercury in the fish, but also other mining contaminants. And to have cleanup goals in the TMDL that are based on sports fish consumption, and to be looking at things from the uses that are not even the tribal traditional uses makes no sense to me. It's not right, because all uses of the waters of California are supposed to be protected. So I think it's important to support tribes in their efforts toward tribal beneficial use development as well, because then you'll actually have in the management and planning of what the regions are doing, you'll have those tribal uses protected in it. Otherwise, you'll clean it up, but it's not even safe to eat one. It'll be safe to eat one fish meal a week, and that doesn't support tribal traditional consumption levels.
I wanted to follow up on Sarah, but I didn't want to if the other panelists had anything to add to that. I don't want to interject before you guys. Okay, so Sarah, from the agency perspective, as when I started here, and Sarah, you've been here for 20 years doing work on the lake. And when I came in, I worked for county, so we worked with the tribes a lot, but there was nothing structured that would tell me coming in new to the system to reach out to others that are doing monitoring, let alone the tribes that are out there doing monitoring. So I think there's something systematic that, I think Anne-Marie, you talked about it earlier, like it'd be great if it was just a requirement. So it's, in the list of stuff that agencies have to do, and then they can't get around it, they can't oversee it, reaching out and seeing what's out there and what's already being done for it to be considered. And I'm learning still as we go, and working with the tribes, learning a lot from the tribes and their monitoring efforts. And we make mistakes all the time. And, from that process of learning is, we can develop these relationships and work on these monitoring programs, and making sure as soon as something new comes across our laps, we're like, oh, let's contact Sarah and let's see what's going on if they have any information to contribute to this, or if they want to be involved in this, or any of the other tribes, we try to do the same thing. And it's just automatic now, where when I first started in this little county agency, it was not automatic. And there was nobody here to instruct me, this is kind of what you need to do. And so we're trying, you know, today's panel isn't going to solve all the problems, obviously, but if we can, you know, plant this seed and make sure that this is a consideration for future scientists. And, you know, when they start their careers and they're going, well, is this going to be on a tribal land? Is there tribes that are doing work like this already? What can we learn from them? You know, what's the plant list here that has tribal beneficial uses that tribes use for this restoration project? If we can start that now and making sure that that's being considered for those that are starting out like I did, in this little agency where it wasn't obvious to do that. And I don't know if that's very clear, but I just wanted to touch on what you said, Sarah. It's also great because you're very involved. So the feedback that you provide for someone like me that's still learning is awesome. And so I think that really helps too. And that's a testament to your program, your familiarity with the program, and all the work that you've been doing.
Looks like no
You're up, Robin.
It looked like no more responses to that last question. Again, anyone who's tapping in, please feel free to drop your questions in the chat and we'll help facilitate those. Yes, my name is Robin Lopez, one of the CalSFS fellows, tapping in from the Ohlone territory in Richmond, California. And first and foremost, thank you panelists for being here and for allowing us the opportunity to learn from you and to digest everything that we're hearing today and how we can move forward with that. This question, I think Anne-Marie and Beth kind of got on the surface level with Kyle's previous question about land acknowledgement. But I want to kind of push it a bit forward at this intersection of perhaps post-colonial theory and ecological knowledge, and diving really into the fact that many of our institutions that we operate with are alongside, they carry immense amount of responsibility. And so the question getting to, how do we hold these institutions accountable for past impact or effects that have holdovers to this very day? And what does it mean or look like to go beyond just the land acknowledgement and into critically building and sustaining relationship with the land and water? And I ask that also from a very biased and personal perspective, having connection to my indigenous elders and ancestors down in Mexico, where there is a very innate sense of connectivity to the land and water. And so I'm curious to hear from hopefully all our panelists, what did that look like? And in the scope of when we're working with institutions or state and federal entities.
Oh, I guess I'll go first. That's a very heavy question. I think everybody, especially all the tribes throughout California, could give you a different answer. And all I could really speak on is how tribes are treated in here in Los Angeles. We have a state policy that recognizes our sovereignty to be consulted on water projects, and time again within the city, that is completely ignored and disregarded. In fact, we are oftentimes just looked at as stakeholders, if we're even looked at that. So I think within the time frame that I'm thinking through and seeing what's going on in the ground and actually being a practitioner, I used to be a practicing planner. Now I'm, dedicated myself to really putting school first, is that I left city politics feeling very defeated, and that there had to be different ways to work outside of the institution in order to get some of our theories and some of our value systems to be recognized. And in a place like LA, I think where I really feel that it's important to fight this battle is one of cultural values and hegemony, and making sure that my people's worldview and values and relationship to land is something that can be digested by everybody who calls Los Angeles home, because we should be the main hegemonic force that is defining how people are interacting with land and water. Because I don't think the city prioritizes relationality. In fact, they prioritize development of the Alley River to bring in more housing, which leads to gentrification, which leads to homelessness. And indigenous values we're against bringing in that type of development. What we're really trying to prioritize is river revitalization, but we're kept from those conversations. So I answer this feeling kind of like I have to understand how the institution runs, but also knows that all the answers and solutions aren't in the institution, and where the power is with the people and influencing the people. And that's why I think that documentary work is so important. Right now, I'm working with the School of Ed to develop curriculum based off the documentary to educate younger people, and using the knowledge that I have learned from my elders to pass down to the younger generations. And I think that scientists need to realize that they could lend a helping hand with helping Indigenous people develop these curriculums, develop, I guess, mass media to help, influence people's relationship with land and water. Because the institution, as historically proven, doesn't value that and looks at sustainability not through the lens of an Indigenous perspective, where it's like, how can we bring up life? It's about economic sustainability and how could we, make more money and develop more projects. So long story short, I think that we have a responsibility to understand how the institution functions, but also realizing that it has its limitations and that we need to look towards our people and developing projects that are influencing, working class folks to be to think like Indigenous people. Sorry, that's kind of long-winded.
Thank you, Anne Marie. I feel like that was a really powerful response, and there's no reaction button here, so I couldn't do the little clap emoji. But you mentioned education and curriculum development. I mean, I see from being in Native American Studies, a lot of times when we are teaching the intro classes, we have a lot of students who are very surprised, never heard really information about settler colonialism before and how it's institutionalized. And I think that, we see that across all age levels. There's really a deep lack of education about the context in which we live in California in particular, but also throughout the Americas. I've been lucky to work with Wintun Elder Pam Gonzalez, who's been advocating for teaching homeland history wherever you are, teaching about Wintun history or Putwin history where we are in Davis and elsewhere, and why isn't that history offered? Because that lack of that offering is a foundation for injustice and exclusion. I also think there's, and this kind of reflects some of what Anne Marie said as well, there's a need for developing more of a culture of partnership, respectful partnership. I see within the university and our quest to do research, particularly at a public university that's in the public interest, but a lot of the culture is often around, developing new knowledge, publishing that knowledge, patenting it sometimes. It's not necessarily broadly incentivized or supported or infused with the ethic of local collaboration or collaboration with the people whose homelands you're working within. I see some movement to change that, particularly there's a system-wide advisory committee, Native and Indigenous Advisory Committee at UC now. There's an advisory committee at UC Davis, but there's still a lot of work to be done. Not only at the institution, but also with our funders who support research to ensure that, not only is it responsive to this kind of broad public interest, but very specifically in the interest of justice and in relationship to the peoples whose homelands we're within and who have been impacted by the development and operation of our various institutions. Thank you.
Thank you, Anne Marie and Dr. Middleton. Sarah, did you have anything to add as well?
Yeah, I guess from a different angle of, the state doesn't slow down. They're constantly, making decisions, making assumptions, and moving forward on policy. And one of the things that we've been doing in terms of some of the advisory committees that I'm on have to do with the agencies being more transparent about their work. And it's, you know, it's not the same. It's only one portion of what you're asking about. But I think it, you know, it's really important to know. I mean, the state is going to be working through the tools and the policies and the laws that they have. And so that's going to continue. And I would, and they don't, and it would be great to, you know, to get them to kind of redo, and they need to redo some of the things that they've based their programs on. On the other side of it, it's also good to, you know, for that to be transparent, for those tools to be available to tribes and to the public, and for people to be engaged. So you know, one of the things that we've done at CalEPA Tribal Advisory Committee is to have each of the boards, departments, and offices of CalEPA to do these deep dives into their programs. How do you find out about enforcement and compliance, and where do you find out where their monitoring data is, and what kind of presumptions do they have about and assumptions have they built into their programs? And I think that we should all be asking agencies and entities to unpack that so there can be better participation with it. Again, it's only one side of what you're mentioning, but I think it's important that it's, you know, the state continues on with its thing. All agencies are doing that, and they need to open it up a lot more. And when they start, you know, when tribes are engaging with them, and you know, tribal people are getting hired up into higher positions. I mean, the governor is a tribal policy advisor. I think she probably has a staff of like 5, you know, or less. But it's something that it's a chance to build on that, because it's so important to get that tribal voice in there, and to have tribal people be in the agencies themselves so that they can work through those ways to incorporate those values and needs and concerns and goals and objectives of the tribes in their programs.
Thank you, all three of you, for, you know, those very intentional and very thoughtful responses. And as you know, you were all answering it, it made me think of a comment and a question as a follow-up, in that kind of what Anne-Marie was alluding to is that, you know, folks who are of Indigenous background are not a monolith, and we should never be treated as such. In every region, every space, and every relationship, it's a different dynamic. And but that also intersected with something I used to hear from elders, and I've heard of recently again. Was a question is always, what kind of ancestor do you want to be remembered as in the work that we do? And it made me think, you know, as I'm hearing from all of you, I think Anne-Marie hit the nail on the head earlier, that there needs to be a culture of consciousness, a change in that. And it sometimes, I think folks wait too late, and who are in privileged spaces when they're in academia, and it needs to be something that happens much younger. When, you know, I'm talking about youth, and making sure this next iteration of future generations is aware and has that culture of consciousness change. And so I'm curious, have any of you ever done outreach work with young folks? And if so, like, what did that look like, if you're willing to share?
Okay, I'll go first. I have a 10-year-old, and I think that's when I really started to come into like my understanding of myself as a Tongva woman, was because I understood that it was so important to pass down values that weren't inherently capitalistic and, you know, empty, as which the system gives us. And I really wanted to be able to pass them down a value system and a love for the land and water. So everything that I've done, whether it be the documentary work, is really for the future generation, is really to leave something to my son, to the next generation, to learn about water from an Indigenous perspective. Because here in LA, it's hard pressed to even learn about water in general, which is a sad thing, but it's also an opportunity to create materials that are indigenously led. And that's why, like I was talking about before, I have prioritized curriculum development and working with different sects and different organizations who want to build youth programs that integrate Indigenous knowledge into their programming. So I have done that work, and it's very fulfilling, and it's very exciting to see that young people get it, and they see they grow up in the climate catastrophe, and they understand the need to change the way in which we're thinking. And when you're younger, you're more able to understand how interconnected everything is, as opposed to when you're older and you're working several jobs and you're tired. So this is the time where we really got to take our youth under our wing and teach them what we know. So.
Thank you for that. Anything to add, Dr. Middleton or Sarah?
I think a lot of my work with youth is more with students and in Native American studies and with ecology, creating respectful learning opportunities like our Keepers of the Flame initiative and students working on different projects, or really just hearing directly from elders and community members who are willing to speak to our classes. I think that I've seen that be transformative for people. And I'm also the mother of a 9-year-old. So some of my work has been advising educators of the elementary school population about what to include or what not to include in curriculum, so as not to perpetuate stereotypes and issues. We're dealing with that now in the 4th grade curriculum, but there have been some positive changes made, and there's also some really good examples of tribally developed curriculums out there as well.
At Big Valley, we have a Boys and Girls Club, and also some education programs, library programs. So we've been able to interact with, or in the last several years, we've been interacting through those programs and the Boys and Girls Club to do youth outreach, because that's how our administrator would like us to do things. So it kind of keeps things more organized and, I guess, tracked. So it's wonderful to do that, because we already have sort of built in people who will give us the limitations of the timing, and it's always wonderful. And we've come up with a variety of things based on what the children and the youth want to do. It's mostly younger ones. It is really, I found it hard to keep the interest of the older ones who are in their early teens and mid-teens. So the younger ones are if one of the things that we've done is do shoreline walks where we can look at the plants and animals and talk about the things that they're seeing. As I mentioned, we've pulled out the microscope so they can look at it under the microscope, and a lot of people like that. There's just been, we had one youth who wanted to basically start taking pictures of all the Big Valley birds and all the Big Valley fish and all the Big Valley plants. And so our hope is to make some posters based on that, with that youth led the things that are important to them. And so it's really just about listening and finding ways to finding the things that they're interested in. And technology is a big one. So we're hoping to have some apps and some tools that are things that they want to use. And we're working with them to develop that. But listening is a big one, most important.
Thank you very much. And there sounds like there's a lot of parallels with working with youth and also working with tribal ecological knowledge. It all seemed to be rooted in just learning how to take a step back and listen. Appreciate all of you for your responses. I'm going to pass the baton back over to Angela.
Thanks, Robin. Great questions. I did want to have a question. I think, Dr. Bethan, you may know this, but is there, it's been like 20 years since I graduated high school, and then after undergrad at Sac State after that, but is there a requirement that you know of, at least for UCs and maybe not CSU, but in California, to include their, like, for general ed requirements for undergraduates, for ethnic study or North American studies or indigenous studies? Is there a requirement for that, or is that still just part of it's a suite of classes that one could choose, but they have an options of classes that they could choose from, and from that required panel? Is that still the case?
So from what I know, we have these general ed requirements, and one category is domestic diversity, but that can be met through a whole variety of classes, certainly not limited to the different ethnic studies departments. So that is an issue. We have been advocating for a requirement to take an ethnic studies course, and we don't have an ethnic studies department, we have different individual departments, or a requirement to take a Native American studies course. Now we're really working with Pam Gonzalez and others to develop this Homeland History course and require some element of that be taught all across the university so that people know where they are and the history of this place. But as of right now, not yet required.
We'll let CalSF know if there's any way we can write a letter or do anything or support. We definitely would throw our support and SFS behind that, because it's all related, as we talked about, today throughout the panel and the session, and it would be a great asset to be able to have that and be a requirement. And I know we were talking about youth before undergrad, but having that, requirement at the undergrad level, I think would obviously be super beneficial and totally get at things that we're talking about today. I did want to let you guys know that before we had this session, I reached out to SFS Parent Society and did ask them. I wanted to ask them some information about what they're doing with either TEK or how do they track their tribal membership. Just because it's something, as a chapter, we're trying to reach out. We want to have more diversity, inclusivity. We want to be welcoming, and we want to create a membership that's everybody and made for everybody. And so starting that to see what's available from the national SFS was one of the things that I wanted to figure out, what they've been doing, what's been successful, and what our general membership makeup is. They're still getting me the data. So it's something that hasn't been tracked well. So that's something automatically that we know, but also that they reached out to me to let me know that they are leading an SFS program to create long-term partnerships with tribal colleges. So it's a strong vision right now, and they're developing that, but that's something that SFS is doing. And I am going to reach out for them and see if there's a place that CalSFS can play in that, because I think that was one of our goals as a chapter, is we wanted to incorporate more tribal members into our chapter. And so that might be an avenue to do that, so we can get those voices in the chapter as we're moving forward. So I also have, I think there was a couple questions. Oh, and then if you guys have been noticing, Anna, thank you, Anna, has been posting links into the chat for all the folks. So like, for example, Sarah Ryan talked about the Kalihi Tribal Advisory Committee, and the link is provided in the chat for that. So as panelists have been mentioning programs, or there's programs at the state that are relevant, Anna has been putting those in the chat. So thank you, Anna, for doing that. And she also posted the recent requirement for California high schools. And did you look at that, Anna? Does that have any sort of ethnic studies or Native American requirements?
Hey, y'all. Yeah, so that was signed, I think, earlier last month, to require all California high schools to offer and have ethnic studies as a graduation requirement. So relatively recent, it's not implemented yet by any means, but just it was relevant to the conversation. I was trying to do it quietly, so I didn't take up time, Angela.
Sorry, we had a little bit of time. I had to spot you out, so thank you. Anna's great. She's behind the scenes. She's been keeping up with the conversation, adding stuff, so I had to highlight her and put the spotlight a little bit there, but that's great. So thanks for the information. That's good to know. We did have one question, more like a comment, I guess. Somebody wanted to make sure that it was mentioned, and it must have been by something that Sarah, you pointed on, that early coordination, collaboration, consultation is the best method to establish and develop good, respectful working relationships with tribal governments, communities, organizations, and groups. And this can include scheduling to attend, present at annual, quarterly tribal council meetings. And this is especially true of state, local educational facilities, potentially of projects within those tribal jurisdictions currently or in the future. But early is the key here for getting those relationships established and transparency, communication, and what folks and agencies are doing. So I don't know, Sarah, if you wanted to touch on any of that. It was looks like it was mentioned something that you had talked about.
Yeah, that's such a good point that Carolyn made. And, you know, it's just like doing an environmental review. If you do it at the very end when everything's been decided that this is how the project's going to go, then what's really the point of doing an environmental review to some extent? Tribes should be a part of the planning of programs that are going to impact them. I think that the Governor Brown's B1011 declaration requires that anything that is going to impact tribes, that they should have meaningful input and an ability to help develop that and to help be a part of it. Otherwise, it's going to be all reaction. And reaction is really, you know, it's expensive in terms of people's energy and their time, and it's exhausting. So it's much better, and you get so much better of a program if tribes are engaged from the get-go. What are their issues? What are the things that they're seeing? How do they, you know, what have they seen change? What's being impacted? Those are the things that I think any you know, if you're managing land and water, you should be thinking about those things. If you're doing research, I think it was brought up earlier. Why are you doing that research? What is it that tribes can get from that? What are the things that they want to say about that? You will just have a much better, more enhanced, more robust, enriched end product if tribes are engaged from the get-go, and just do your homework to find out which tribes those are. And it might not be, you know, if you look in the phone book or you look online, you might not see a tribal office in your county, but as you know, in the map that I showed earlier, they're everywhere. There are Native peoples all, you know, throughout California who have a claim and an interest and a concern about every square inch of this place. So be engaged and do your homework. Get them involved if they want to.
Great, thank you. Did any of the other panelists or fellows want to comment on that? Anything else? Okay, so I have one more question. This is left over from earlier from Sarah's, and this will be kind of like the last question. It's a little different, but it's for each of you guys. And the question is, which water pollutant is most significantly affecting traditional cultural subsistence of California tribes? And I bring it up because we have kind of a regional diverse panel here. So we have somebody, you know, we have a student, we have a educator, professor, and we have somebody that's working on, you know, implementation management here. So I like the diversity. And so I think the different perspectives of answers to this, and I know it's like one pollutant, there's like so many, and maybe there's, you know, ignorance is a pollutant. I don't know. But so, you know, I want to just kind of explore this a little bit as our last question for this panel. And yeah, so let's see what you guys got for this one.
Well, I'm happy to jump into that one. So it is hard to narrow it down to one, and I, you know, in Clear Lake, we're looking at 4. We're looking at bacteria, so E. coli that's impacting people, and that's coming from all sorts of sources. It's coming from septic systems. It's coming from sanitary sewer overflows. It's coming from runoff from ranches and agriculture that has manure for fertilizers, and houseboats. So that's one. Cyanotoxins, which is, you know, the result of nutrient enrichment of a water body, which is coming from multiple sources. And let's see, Mercury. Mercury. So I understand that there are no water bodies in California where you can safely consume a traditional level of fish meals for a week. Like the mercury levels are so high, and the methylmercury levels in the fish are so high that there's my understanding, there's no places where you can eat 3 or 4 fish meals a week, and that's not even subsistence consumption there. So you can't not talk about mercury pollution and methylmercury in the fish when you talk about pollution. So I think those and it really has to do with land management and land management decisions. So I would say those on Clear Lake, and I suspect those would be the case in many other places as well.
Thank you. So being more of a social scientist, I don't know if I can name a specific pollutant. I have spent a lot of time studying infrastructure, though, and the imposition of infrastructure on tribal homelands and waters. So I'm thinking of dams in particular, and how they exacerbate the impact of any existing pollutant. Dams as well as diversions. Because they lower the water level, which can lead to increased water temperature, which can lead to higher and concentrations of various pollutants, and a heightened impact on human health. So I'm going to recognize Joe Hostler here from Yurok tribe. I was talking with him about climate change once, and he said, well, how can we talk about climate change until we talk about these dams on the system and their impact on the river overall? So whether it's a pollutant or broader systemic change, I think the infrastructure plays a key role.
I definitely agree with Dr. Middleton about how infrastructure plays a key role out here in LA. Most of the river is channelized, so we don't even depend on the river for any type of drinking water. So, I mean, I can't speak to specific pollutants, but I will say that we are completely dependent on imported water. So we definitely need to clean up our own aquifers and become dependent on our local sources. And there is measures and money to start doing that kind of stormwater capture work. And, you know, whether tribes will be involved is something that, you know, will be documented and what our tribal community will push for. But as of right now, our water is polluted to the point where it is not useful to anybody.
Thank you for those responses. Did any of our fellows want to add anything to that before we wrap it up? And I did just do a call out, last chance for any of the attendees to ask any questions of the panels before we finish for the day. No, we're good. All right. Okay, so I think that's it. Thank you so much to our panelists and speakers. Thank you, Ada Stevenson, too. I'm sorry she couldn't fully participate. Her information she provided was wonderful and valuable. And thank you, Ada. Let her know for us, Dr. Beth, too. And is there any last parting words from our panelists today? Good. Okay, you guys said a lot. Thank you so much. Oh, Sarah, yes.
I just want to thank you, Angela, for putting this session together, and it was great to see my fellow panelists. And a shout out to Ada, who I know is not on the call, but she and I go way back, and she does great work. So just really great to hear her voice. And thank you for this.
Good. Thank you so much. So this will be this is recorded, and it will be available on the Water Board YouTube channel. We also like to extract it and put it on our website or embed a link. And we have a website, we have our own standalone website, and we also have one on SFS. And then if there's anybody interested in partnering with tribes, Anna did put a link in the chat for the Office of Public Participation, Tribal Affairs. So that's a good step if you want to reach out and explore the information at that link, and then share this panel with anybody also that you think might find it valuable and have information for you. And then again, thank you so much to the panelists for spending this time on a Wednesday to come and talk with us. It was a great discussion. I really appreciate you, all that you're doing and your input. And thank you, fellows, for moderating. You did a great job. And thanks so much for contributing to CalSFS. Thanks to all the attendees. I'm not sure if Allie wanted to get on and say anything. We're ending like a little early. But if you want to say anything for the parting of CABW, or what do we have after? Just want to check with the.
I can't even get I'm trying to come back. Sorry, hold on.
It's been a long time since you've been gone.
I'm here. Okay. Yeah, well, thank you all. Everybody's expressed a lot of gratitude, so I don't want to keep you all too much, but thank you all very much. Appreciate your time. That was a great discussion. We will be sending out a survey. Just let us know how this went, get feedback from everybody. I think Shuka's putting it in the chat, and then we'll also distribute that through email lists and all that good stuff. And then, yeah, the recording will be available. We'll do what we do and send a bunch of emails to you all to let you know where to find that information. And I think that's it as far as logistics. So again, thank you everybody for another great CABW SFS, and look forward to the in-person Thule boat demo next year, hopefully.
Can't wait.
It's gonna happen.
Yes.
All right, thanks everybody.
Thank you guys.